नमस्कार नीरज सत्यदेव।
सर नमस्कार सर।
नमस्कार मंजू शर्मा जी।
नमस्कार नीरज।
नमस्कार नमस्कार।
नीरज तुम लोग चाय पी रहे हो? ये तो ठीक नहीं है।
वाईफाई के थ्रू भेजो चाय।
तो जी नमस्कार। आपको दिखाई नहीं दे रहा है?
अभी दिखाई दे रहा है। हो नहीं रहा है। मैडम
हाँ नमस्कार नमस्कार मित्तल जी।
नमस्कार सर। मैडम
हाँ बोलिए।
कल प्रोफेसर कस्तूरी रंगन सर आपसे बात करेंगे आज।
हाँ हाँ।
वो शायद व्यस्त हैं, प्राइम मिनिस्टर के साथ कोई मीटिंग है।
क्या चीज? कौन?
प्रोफेसर कस्तूरी रंगन जी।
अच्छा।
वो गैलरी के
वहाँ कावेरी में नहीं जा पा रहे हैं।
वो नहीं जा पा रहे। आपसे बात करेंगे आज।
अच्छा ठीक है। नमस्कार डॉक्टर साहब।
सर नमस्कार।
तो अशोक
मिश्रा जी भी आ गए हैं सर। तो
अशोक मिश्रा जी से अशोक मिश्रा जी आप जरा चले जाइए वहाँ मैसूर 3 तारीख को। हाँ
सर म्यूटेड है सर।
आप म्यूटेड है।
3 तारीख। हेलो हाउ आर यू? 3 तारीख को मेरी यहाँ पे मीटिंग्स है 2-3। अगर
हाँ
2 को होता है 4 को होता तो हो जाता, पर 3 को जरा मुश्किल है। देखता हूँ मैं टाइम।
देख ली। एनीवे नीरज प्रोफेसर रंगराजन और प्रोफेसर पद्मनाभन दोनों से बात हुई।
पद्मनाभन सर से बात हो गई है और होपफुली सर आ गए। नमस्ते सर। होपफुली शायद सर विल ट्राई।
हाँ तो प्रेसिडेंट से बात कर लेंगे उसके बारे में। हाँ मीटिंग के बाद जो है वो। नीरज
जी मैडम।
तो हम लोग शुरू करेंगे ठीक 10 बजे के। अभी और लोग ज्वाइन करेंगे कुछ लोग। हाँ
10 बजे ठीक शुरू करते हैं।
नहीं तो
कई लोग तो उसमें ज्वाइन करेंगे ना यूट्यूब में।
प्रोफेसर पद्मनाभन should we start or wait for some time?
अब 5 तक कर लीजिए, 5 मिनट में।
हाँ
5 मिनट बस 5 मिनट बाद शुरू करते हैं।
ठीक है।
Many people would have joined by YouTube. Many people would have joined by YouTube. So let us start on time.
Yes sir.
Professor Padmanabhan, Balram has got a parliamentary committee meeting he has been called for. I can't hear you.
Sir, you have to unmute yourself.
Adi, can you hear me now?
Yeah, we can hear you.
Yes.
I saw the mail.
Professor Balram Bhargav has gone for a parliamentary committee meeting.
Right.
So we don't know his schedule because the meeting they have not given any schedule when they will finish. So he had to go there. The parliamentary committee, nobody can say no.
That's right.
Yeah. So, Neeraj, what did his office say? Tell you?
Actually, already I think the office has joined. TGICMR, you might be seeing on the screen. Office has joined, but there is nothing certain about when sir will join. So we are waiting actually, madam. whenever Professor Balram Bhargavaji is joined,
Okay.
but his office has joined. DGICMR, you may see, has joined.
All right. So can we start now?
Just 3 minutes after.
2 minutes. Yes.
Sir, we have a very tight program. We have to go till 5:36.
Yeah.
And we will have 10 minutes lunch break.
Half an hour lunch break.
Half an hour is too much. 15 minutes is enough. Because all of us can start eating here, get our food on the table.
Sandwiches.
Sandwiches is a very good idea, yeah.
Yeah.
And Dr. Jaipal Mittal will provide us sandwiches.
Online.
Yeah, online.
संतोष कहाँ है?
संतोष ने नहीं ज्वाइन किया क्या भाई?
ज्वाइन किया संतोष ने, लेकिन मतलब यहाँ नहीं बैठा है.
हाँ, परम, how are you?
Morning, everyone.
Good morning, परम.
नमस्ते मैडम. नमस्ते मैडम.
ठीक है सब?
गुड मॉर्निंग
जी मैम, ठीक
मैडम.
है.
हमको बस आप भी कह दिया करिए कि सब ठीक है तो.
हो गया मैम. नमस्ते मैडम. नमस्ते मैडम. सर नमस्ते सर.
नमस्ते Neeraj, I think we should.
So we welcome everybody. Annual session of the National Academy of Sciences India, the first science academy of this.
आपकी तबीयत कैसी है? Are you okay? Dr. Parker, are you okay?
We are now starting the 90th annual session program as well as the symposium on towards the new healthcare regime for the nation. This program is for 3 days, starting from today and ending on 27th February 2021. So we feel proud and privileged to inform everybody who have joined while welcoming them from the core of our heart that this is the oldest science academy of India, completing 90 years of its successful existence. And this academy was founded as the first science academy because Professor Meghna Saha was not only an eminent scientist and the world famous scientist at that very time, but also a very prominent figure for the social reforms. So, with the idea to make the country move towards cultural improvement, this academy was founded for scientific endeavors to make this country free from many orthodox and call all the scientists, Indian scientists at that very time, on a single platform to provide solutions, scientific solutions to the problems of our country. So, first of all, we think to pay our regards, homage to Professor Meghna Saha, who was the first president of this and the founder of this academy. We are very much grateful to such a pious soul who contributed a lot to the development of this country. Regarding this symposium, the idea of this symposium was discussed in the council and it was basically mooted by respected Madam Professor Manju Sharma ji. When the whole country was struggling with the pandemic, COVID-19, it was decided and discussed, seeing the scenario of last few years, that several pandemics came and several communicable diseases spread in our country, that we should have a new healthcare regime. The details will be discussed later on in the symposium. But now we wish to start, but with one note, from 1930 to this date, 2021, a single thing which I would like to quote here that makes the clear sense of accomplishment for all of us, where we can have pride that now, today in the newspaper cities, that USA wants to have collaboration with India for signing MOU for the health and biomedical research. So this is the advancement of our country from 1930 to this date, that the so much developed countries now they want to make their collaborations with it. So with this, we start the symposium, and I would like to request our General Secretary, Professor Satyadev sir, who was Honorable Vice Chancellor of APS University, Riva, and presently he is the General Secretary of NASI headquarters as well as Emeritus Professor of Mathematics in the Harishchandra Research Institute of Mathematics and Mathematical Physics, Allahabad, which is under the Department of Atomic Energy. He is the recipient of many awards, including Indian Science Congress Gold Medal, MP Single Memorial by the Prime Minister of India, elected Academic Secretary, Indian Mathematical Society, Distinguished Service Award by the Mathematical Association of India for his outstanding contributions to the cause of teaching and research in mathematics. With these few words, there are many things to tell about sir, but time is short. So with these few words, I would like to request humbly Professor Satyadev sir to please welcome formally to everyone.
Good morning, everybody, and welcome to the 90th annual session of the National Academy of Sciences, India. As you know, this is an extraordinary annual session of NASI being held in the month of February rather than in November and December, which used to be the case earlier. This is because of the COVID-19 pandemic, which has affected the entire world, not only us in India. It's my pleasure to welcome all of you in this inaugural function of 90th annual session. First of all, I would like to welcome our President, Professor G.P. Padmanabhan, former Director, IISc Bangalore, who has been guiding us for the last 2 years in all our activities. He has been a constant source of encouragement to the Academy, whether it was presiding over the council meetings, presiding over the webinars conducted by the Academy, or by the local chapters, or it was celebration of the other important occasions of the Academy, or even correcting the drafts for which we have been sending the letters to the various government bodies. So we welcome our President, Professor Padmanabhan. I would like to welcome Madam Dr. Manju Sharma, former Secretary, Department of Biotechnology, Government of India, and the past President of the Academy. Year after year, she has accepted the request of the NASI Council to organize a symposium on an important topic of science during the annual session. This year, the theme is, as Dr. Neil Kumar has mentioned, the theme is towards a new healthcare regime for the nation. I have no hesitation in saying that she is like an engine of almost all the activities of the Academy and has been constantly working on the new initiatives of the Academy. Whenever she takes up a project, she implements it with perfection, devoting all the energy that she has. I would like to welcome all the invited speakers of the symposia who are going to talk about important subject matters pertaining to the contemporary science. I am very happy to welcome Professor Balram Bhargav, Director General of ICMR and Secretary to the Government of India, who will be giving the inaugural address in this session. I cannot help mentioning the fact that all the talks given by Professor Bhargava are highly admired by one and all, simply because he talks really like a very competent Secretary of the Department of Health with facts and figures relating to our country. We get a very good picture of what is going on at the government level in health sciences. There are two main parts of this session. One is the symposium and the other is the annual session of the physical as well as the biological sciences. I welcome the Sectional President of Physical Sciences, Professor Shank Mitra Bandyopadhyay, Director ISI Kolkata, and the Sectional President of Biological Sciences, Professor Smita Mahale, Director of National Institute of Research in Reproductive Health, Bombay. Both of these sectional presidents have worked very hard to get excellent speakers for invited talks in their sections, which we will have occasion to hear later on. I welcome all the fellows and members of the Academy who are online at this moment. I also welcome all the past presidents of the Academy who have found time to be present on this occasion. I welcome all the award winners who will receive their awards and give talks on their research during this session. And I also welcome all the chairs and co-chairs of various sessions. Thank you very much.
Thank you, sir, for a warm welcome of all the fellows, members, council members, past presidents and presidents, and the participants in this program. We are really thankful to you, sir. Now we move to the second part, that is the genesis of the program. Genesis of the program, as I already informed, the idea was mooted by Madam Manju Sharma ji. So I would like to request her, but before that, although she doesn't need any introduction, but in brief, it is my duty to inform because there are several other participants connected with this program from India and abroad. So I would like to introduce Madam. She played a significant role in establishing several institutions in the country. She was the president of the National Academy of Sciences India and also the Indian Science Congress Association. She is well recognized for her manifold significant contributions, particularly for the welfare of women who regard her as a role model. Here I would like to say that this academy also regards her as the icon because she is the source of inspiration for many of our programs. And she has led this academy for last about 30 years. She earlier served as the Secretary Department of Biotechnology in the Ministry of Science and Technology, Government of India, and was most recently the President and Executive Director at the Indian Institute of Advanced Research in Gandhinagar, Gujarat. She is still very active as the advisor, NASI new initiatives, and also as the NASI DST Women Scientist Chair Professor. She is Padam Bhushan and she has also received many awards. I cannot tell all this because it will take another hour of lecture. So with these few words, I request Honorable Madam to say something about the genesis of the program as well as the 90th anniversary of this academy.
Thank you, Neeraj, and namaskar to everybody, our Honorable President, NASI, and all the past presidents, and the distinguished guests who have joined us for this webinar. We were going to celebrate 90 years of Academy on the 4th December when we were supposed to organize this annual session. However, as all of you know, due to unfortunate circumstances, it got postponed. Just to refresh the memories of all very, very distinguished experts who are here, I just want to say a few things about the Academy, which has completed 90 years. 1930, this Academy was established, and it is one of the oldest science academies in the country. In the inaugural session, the first president, the founder president of this academy, Professor Meghnad Saha, made a very sort of interesting remark for the academy. And I just want to say, I won't read his whole quotation. Everybody knows that, and Neeraj may be projecting it. But the most important point, what he said, but the main function of the academy should be towards cultural improvement by contributions to human knowledge. This was the last part of the sentence. On another occasion, when he was addressing a group of scientists, Dr. Meghnad Saha said, scientists are often accused of living in the ivory tower and not troubling their mind with realities. And apart from my association with political movements in my juvenile years, I had lived in ivory tower up to 1930. He says himself, but science and technology are as important for administration nowadays as law and order. I have gradually glided into politics because I wanted to be of some use to the country in my own humble way, unquote. And when Professor Meghnad Saha established the academy, a very interesting comment came from Professor Albert Einstein. And he wrote, Professor Meghnad M.N. Saha, to whom physics, particularly astrophysics, is indebted for many valuable contributions, has reported to me about the Academy of Sciences, which has been called into existence at Allahabad with a high object. So that was a very encouraging comment from Albert Einstein. The Academy today has progressed very rapidly since then. And today we have more than 1,800 fellows and about 1,800 members. Membership is a very unique feature of this academy. The mandate given to the academy was science and society. And I'm very, very happy to say that the academy has really worked very hard to implement, to develop many programs of relevance to the society. And these programs have had very significant impact all over the country, in different parts of the country. Every year, as Dr. Sadhdev said, we have been organizing the annual session along with a symposium on a subject of national importance and human welfare. This year, the council suggested the present symposium, which is towards a new healthcare regime for the nation. And after the approval of the council for the title of the symposium, I had very detailed discussions with Professor Balram Bhargava, and the present program, which is given in the, which is circulated, was developed in consultation with Professor Balram Bhargava and also a small committee which the Honorable President had constituted. However, before I say something about the symposium, there are two more issues which we have to, I want to place before the August audience. One is that the COVID-19 pandemic completely changed the entire scenario of various programs, activities of the academy, etc., etc. But in spite of that, in spite of this very drastic change, people could not travel and we had to only meet online. The academy did not just stop its working. It organized more than 20 webinars in 1 year on nutrition, a subject which is very important for the country and which was announced as a Poshan Ma by the Honorable Prime Minister in September 2020. The second subject, which all of you know, the pandemic COVID-19, when it was there and how we felt about it and how the Honorable Prime Minister thought that this particular pandemic, we have to create awareness amongst the people of this country. Jagarukta Abhiyan. People must know how to prevent, how to go about the appropriate behavior of the people. And immediately NASI thought that we should organize a large number of webinars on this subject, Jagarukta Abhiyan. We have had more than 16 webinars in different regions of the country. And I get a feedback that there has been a very, very good response to all these webinars. Hundreds of scientists and medical professionals, they have joined the seminar, and it has been a great pleasure to know that with the approval of the president, the webinars which were organized, they have really benefited to some extent. The main preventive measures for the COVID-19, that is wearing a mask, washing hands, and keeping social distancing. So we did during this year, in spite of all the hurdles which we faced, the many activities around the country. And I thank many of the conveners and chairmen of the local chapters have joined them very quickly, as they have been very, very cooperative.
Neeraj, you need to mute the.
They have been very, very cooperative, and they have joined us in organizing these webinars, both on nutrition and we are going to discuss many important areas, and we start with the epidemiology and then drug discovery, all related to COVID-19, also drug discovery for malaria parasite, cancer, many infectious diseases, brain research, public health emergencies, diabetes, enabling ecosystem for biomedical research, development of therapeutics, Ayurveda innovations, etc., etc. And in all these areas, we need to do cutting edge research. We need to do translational research, and also they have to be product and technology oriented. It has been demonstrated that given a task during a pandemic, we, the scientific community of the country, the doctors, medical professionals, and also the industry can do a lot, and they have one objective, that is human welfare. And we have seen during this last 1 year that how the scientists have come forward, how the industry has joined hands, and all the science departments, different ministries, concerned ministries, they have played a pivotal role in controlling the pandemic situation. And today, India stands on a very strong footing in the world on the prevention and control of COVID-19. So I want to thank all very distinguished speakers who have agreed to our request to be a part of this symposium. And I also want to thank all the chairmen and co-chairmen who have readily agreed to chair the sessions. With these words, I want to stop here, and I will request Padmanabhan, our president, to give his presidential address. And maybe Neeraj would like to say a few words about our president, about who does not need any introduction. The whole country, everybody internationally and nationally knows who Professor G. Padmanabhan is. So, sir, after Neeraj says something, we request you to give your presidential address. Thank you. Neeraj?
Neeraj?
Neeraj, please. We can't hear you. Something wrong with
There's some audio.
system with Neeraj? Huh? Neeraj?
We are not able to hear.
I don't know, there's something wrong with their system.
Can you hear me, madam?
Yeah.
Now
Can
we
you hear
can.
me?
Yeah,
You,
please.
Neeraj, I request you to
Okay.
kindly say a few words
Yeah.
about.
Actually, Honorable Professor Padmanabhan sir, who is the president of this Older Science Academy, was the director earlier of Indian Institute of Science, a premier scientific institute of India at Bangalore. He was instrumental in ushering in recombinant DNA technology in the country and worked closely with several government agencies to further the cause of biotechnology in the country. He helped and promoted vaccine industry in this country, and also he is well known for showing the anti-malarial property of curcumin and its efficacy in combination therapy in 2004. Professor Padmanabhan sir is the recipient of several prestigious awards, including the Padam Bhushan and Shanti Swarup Bhatnagar Prize. During his tenure as the president, the Academy has touched several milestones, and we are very much grateful to Professor Padmanabhan sir. Sir, please say a few words as the presidential remarks.
Thank you, Neeraj. Santosh, you have the slide? Yeah. Now, it's not going to be a few words. This is perhaps my last address as president on a technical session. And Professor Manju Sharma has given the brief history, and I will have occasion to come back addressing the AGM. But I thought, I will use this occasion to talk about science. The science I have done, 60 years of science at IISc. Next slide. I joined this Department of Biochemistry, Institute of Science, in January 1961. And that makes it 60 years and 1 month. Can you go back? That is Dr. P.S. Sarma, my guru. He was a well-known biochemist, worked with LVM at Wisconsin. Next slide, please. I was given 2 projects to start. One was on Kesari Dal, to find out, Kesari Dal is supposed to cause neurolathyrism. And the other one was an entirely unrelated project on heavy metal toxicities in Rospora, it's a fungus. I was successful in isolating the neurotoxin. Next slide, please. And it was characterized as oxalyl diaminopropionic acid by my colleague, S.L.N. Rao. I continued to work on its mechanism of action, neurotoxicity, showed it's a glutamate agonist, and published 2 papers in Nature. I must tell you, those days publishing in Nature was not a big deal. Only later on it became very difficult. Next slide, please. But I want to tell you, till today, we do not know whether this dal causes neurolathyrism. Although we had a toxin, internationally this toxin is known as a glutamate agonist, but the dal as such, does it cause paralysis? We do not know, and there have been associations demanding the lifting of the ban. Many states have banned the cultivation of these seeds. More recently, there have been some steps to lift the ban, at least partially. So I must tell you, this is an unsolved problem, whether the dal actually causes neurolathyrism, although it has a toxin. I'll go to the other project where I worked on the heavy metal toxicities, especially, if you work cobalt toxicity in Rospora, it produces iron deficiency. And in particular, it secretes a peptide belonging to the cytochrome series with extraordinary binding affinity for iron. I characterize this by my PhD. This is a Desferry coprogen, 10 to the 30. EDTA is 10 to the 11. So you can imagine the affinity of this molecule. This is cytochrome. It has 3N hydroxymate bond, ornithine, and hydroxyornithine. I actually want, and interestingly, the fungus will use this iron complex if you provide. It will reduce the iron to ferrous, and iron is released, and it will use it for heme synthesis. Now, that is what I wanted to do, biosynthesis of this, and my required labeled ornithine. And my grant was 5,000 rupees for a whole year. Young people around must realize what it was to work in the 60s. 5,000 rupees for a whole year. So that is how we did this study. But interestingly, we thought this very oxamine, this iron chelate, can be used in thalassemia. If you inject it, they will remove the excess iron. These children are injected with, chelators even now. We did animal studies, but unfortunately at that time, my guru, Professor Sharma, died untimely death. And I didn't know how to take this forward. Next one, please. But what happened was he became a theme for my central theme in many of my projects because the iron is donated to porphyrin and it gives rise to heme. And this is the structure of heme. We know many hemoproteins, hemoglobin, cytochromes, all of them contain heme. Next one, please. And I went to University of Chicago in 1973 as a Fulbright scholar for a whole year. And here you see Murray Rabinowitz. He had a muscular dystrophy, and his wife. And you can clearly see the commitment of this person. And I had seen him deteriorating in every one of my trips, but he still didn't give up science. There was this, Murray Rabinowitz. I went to his lab because they worked on cytochromes. Next one, please. I learned all the techniques. I visited Murray's lab 10 times, 73 to 86, picked up techniques of gene cloning, sequencing, all the recombinant DNA techniques which I was able to introduce in India. I learned at the University of Chicago. But I did not take any of my projects. I worked on any project they wanted, but I was learning only techniques. At the end of the year, I said, Fulbright scholar, I'm going back. Murray said, why do you want to go back to India? What will you do in India? I said, sir, I have a small lab. I have to build it. And if you can help me, I will build it. And, he was so generous that he said, you come to my lab anytime, any length. I went to his lab 10 times. 10 times between 73 and 86 for periods ranging from 1 year, 6 months, 2 months, 1 month, all kinds actually. I moved from the fungal system to the rodent system to the rats because I wanted to do molecular biology in animal systems, in higher organisms, because in the 60s and 70s, it was believed all the Nobel Prizes were all in prokaryotes, in E. coli, for example. And the eukaryotic system was considered as a black box. But I thought, we should get into higher organisms. So I took this liver cytochrome P450. And my question was, what is the role of heme in the synthesis, in the biosynthesis of this protein? Because we know about the structural role. Can you go back to the previous slide? Looking at the mechanism of induction of liver cytochrome P450, and that time only 2 species were known: phenobarbital inducible, methylcholanthrin inducible, 2 species. Today, I think 150 species are known in plants, in bacteria. It was known. Gonzalez, Madam Sharma knows him. He was our first director of ICGB. He worked on cytochrome P450, Pseudomonas butida. And there are 150 species known today. Now, anyway, 2 species I injected phenobarbital, and within 6 hours, I can detect the messenger RNA by translation. Next one, please. Then we did a lot of studies. I have a lot of slides, but I cut down many because I thought I shouldn't speak for more than half hour. That was the duration I told Neeraj I will take. But anyway, I'm giving you the final take of this project. How heme regulates this gene. That was the answer we proposed. That in the normal people, In the normal animal, this gene is poorly expressed or not expressed. We had identified negative element, positive element, by gel shift assays, by transcription studies with nuclear lysates and so on. And we propose that the negative element which protein binds and prevents transcription initiation, and we had evidence for that. If you inject phenobarbital, this becomes phosphorylated and it moves to the positive element. Once it moves to the positive element, everything else falls in place, including the enhancer and the transcription stage. And we could publish in PNAS. I must tell you, from India, we were able to publish at least half a dozen papers in PNAS. I am just telling you for the young people, it is very, very important to publish big, good papers from India, and it gives you a lot of pride. Next one, please. So what is the take home for all the studies we did for 20 years? Heme regulates translation of protein synthesis. Heme regulates translocation of specific proteins across membranes. I told you about ALA synthase, the first enzyme of the heme pathway, which is nuclear coded and has to be transported across the mitochondrial membrane, and heme regulates that process. Heme regulates transcription. I gave you a little glimpse of that. Heme regulates differentiation of hematopoietic cells. We didn't do anything in this area. There are other groups which we did. Finally, we wrote a review. Heme is a multifunctional cellular regulator, which was published in Trends in Biochemical Sciences in '89. Actually, I had called heme as a second messenger, but the editor said, I'm very happy with your review, but I will not accept you to call it as a second messenger. I will allow you to call it as multifunctional cellular regulator. So I accepted it. I was desperate to publish it anyway. Next slide, please. So this is time, this is early 80s. I have spent 20 years in this area. And then that was the time Astra came to Bangalore, which became AstraZeneca. For some reason, they came to me and said, will you help us set up an institute, Astra Research Institute? And they had to get permission of the government, and it took 2 years for them to get permission. But in the meanwhile, they started a research as a project mode in my lab at the Indian Institute of Science in the Department of Biochemistry. And I started 4 projects for them: malaria, tuberculosis, cysticercosis, and diarrheal diseases. And I recruited half a dozen of our own department and institute students who had finished PhD and were going abroad. I said, this is a new challenge. Why don't you take it up rather than going abroad and working as a postdoc? So some of them agreed, and 6 of them. And malaria was started in that mode. And it was the early 80s. And the Astra worked in my lab for 2 years. We recruited the first employee of Astra Research. And I must also thank Astra. For 10 years, they supported me, my research. And I thought, why should I do malaria research only for Astra? Why not do it myself? What am I doing working on all other things? So I should do something in organism or a system which is useful to the society. That is how I started malaria research. And there was a heme connection. I will tell you what is that heme connection. Next one, please. For those who are not in the field, how is malaria caused and the cycles, I want to very briefly tell you. The mosquito injects sporozoites, very quickly goes into the liver directly, and several hours, you get merozoites, which will infect red blood cells. And you will get asexual cycle ensuing, the ring stage, the prophocyte stage, and the cells will lyse, and the parasite will be released and infect fresh cells. That is where you get the chill and the fever. But in all the process, it also generates a precursor for the sexual form, that's the gametocytes. Now this goes back into the mosquito when it bites through another channel. So in the gut, you have macrogametes, microgametes, they form oocyanides, oocysts, and then sporozoites. So the sexual cycle is in the mosquito, the asexual cycle is in the red blood cell. This is in brief the life cycle of the parasite. And you have simple malaria, which can be cured. Basically, as I said, it only lyses the cell, anemia will be caused, and of course, toxins are released and so on. But drugs which work, especially artemisinin derivative is the only one which works now, even though resistance is spreading, binds heme actually. Chloroquine binds heme. Because what happens is the parasite is living in a sea of hemoglobin. And all the hemoglobin is taken up by the parasite and then taken up to degrade it in the food vacuole. The parasite degrades the hemoglobin in the food vacuole and gets tons of heme. And the amino acids generated, it uses for its own protein synthesis. So parasite doesn't, many of you biochemists would know heme is made into bilirubin, biliverdin, and bilirubin. Parasite doesn't have that system. Parasite will simply polymerize the heme into hemozoin, a polymer, which is inert so far as parasite is concerned, but is very toxic to human. I'll come to it later. Now, this is the simple malaria which is curable by, say, a drug like chloroquine till it become resistant, because it will bind heme and prevent hemozoin formation. Heme will accumulate and kill the parasite. But then you have issues when Plasmodium falciparum, as you know, there are 5 species of Plasmodium are important. 2 of them are most important, accounting for 95% of the malaria, falciparum and vivax. These 2 species, for example, are almost 50-50 in India. Falciparum in particular, all the mortality, I should say, is due to falciparum. Vivax causes morbidity and huge problems, although death may not be as high. So what does falciparum do? One of the issues with falciparum is the RBC, infected RBC reaches the brain and the blood-brain barrier is broken and leukocytes enter in and all kinds of complications, and you can get cerebral malaria, as well as organ failure if other organs are also affected. It can also have this problem, placental lining it can prevent, and therefore pregnant women has problem. Malaria is, the birth weight, the fetus, growth of the fetus is seriously affected. Next one, please. So this is briefly the life cycle of the parasite. Parasite is in a sea of hemoglobin. But I asked this question, does it make heme? It rather looked like a stupid question to ask, because it has so much heme coming from hemoglobin. Why should it make heme? That is the answer you will see at the end. Next one. We were, interestingly, previous slide, we showed that the parasite can make heme. And it was, people were skeptical in the beginning because, as I said, why should it make heme when it can get so much heme? But in early 2000, the falciparum genome was available. Entire sequence was available. And then it was found all the putative heme biosynthetic pathway genes are present. Therefore, we were vindicated. And so we got into it great vigor, cloned every gene of the pathway, and very interesting pathway in the parasite. I don't have time to get into, except to say, the first step is in the mitochondrial ALA synthase. Then the intermediate goes into a thing called as apicoplast. The parasite has apicoplast, which is a rudiment of chloroplast. Very interesting. And then it comes out, and in the cytoplasm, there is one step. And the final step, again, back in mitochondria. The point you need to remember, the first enzyme and the last enzyme are in mitochondria. And, there are 3 compartments involved in heme biosynthetic pathway. And it took about 15 years, and we have several publications. Next one, please. Next slide. Then, we were followed by many groups all over the globe, McFadden, Roos, many groups are followed. And this question has been bothering me. That means, you have RBC on the left, you have reticulocyte on the right. This is very important to remember. Falciparum will infect all blood stages. But reticulocyte is preferred by vivax, also by the animal model, Plasmodium berghii in the rat model, and the mouse model prefers reticulocyte. No, there's a difference between RBC and reticulocyte, as you know. RBC is a bag of enzyme, basically. But even that was shown to have part of the heme pathway, if not complete. It can't have the first step, it can't have the last step, but it will make ALA probably imported from the host, and then it will make porphyrin, protoporphyrin. It has to be imported into the parasite and use ferrochelatase to form heme. Whereas reticulocyte can make completely heme, it has mitochondrion. It has the first enzyme ALA synthase, last enzyme ferrochelatase. It can make heme and import. Then we had shown parasite makes heme. And we know heme comes from hemoglobin. So much heme. So we were wondering this question, why does the parasite have so many mechanisms to have heme? Biosynthesis, hemoglobin, heme partly made by RBC or reticulocyte. Next one. We thought maybe the heme is not required for the blood stage. Maybe it is required. Why does the parasite have so many routes to acquire heme while it is getting tons from host hemoglobin? This is the question. Next one. We thought it may be required for parasite development in the mosquito. It may be required for the sexual stage. So what we did was to make out knockout parasites. Knockout means the first gene, ALA synthase gene is knocked out here. These are all classical methods by which you can make out knockout parasites. We can also make knockout parasite of the last gene. So we have wild type parasite, you have a knockout, first gene knockout, you have a last gene of the pathway knockout, and these are all proof, northern, western, and clearly you can see the western here. In the knockout wild type, you see ALAS protein, you don't see it in the knockout. Similarly, ferrochelatase you see in the wild type, you don't see it in the knockout. This is just a proof. But all of them grew well in the mouse. If you inject in the mouse, there may be a small lag, but all of them grew wild type knockouts. All of them grew. Next one. So it was concluded, with knockout parasites. Next slide. With knockout parasites that heme is not required for the blood stage. This is what everyone was claiming, quoting us. Can you go to the next slide? Well, to cut the story short, in the mosquito, as I said, it forms ookinase, oocysts. Ookinase was not affected. Oocysts were affected. I am not presenting data short of time. But in the case of final sporozoites, these are the sporozoites in the wild type. You don't see sporozoites in the ALAS knockout. Similarly, these are the sporozoites. You don't see sporozoites in the FC knockout. That means if the parasite cannot make heme, it cannot develop in the mosquito. So next one, please. This was a major publication which came out from our lab showing that the parasite requires heme to develop in the mosquito. We published it in PLOS Pathogens, and this was very widely quoted because we were the first to demonstrate why does the parasite make heme. So it is growing in, it can grow in the mosquito. Next one, please. But still this question, why would it make heme in the blood stage, waiting for mosquito stage? Next one. Why does the parasite make heme in the blood stage? It cannot be in waiting for the mosquito stage. Finally, we have an answer. This is a 20-year-old enigmatic question, and we have the answer. And we have unpublished data. I will very briefly give you that. Next one. We have now shown that the parasite makes heme to cause virulence. It is necessary for its virulence. And we have used the animal model, C57 black. If you inject Anka strain, it will produce cerebral malaria. It will not produce it in biopsy mice. For example, what happens, clearly you can see the blood-brain barrier breakage in the C57 black. This is the wild type, and this is the biopsy. If you inject Evans blue, you will see the brain stains blue, but not biopsy mice. So biopsy mice do not show cerebral malaria. But they will all, parasite will grow, they will all die later of anemia. But C57 black will show cerebral malaria and die in about 10 days. Next one, please. This is the model we have used, and then we have huge amount of data. I will not get into it, but I can tell you it doesn't produce cerebral malaria. Knockout parasites do not produce cerebral malaria. That's the biggest finding. This is the wild type. You can see the Evans blue staining of the brain. The knockout, whether it is the first gene or the last gene of the heme pathway, you don't see blood-brain barrier is not broken. And this is all various histology studies, FACS studies, looking at leukocyte migration, looking at CD3 cells migration, looking at IgG devastation, looking at amyloid precursor. For example, you see amyloid precursor protein. It's a typical marker for cerebral malaria in the wild type. You don't see it in the knockout. And these are all various, as I said, various parameters which one typically uses to show cerebral malaria. Next one, please. The bottom line is the knockout parasite does not make cerebral malaria. And we thought it is the food vacuole that is affected. The virulence is caused by the food vacuole, which makes a hemozoin. Therefore, what we did proteomics of the food vacuole. And to cut the story short, we probably, there is only one paper on the proteomics of food vacuole. Very difficult to do. And we were, after a great struggle, we have been able to do the proteomics of food vacuole. And we have done the food vacuole with the wild type, with the FC knockout. And, the main result is we have got a very good proteomics data. But three things are very, very important. The pH of the food vacuole is very important. I told you for hemozoin formation. Heme has to be converted into hemozoin, the heme that is derived from hemoglobin. But the pH is compromised in the knockout parasite. The membrane is, unsaturation is compromised. And the proton ATPase is compromised. Next slide, please. I can show you one or two data. You can clearly see wild type, this is the knockout. This band by Maldi, we could show this is proton ATPase, type 5 proton ATPase. Absent in knockout. This is one batch, this is another batch. Clearly showing the pH, acidic pH is not there for hemozoin to form. Next one, please. Then one data on saturation. We have a lot of data. This is just oleic acid. Oleic acid, methyl ester in the wild type, oleic acid, methyl ester in the knockout. You don't see the knockout. So can you go back to this? So the unsaturation of the membrane is seriously affected. The pH is affected. You don't see the proton ATPase. I think these are the reasons why the food vacuole is compromised. The functionality of the food vacuole is compromised. So we have done the proteomics. We have a huge amount of data. Next one. But these are the three main findings why food vacuole compromisation, where hemozoin is formation is seriously affected. You need heme, and the hemozoin is considered, or free heme is considered as a pathogen-associated molecular pattern. They are considered as PAMs, DAMs, and so on. For example, lipopolysaccharide belongs to these kind of categories. So food vacuole pH, unsaturation of the membrane, proton ATPase, these are responsible for poor functionality of the food vacuole. And that is why we believe the knockout parasite is able to prevent cerebral malaria. Next one, please. And this is just ferrochelatase knockout can prevent cerebral malaria. So we thought, can I have a drug molecule? Fortunately, we could find out repurposing possibility. Griseofulvin, it's an antifungal, which is extensively used medication to treat the fungal diseases, skin diseases. But interestingly, it also inhibits ferrochelatase. So we thought, will it be like a ferrochelatase knockout? Next one. I don't want to go into, I can only tell you all the data which we got with ferrochelatase knockout. We could reproduce it with griseofulvin. By giving griseofulvin, we repeated all the data which we generated with ferrochelatase knockout. We could repeat it, indicating griseofulvin can be a very good adjunct drug to treat cerebral malaria. Next one, please. This is unpublished data. We have heme biosynthetic pathway. Interestingly, a small amount of heme regulating a major amount of pool of heme derived from hemoglobin to make a heme in the food vacuole, because the food functionality of food vacuole is compromised in terms of pH, in terms of membrane unsaturation, in terms of proton ATPase. Next one. And the next one. This is the cartoon I'm trying to show. This is hemoglobin heme, and this is biosynthetic heme, probably even smaller, it's minuscule. And this is regulating this. This is something very, very interesting, and we are trying to publish it in a big journal. Just 2-3 slides on curcumin. Neeraj mentioned this. This is the other molecule, natural molecules, which we have been looking at for potential antimalarial. So griseofulvin is something very recent. Next one. Can curcumin be an adjunct drug to treat cerebral malaria? Curcumin from turmeric, so many diseases it has been used, either by clinical trial or even traditionally, but it is not a drug for any application. So I have been very particular, it should be at least used as a clinically, it should be approved. People talk about bioavailability and so on. All these have been sorted out. People are still in a different world and keeps on saying, not only curcumin, there are many natural products, I must tell you. They are all anti-inflammatory. Medicinal plants give many natural molecules like curcumin. They are all anti-inflammatory, very poorly bioavailable, I must tell you, many of them. But they all have this property to have memory cells generated. They all affect the immune chemistry of the host, and they are present for a short period, a short half-life. Next one, please. And I don't want to get into great data, just clearly showing, if you give, have used nanocurcumin also made by National Institute of Immunology. For example, you look at the brain, and you can clearly see if you have a luciferase labeled parasite, you can detect wherever there is RBC with the parasite. Blue means low parasitemia, red means high parasitemia. Look at parasite is loaded, but if you give nanocurcumin, you don't see any parasite in the brain. RBC containing parasite in the brain. Similarly, with the blood-brain barrier, this is the wild type, and this is the curcumin or nanocurcumin. Blood-brain barrier is not affected, is affected. Next one. So, I believe, keep on going. Next one, next one, next one, next one. This is the summary of the curcumin slides. Next one. There are, how does cerebral malaria is caused in the mouse? It is essentially because of inflammation as well as migration of RBCs into the brain containing parasites, multiple complex things. And what does curcumin do? It prevents completely anti-inflammatory effects. And that is, but parasite will grow. Therefore, the animals die of anemia, but they're not of cerebral malaria. So we thought a combination of artemether and curcumin will be ideal, and we could show all the mice to be survived. And we, if you just use a combination of artemether and curcumin. Next one, please. And we have published it, we have patents and all that. So we have two drugs. One is the griseofulvin. We think it will be a thing to be taken forward. Curcumin, fortunately, after a battle of 10 years, with the DCGI has given approval. Amit is here, director of NIMR. They will take up the clinical trial in 2 hospitals, one in Odisha and one in Chhattisgarh. ILS Bhubaneswar is a collaborator. Of course, we will be there. And IPCA Mumbai has also agreed to, is an antimalarial selling company. and they are part of this thing. You know, I see light at the end of the tunnel. Unfortunately, the two hospitals have become COVID hospitals. We are waiting for them to give clearance so that we will go ahead with the trial of curcumin. Next one, please. You know, it took a lifetime for me. Maybe I have taken more than half hour, 35 minutes to identify two therapeutic molecules. One, gruciafulvin, which I think is unpublished. I think it has a tremendous potential because it's an FDA-approved drug for fungal therapy. And curcumin, finally, after a battle, it has reached the clinical trial stage. And NMR will be doing the trial. And I'm hoping, you know, during my lifetime, I'm only 83, I will get the results to see, you know, that it is an effective drug. Because we don't have a drug for cerebral malaria, I must tell you. The mortality in cerebral malaria is very high. As WHO itself says, it is 25%. Therefore, it is, you know, we really need an adjunct drug. Next one. I also quickly, what are my other activities apart from the science I do? I have been part of the team which developed hepatitis B vaccine. Many of you would know. Dr. Rangarajan was leading in my lab. I was part of that group. And I've chaired several committees, DBT, DST, CSIR, ICMR, all these. I've been closely interacting with DBT right from its inception as NPTB in 1982. And that interaction continues with DBT. And I chair the BIRAC, which is a big thing for the last 10 years, and the technical evaluation committee of COVID vaccine for DBT. You know, I should acknowledge all my scores of students inside and outside IISc campus. I have interacted with generation of scientists, young, old, middle-aged, biotech entrepreneurs. But I think all the pinnacle of all recognition is to have become president of NASI. And you know, this is something, a recognition which I truly cherish. And I will have another occasion to talk to the fellowship, I think AGM probably. This time I thought I will confine myself to science. And I should thank Neeraj and all at the headquarters for all the cooperation. Last slide, one more slide. There's one more slide, and it is an important slide. Yes, can you go back? There is one special person to thank for all that she has done to me, Dr. Manju Sharma. Thank you so much. Thanks, everybody. Yeah. I'm done.
Thank you.
Thank you, sir. We are also very much grateful to you, sir, for your continued guidance and a marvelous leadership leading to academy to and touching several milestones. Regarding your work, I cannot make any comment, but it is your manifold contributions in last several years, more than 60 years, and your role in cytochrome P40 transcription and fixing the role of heme, as well as your work on the curcumin. All these things are really great contribution to the science, and we all are very much grateful to you, sir. I have also read your biography, sir, which has been published by the Vigyan Prasar, and it is really a very, very interesting story with harmony with the struggle for the existence as well as the survival of the fittest. And you are most fit, I must say, even at your age of 83. Sir. Thank you, sir. Now we have with us Honorable Professor Balram Bhargavaji, the Director General of ICMR as well as the Secretary to the Government of India in the Department of Health Research. He is one of the youngest, I must say, youngest fellow of this academy. Why youngest? Youngest for his ability, agility, and the dynamism. Although he is very senior in terms of his scientific significant contributions. he is one of the foremost leaders in biomedical innovation, public health, medical education, and medical research. Earlier, he was the professor of cardiology at All India Institute of Medical Sciences, New Delhi, and also served as the executive director for a Stanford, India Biodesign Center, School of International Biodesign. The government of India honored him in 2004 for his manifold contributions as Padam Shri by the President of India. He also received other prestigious awards, including S.N. Bose Centenary Award by the Indian National Science Congress and National Academy of Sciences Platinum Jubilee Award, Tata Innovation Fellowship, and O.P. Vaseen Award, and many more awards. I cannot take all the names here because the time is short. But his contribution is really great, and we feel pride and privilege to have him as the person who is inaugurating this 90th annual session of this event and the symposium. Thank you, sir. We invite you to say a few words.
Thank you so much, Dr. Neeraj, for your very kind words. Honorable Dr. Manju Sharma, Professor Padmanabhan, Professor Satyadev. I think we've had an academic feast by the lecture by Dr. Padmanabhan today. It is an inspiration to not only, I think it's an inspiration to several generations of the kind of work that has been done, and it has taken a lifetime to achieve that work with complete dedication and complete focus on malaria, curcumin, and the role of cerebral malaria, the role of grisofulvin, the antifungal in combination with curcumin. So congratulations, sir, and it's an honor to share the dais with you and also the dais with Madam at this point in time. Now, I think the annual scientific session has the title is that towards a new healthcare regime for the nation. Now, I do not have any slides, but I will talk some general points regarding the healthcare of the nation and in what direction are we heading. We know that the healthcare of the nation has been tested during this COVID pandemic, and we have realized 3 or 4 important points from this pandemic. That one, we have to get out of our silos. We have to work together, whether it be within the organization or within the institution or within the laboratory, within the science body, or as commitment for the entire nation. That is the first point. Second point, it is very clear that we can do it, and we have to work within a time-bound manner, and we can do it because the health of the country is very important. Our life expectancy, although has increased, but still we are lagging behind in many parameters, whether it be your infant mortality rate, maternal mortality rate, etc. So those are issues which we need to address. So we cannot rest even for a minute as far as, so first is breaking out of your silos. Second is we have to have it time bound. The third point is that we have a very good time in terms of a very great leadership, and health has become a very important focus for this nation, for national development. And if you have a healthy society, you have a healthy nation, and that leads to a healthy economy. So that is imperative and most important as far as the development of the country is concerned. The fourth point is that from this pandemic, we realized that at this point, the governance, we have an excellent leadership who is ready to support healthcare in every which way. And the basic philosophy of healthcare as in the current leadership is to have a holistic healthcare. It has to be an integrative healthcare, and it has to be a preventive healthcare. So a holistic healthcare, a preventive healthcare, and an integrative healthcare leading to and based on the principles of Atmanirbhar Bharat with our self-sufficiency, whether it be in diagnostics, whether it be in treatment, whether it be on manpower, whether it be on training, so that we have to become self-sufficient in every which way. The other little point that I want to emphasize here, and that is more so in our scientific community, is that we have to get out of our Macaulay mindset. We have to get out of the Macaulay mindset that anything that is non-Indian is good. That mindset we have to change if we have to show that the scientific as well as the healthcare prowess of our nation. We have to get out mentally out of this Macaulay mindset, which was clearly instilled in our mindset when by the East India Company, and at that point in time, that anything that if we have to rule over a country which has such excellent traditions, such excellent diversity, such excellent culture, we have to break the backbone if we have to rule, that they should realize that anything Indian is not good, anything which is international is good. So that was the 1834 Macaulay's the mindset, where he started it at the school level, and gradually we are still seeing the repercussions of that in our society, and that is an important mindset change. And this government seems to be supporting this when they talk about Atmanirbhar Bharat and trying to get self-sufficiency in our scientific program as well as in our healthcare program. The healthcare budget of the country has remained and stuck at 0.9% of the GDP for several years, several decades. There has been talk about raising the budget from 0.9% higher since the early 90s or even late 80s, that it has to be increased. But every time it was shot down by various people saying that India doesn't, the healthcare system in the country doesn't have the capacity to absorb that money, and therefore there will be tremendous amount of wastage. So it was never raised. But after, in this millennium, the governments, successive governments have realized that health is becoming an important agenda, not only for politics, but also for the development of the nation. And therefore, they have started realizing that we must spend more on healthcare. Now, at one end of the spectrum, we have India with 0.9% of GDP of health on healthcare. And at the other end of the spectrum, in the United States, we have the 19% of the GDP of a $19 trillion economy spent on healthcare. 19% of $19 trillion economy, which is more than the economy of India as a whole. That is what US spends on healthcare. We do not need to go, the pendulum should not swing that much. However, it has to swing to a certain level wherein countries which have had an exemplary socialist healthcare, whether it be the Scandinavian countries or many European countries or the United Kingdom, where the GDP on healthcare hovers around 7-8% of the entire GDP. So if that is the target and we are at 0.9%, we really need to get our act together in a big way. In terms of the socialistic, I'll just touch upon the socialist healthcare a bit here, is because the committee in 1942-43, which was decided how the healthcare is going to develop after independence, clearly said that it has to be a pyramidal structure with tertiary care, secondary care, and primary care. And the primary care has to be the focus, and then secondary care and tertiary care. This went on well gradually. It was going at a slow pace in the 50s and the 60s and the 70s. But in the 80s, with liberalization of healthcare occurring in the early 90s and 80s, we had more of tertiary care spin to the whole healthcare system, and primary care suffered in a big way. Similar thing happened in the United Kingdom when tertiary care was being promoted in a very big way in the early 80s. And at that point in time, the NHS took a conscious decision. They took a conscious decision of promoting GP care and general practitioners care. And the general practitioners were incentivized to be more than tertiary care specialists. And they were able to retain their pool of general practice, their pool of primary care in such a way that it is exemplary for the entire world, how their pyramid is so robust. Even if you look at countries like Scandinavia, where they have excellent tertiary care, the state provides for tertiary care at every level. State provides for PhD, totally funded, and from cradle to death, the entire healthcare is taken care of. However, the primary care and the GP system in the Scandinavian countries is still not working as well, is not there at all. That is the biggest problem. They have, again, the pyramid, the apex of the pyramid is strong, but the base is not there in countries like Scandinavia. So India has been confused whether it has to go the socialistic way or it has to go the privatization way. But I think there has to be a balance, and this government, with the launch of the PMJ program and the vision of setting up 150,000 health and wellness centers across the country, out of which 60,000 have been set up, which will be advising and treating on preventive, promotive, as well as the investigations for the non-communicable diseases, mental health, and dental healthcare. They have 12 pillars on which this health and wellness centers will be working, and 60,000 have been established, and they need to become more robust with the use of integrative medicine and preventive medicine, with the vision of having established 150,000 by the end of 2022. So that is the important goal, which is somewhere Buddha's middle path between private medicine and tertiary medicine, as well as socialistic medicine and primary medicine, somewhere in between. This is the path that the government is taking today, and I think that will hold us in good stead in the future to come. In terms of the budget announcements in this financial year, we've seen that the healthcare is now being viewed holistically, with healthcare, water, sanitation, hygiene, maternal care, child care, all. The budget for the entire whole expand for all this is 220,000 crores. For healthcare alone, Ministry of Health is 70,000 crores. It was 60,000, that has gone to 70,000. But if you combine all this, this becomes about 220,000 crores in the current budget that has been declared. So if we have safe water, if we have clean water, if we have good maternal child health care, and if we have good preventive care, we will be able to spend less on treatment care, which has been the focus in the past few decades. That treatment, tertiary care, private treatment, and we are spending too much money on that as a nation, and that will probably change once we have this preventive, promotive, and holistic care centers set up. Many of them have been set up. We have seen the ASHA workers have been the backbone of our primary centers and the health and wellness centers, and we need to scale them up. At the current moment, we have about 9 lakh ASHA workers, and there is a plan to increase or double the number of accredited social health activists or ASHA workers, which have contributed immensely to healthcare. Another factor that is coming into our integrative care is the role of Ayush physicians and the role of the Ayush doctors, how they will pitch in to health in our primary care or in our healthcare ecosystem as a whole. Now, the other declaration in this budget has been which we have benefited from the Department of Health Research and the ICMR, is the aspects of preventive biosecurity for the nation, One Health for the nation, and providing principles and know-how with science and technology driven for disease elimination. the promise of tuberculosis elimination by 2025, and we have heard an excellent lecture on malaria eradication by 2030 also remains the most important vision that we have benefited from this budget in terms of the setting up of the One Health Center at the Maharashtra Agriculture and Fisheries University in Nagpur, right in the heart of India, in the center of India, the Institute of One Health with a BSL-4 laboratory or maximum containment laboratory there, and the setting up of 4 regional national institutes of virology in Chandigarh, Dibrugarh, Pune, and Bangalore, and one more in the north if possible. So we are looking at that, and if we are able to set up these NIVs in the very near future, they will provide us self-sufficiency or Atmanirbharta in biosecurity and biosafety, and we have to work very closely with and again what we learned from the pandemic, we have to get out of our silos, we have to work with other agencies. Many of our science agencies are competing with each other. More important is that they should be synergistic with each other, depending on what is the cabinet rules of business for that ministry, and that should entirely be ensured to and stuck to as an important legal framework so as to provide synergy in the science as well as healthcare that is going to be delivered to the nation in the next decade or so. The important focus from this budget again is that we are getting the NIVs, we are getting the Institute of One Health, and we have also got funding for setting up an Institute for Disease Elimination Science and Health, which is called DESH, D-E-S-H, Disease Elimination Science and Health, which is mainly will be looking at elimination of tuberculosis and the science behind it, and even after elimination, how that resurgence does not occur, and for malaria. So those are also the 2 priorities that will be from the ICMR DHR perspective for this. We have also are setting up have set up parts of it, but the clinical trial network. What did we learn again from the pandemic? That we liberalized the research. We were able to conduct research in non-elite institutions, and they were performing very well. As we have seen for ISRO having a lot of triple IIT and NIIT people helping out in ISRO in a big way. Similarly, we have found regional medical colleges, have worked so well in research, and they need to be plugged into our entire national network. So we have set up a national clinical trial network for doing clinical trials on various areas, which can be deployed from top down at all these institutions, as we had done for the plasma trial and other pharmacological agents during the COVID pandemic. The other learnings which are very important and from this pandemic again is that why we got this pandemic in this country. And we know, we discussed last time about climate change, global warming, damage to our ecology and environment, poor funding of public health, poor public health, and number of public health specialists are small. So those are areas again where the government will have to really take care of, and we as a nation have so much of biodiversity, whether it be in the Northeast, whether it be in the Silent Valley in Kerala, whether it be in the Western Ghats of the so we have a great biodiversity, and we have to preserve that in every which way so that we do not have more of these pandemics which are related to the changing ecosystem for the bats and other animals who have to move out of their own forests because we are encroaching on the forests. If we encroach on the forest, we will ultimately damage our cities by getting all these kind of pandemics. That is very, very important. And global warming remains an important agenda wherein we need to do much more as a scientific community so as to prevent and keep a healthy nation. And if we have the healthy nation. Now, if we look at the perspective of testing all our passengers who come to our country. Yes, we will test them, but we will be in an isolated bubble. However, we vaccinate our people, we vaccinate a region, we will be able to eradicate the disease in this region. But ultimately, man is a social animal. We have to live in this whole world as an ecosystem. So the disease has to be eliminated. And that is why the nation to donate vaccines to more than 30 countries now and try to break the chain of transmission of the virus, not only in your community, not only in your city, not only in your nation, but the entire world is our agenda. Unless we break the chain of transmission of the virus in the entire world, we will not be able to eradicate this disease. And this will again be endemic and resurface, as has happened with SARS, as has happened with MERS and other diseases. So that is another area where we have to have and embrace our entire and our principle on Vasudhaiv Kutumbakam, world citizenship, and that is what has been the mantra which we have worked at the ICMR and the DHR, that we have to take the whole world into account. So with the other point that we have to emphasize now, as our nation is facing although our life expectancy has now gone up to 70 years, but we have a pandemic of, or rather an epidemic of non-communicable diseases, diabetes, hypertension, strokes, cancer, renal disease, high dyslipidemia. We are the world capital of diabetes. We are the world capital of hypertension also now. So those are things where we will have to address, and these non-communicable diseases can clearly be tackled by preventive and promotive healthcare, and mindset change for practicing a healthy lifestyle and a healthy diet, which will really help us prevent these diseases and taking the toll on the target organs, which are the eyes, the kidneys, the heart, and the brain. Because if we are able to control these diseases in our environment, 40% of the DALIs lost in the country are because of communicable diseases, and we have traditionally a robust network to tackle these communicable diseases, but they are all, as I mentioned, holistically linked to proper sanitation, proper hygiene, women and child care, maternal care, poor. If I were to ask what are the 4 major killers in India, I would say 1 is air pollution, 2 is hypertension, 3 is cancer, and 4 is poor nutrition. These are the 4 pillars of our poor healthcare, which contribute to the entire deaths in our country, major deaths. Poor nutrition in children is still a problem, even 70 years after independence. It has to be taken with the bull by the horns, and we need to really get our act together. Poshan Abhiyan is a big mission. The DBT, the ICMR, they are all playing their little bit role, but I think this has to be a huge mission. And we did the midday meals and other things did suffer during the pandemic, and they need to be again started back in a big way. Air pollution again linked to climate change, linked to global warming, linked to the pandemics, need to be managed in a big way. Hypertension, by controlling hypertension, it accounts for more than 20 to 25% of the adults in the urban population, and in the rural population, and up to 50% of the adults in the urban population suffer from hypertension. 30% are being treated, out of those 130 are being treated for hypertension, and less than 20% are being treated adequately. So if that is another major killer, and we need to reduce salt intake, change our lifestyle, fresh fruits and vegetables, and control hypertension. So these 4 killers are and cancers. Again, cancers are increasing, more related to air pollution, related to more chemicals, more use of. So all those things are contributing to cancer. However, we've recently received, we've been conducting the cancer registries, more than 250 cancer registries since 1982, and we've just submitted the report to the Ministry of Health. and we are seeing a surge of a large number of cancers in the Northeast region. And because of the basis of that report, the government with the Tata Grid and others are setting up several cancer hospitals in the Northeastern region. But again, I would finally say that this is our Karma Bhumi. We have to spend our lifetime and learn from people like Professor Padmanabhan, Professor Manju Sharma, and senior people who are present, having spent their lifetime for the betterment of the citizens of our nation. I wish all success for our future. Thank you so much for your attention.
Thank you very much, sir, for a very nice and descriptive, I must say it is a descriptive lecture about the role of the medical field in the development of the nation with the adoption of preventive and biosecurity measures, and especially in these days, the role of the ASHA workers, even the three-layered structure of the healthcare system, all those things which have been emphasized by your good self is really very much the need of the hour. And due to all these concerted and coordinated efforts, the country is developing. And today news that that USA has tried to get a MOU prepared for the India healthcare system and the medical resources research. That is a great news, because that shows that our experience, skill, and our infrastructure has got to the mark. And we are very much thankful to you for your regular, concerted, and dedicated, devoted efforts, which has made this possible, sir. Thank you very much, sir. This is the now end of the session, and regarding the very experienced and very scholarly lectures which have been delivered in this session, I would like to say that the need of imagination as a sense of truth and as a feeling of responsibility, these are the three forces which are the very nerve of science and education. And all three lectures which were delivered by Honorable President, Honorable Madam Manju Sharma ji, your good self, Professor Balram Bhargava ji, and Professor Satyadev ji. These were the mark of this. These were very much true to this quote given by somebody, and it shows how our leaders in science are guiding us, are showing the path of success to the fellow countrymen and the fraternity, scientific fraternity. We are very much grateful to all other chairpersons, co-chairpersons, and the distinguished speakers. I cannot take the names of everybody, but for the next session, I think Professor J.S. Tyagi has come, and Professor Paramjit Khurana ji has also come, and I've just seen Dr. Amit P. Sharma ji has also come. So I am also very much grateful to all of them and the other co-chairs and chairs like Dr. Sanjeev Sinha ji, Professor Ishan Patro ji, Dr. Beena Ravi ji, Professor UC Srivastava ji, who is sitting here. He is also the treasurer of this academy. Professor V.M. Katos, Professor A. Lakshmaya, and also the two sectional presidents, Professor Sanghamitra Bandyopadhyay ji and Dr. Smita Dilip Mahalay ji. We have also with us several past presidents. Professor Akhilesh Tyagi ji is here, Professor J.P. Mittal sir is here, Professor Ashok Mishra ji is here, Professor V.P. Kamboj sir is here, and several senior fellows are here. Professor Jaya Tyagi ji is also here. So we are very much grateful to all our fellows, members, council members, past presidents, and our honorable president for giving us this opportunity to come on the international platform to celebrate our 90th annual session as well as the symposium. We are very much thankful to all the participants and everybody. Thank you very much. Now we are going to start after 5 minutes the next session. We will take only 5 minutes break, as madam has said, because all the participants are here. So please Don't go anywhere. Please be connected. We are going to start within 5 minutes. Thank you very much.
I think, Neeraj, we don't need the break. Hear me?
Yes, yes, madam.
Break. Straight away go, but I will request the chairman and co-chairman to introduce the speakers and introduce the session. And Neeraj, from now onwards, we hand over to the chairman and co-chairman for various sessions.
Yes,
Okay?
yes, madam. Yes, madam.
Jaya ji, आप हैं, so please शुरू कर दीजिए. Mr. Jaya,
Madam, let me introduce. Actually, I would like to request Dr. Santosh, because he is coordinating this event, to kindly introduce the chairperson of this session one, so that after that the chairperson would introduce others and start with it. Dr. Santosh.
very briefly, Santosh.
Very good morning to all of you, and welcome to you all in this technical session.
See you.
One.
Can't see you.
Yeah, madam, I'm here, madam.
Switch on your video.
So I request the chairperson of this technical session one is Professor Jaya Tyagi, madam, and the co-chair is Professor Paramjit Khurana, madam. Both are here, I think so. Professor Jaya Tyagi is our NASI senior scientist and is former head department of biotechnology, All India Institute of Medical Sciences, New Delhi. And Professor Paramjit Khurana, madam, is our general secretary, outstation, and is former head department of plant molecular biology, South Campus, Delhi University, New Delhi. Our three speakers are also here, I think so. The first one is Professor Manoj Murhekar, and the two other speakers are Dr. Amit P. Sharma and Dr. Raghavan Vardarajan. So I request to the chair and co-chair to this session to kindly proceed the session. A brief introduction is also here, and I request to the chair and co-chair to kindly invite the speaker with their brief introductions. The first speaker, Dr. Manoj Murhekar.
Thank you very much, Santosh ji, for this opportunity to chair this session along with my colleague, Dr. Paramjit Khurana. And just to be very, very brief, we're going to have we have a lineup of three eminent speakers, and as already indicated, there is 25 minutes per speaker. And my request is that we may put the questions to the speakers in the chat box, which may be taken up at the end of the session if there is time permitting. So with these very few brief words, I would like to invite Dr. Manoj Murhekar, who is at present director of the ICMR Institute, National Institute of Epidemiology at Chennai. Dr. Murhekar is a committed epidemiologist with undergraduation in medicine and post-graduation in community medicine. And he is now at Chennai, but before this, he has done some very outstanding work at RMRC, which is another ICMR institution in Port Blair. And he has numerous publications to his credit and awards. I will not go into those details. It is up on your screen. And he is actually going to speak on a very relevant topic. And I'm sure we are all waiting to hear from him directly about the COVID Zero survey and an insight into pandemic in India. So with those very few words, I request Dr. Murhekar to kindly deliver his talk. Dr. Murhekar, could you please project your slides?
Sir, you are muted, sir. Manoj, sir, you are muted, sir.
Good morning. Can you hear me?
Yes.
Yes, we can hear you. Yes.
I need a sharing access basically. I am not able to share my slides.
So,
Take me a code.
Dr. Neeraj, please allow him to share his slides.
Sir, share button is enabled, sir. Please, sir. Yeah, okay. I'll do that. Just a minute. See my slides now?
Yes, yes. Perfect. You can see it. Please go ahead.
Slides now.
Yes, we can see.
A very good morning once again. At the outset, I want to thank Dr. Sharma for inviting me to talk in this symposium. Well, I'm going to talk about COVID zero surveys and an insight from these surveys into India's pandemic. Well, I'm going to cover some of these topics. First, we talk about which most of us know. And then I will talk about the insights from various zero surveys that have been conducted in India about the spread of COVID, about the testing rates, about the mortality, different high-risk groups, and lastly, I will briefly touch about the herd immunity that everybody talks because of or against COVID. Well, very briefly about the COVID timelines in India. The first case was reported in India on 30th of January, and this case was a medical student who was studying in China, Wuhan. She had come from Wuhan, and she was from Kerala's Thrissur district. On the same day, WHO declared COVID-19 as a public health emergency of international concern in view of rising number of cases as well as deaths, mainly in Wuhan in China. Well, in February, we did not have many cases, and by March, we had little more than 500 cases. That included few clusters among Italian tourists as well as some family clusters, mainly related with travelers from Italy and some European countries. But in view of increasing number of cases in the country from different districts, Government of India took a decision for imposing a nationwide lockdown, which started from 25th of March. Initially, it was for 3 weeks, but the number of cases and the number of districts that were affected because of COVID were increasing, and as a result, the lockdown was extended till 3rd of May, and then till 17th of May, and lastly till 1st of May. The unlockdown process again was a phased process starting from 1st of June. Then the several efforts were ongoing globally to produce a vaccine, and on 30th of June, DCGI gave approval for trial of indigenous Covaxin vaccine. On 26th of July, we also have DCGI, the Covishield vaccine trials began in India. Number of cases were increasing in India steadily, and on 7th of September, India overtook Brazil and became second largest country with number of COVID cases. And it was September 12 when India reported highest daily number of cases. In fact, it was little less than 1,97,000 cases were reported. And from end of September, we have been seeing gradual decline in number of cases. Although we are seeing little upsurge in the last couple of weeks. On 31st of December, on January 3rd, DCGI provided emergency use authorization for two of vaccines, that is Covishield and Covaxin. And as we all know, January 16, the first phase of vaccination began in India, primarily targeting healthcare workers. From February, it was the frontline workers, and from March, it will be the elderly population who will be targeted for vaccination.
Well, we have nearly 1.1, more than 1.1 crore cases and 156,000 deaths reported till date. Well, now coming to my topic of presentation about sero surveys. So why basically we need these sero surveys and why these are important in the context of COVID? Although the facility-based surveillance, which is ongoing in all the districts in India, provide useful data to know the trend of infection. But this surveillance depends on several factors that includes the testing strategy of the country at that or district at that moment in time. It also depends on number of tests that were done. Also about the type of tests that tests that are used for diagnosis. Another very important thing is that most of the COVID cases are mild in nature, or they do not have any symptom or symptomatic cases. And such individuals are less likely to seek healthcare and less likely to be detected by facility-based surveillance. And in that context, serological surveys or serological or seroepidemiological studies are very important. In fact, they can answer two important questions in the context of COVID. The first is they can tell us as to what proportion of population has been infected in the past. So that is first important question that sero surveys can answer. Then sero surveys can also help identify the geographic distribution of the disease, identifying hotspots, high-risk groups, and so on. And the second important question which sero surveys can answer is what proportion of population is immune to disease or infection, although with certain limitations. Well, in May 2020, nationwide sero survey, and following this sero survey, ICMR issued guidelines to all the states to conduct seroepidemiological studies, both in general population as well as high risk groups. And as you can see, some of the cities where sero surveys have been done. This is not exhaustive list. It's some of the cities where landmark studies have been done. And several states also have conducted seroepidemiological studies estimating the prevalence of SARS-CoV-2 infection in these states. Well, I will talk about the ICMR nationwide sero survey, and also then touch about other sero surveys that have been conducted in the country. So we conducted the first sero survey in May when the country was in lockdown. And this survey was conducted in 70 Indian districts from 21 states. And the objective of this survey was to estimate the age-specific prevalence and also identify different risk factors of infection. And also comment about the testing as well as fatality because of COVID-19. And since after the first survey, ICMR also conducted two more sero surveys. So this provided an opportunity to study the trend of infection over a period of time. So as I mentioned, all the three sero surveys were conducted in these same 70 districts and same 21 states. Very briefly, I will talk about the study design of these surveys. At that time in May, when we were planning for this sero survey, the different districts in India were color coded as red, orange, and green based on number of COVID cases reported. So we decided to make 4 data. We divided districts in 4 data based on the incidence of reported COVID cases. And from each data, we selected 15 districts randomly. And from each district, then we selected 10 villages or wards. And from each of the cluster, we selected 10 eligible adults with 1 adult per household. So we had decided a sample size of 6,000 adults per stratum, that is 400 individuals per district. So for the subsequent 2 rounds, second and third sero survey, we did the survey in the same district, same 700 clusters. But the second survey, in the second survey, we included individuals who are aged more than 10 years. And in the third survey, besides general population, we also covered healthcare workers. Well, selecting appropriate lab serological test is very important for any sero surveys. That is especially true if the prevalence is low, and if the specificity of the test is low, we are bound to get very high false positivity. And at that time, the COVID Kavach ELISA developed by ICMR NIV was only indigenously available ELISA test. We decided to use this test for serological testing. And since the test did not have 100% specificity, we decided to test all the positive samples with another ELISA, that is European ELISA, which has a higher specificity. And this testing yielded better specificity, that is 99.9%. In the second round, we used a chemiluminescent immunoassay which detected antibodies against nucleic acid protein. And we know that there was a lot of literature documenting decline in nucleic acid and decline in IgG antibodies in general, and nucleic acid antibodies in particular. In the third round, we decided to test all the sera with two tests. One was Abbott test detecting IgG antibodies against nucleic acid protein, and second was antibodies against S1 RBD. That is what we did in the third round. So coming to the situation or seroprevalence in May. So this describes the seroprevalence in the country by May. As you can see, the overall we tested about 28,000 individuals, and the overall seroprevalence after adjusting for the test performance was 0.73. And you can also see that in different strata that was created based on the reported incidence of reported number of cases, the seroprevalence was not different. So low seroprevalence, less than 1% of the population was exposed. But what it indicated was we were in very early phase of epidemic, and infection had not really spread much, mainly because of the lockdown, the stringent lockdown that was in place. It also indicated that there is a high susceptibility of population because 99% of the population at that time was not exposed. And therefore, it was reasonable to expect increase in number of cases in coming months. A similar prevalence that was observed in different strata indicated two things. First was there was under-detection in low incidence areas, and again, need for more testing. And also it highlighted issues related with access to testing, especially in some of the northeastern states. Well, in the second round of sero survey, which was conducted in August, 17 August to 22nd September, we surveyed about 29,000 individuals. And at that time, as I mentioned, we included individuals aged more than 10 years. The overall prevalence was 6.6%. But for comparison purposes, we estimated seroprevalence in adults, which was 7.1%. So the seroprevalence basically has increased from 0.7% in May to 7.1% in August, about tenfold rise. In December, when we conducted the third survey, the seroprevalence has increased to 24.1%. And this seroprevalence is based on presence of antibodies either to N or S1 RBD antigen. So this increase that we saw, about tenfold increase between May to August, that was seen in all 70 districts. But this increase was highly variable in districts such as Kulu and Palakkad. The seroprevalence increased from 0% to 0.5%. There is a typo. Whereas a district like Ganjam in Orissa, it has highest seroprevalence in second by August, that is 42.5%. Again indicated because Ganjam was one of the districts that there are a lot of migrants coming to their hometown. And that could be, that was one of the reasons for higher seroprevalence. This tenfold increase over a 90 days period revealed infection doubling time of about 27 days. This also had an implication for vaccination because we thought that if infections are doubling at a rate of 27, doubling in every 27 days, so we would reach that herd immunity threshold of 50 to 60% by December. That is what we were seeing a decline in number of cases by that time. Between August to December, the seroprevalence has increased at least by 2.2-fold when we compare the prevalence by nucleocapsid antibody. But this time, the prevalence, the increase in prevalence was not in all the districts. In fact, in 55 of the 70 districts, the prevalence increased. But in 15 districts, the prevalence declined. And the main reason for decline was decline in nucleocapsid antibodies. That is well known. And between August to December, the infection doubling time in the country was about 6 to 8 days. So as I mentioned, between August to December, the increase was seen in 55 districts. And the highest increase was seen in Kulu, which had a lowest prevalence in second survey. Whereas in 15 districts, the ratio of anti-N positivity by August and December was, it ranged between 0.37 to 0.81. And Chennai had again the decline in seroprevalence, and because this were the Chennai was one of the cities which had the peak much earlier, and the decline in this ratio indicates decline in antibodies over a period of time. The serosurveys can also talk about, can also tell about who is basically at higher risk of infection. So, so this slide describes the age and sex distribution of IgG antibodies. So as you can see, the seroprevalence at the time of second survey was not different by sex as well as by age. So the lower or comparable seroprevalence in younger children, that is 10 to 17 years, and elderly is a very noteworthy finding, because at that time schools were not open, and also the non-pharmaceutical interventions were in place, were implemented strictly in all the states. And despite these schools being closed and despite non-pharmaceutical interventions, children and elderly had a comparable seroprevalence, again indicating a household transmission, indicating that these children and elderly are exposed to adults who are active, who are working outdoors, and again indicating a household transmission. In December, again, we got a similar picture. There was increase, as I mentioned, but one again important finding was 18 to 44 age group had lowest seroprevalence. But eightfold, whereas in groups more than 0.4. So this distribution again is not consistent with the distribution or incidence by age group in India. And this again indicates a decline, because this was the age group which was earlier affected in early phase of pandemic. And the lower prevalence that we observed indicated a decline in antibodies that this age group have acquired. Well, coming to the incidence by the prevalence by residents, in August, again, we got a gradient. The prevalence was lowest in rural areas. Only 5% of the rural area was affected, as against about 17% in urban slums and 9% in non-slum areas. A similar pattern was seen, but what we saw by December was increase in seroprevalence was much higher in rural areas. The prevalence increased by about 2.5-fold in the intervening period of August to December. Whereas in urban slums, the prevalence didn't increase because it has really sort of saturated. And similar findings were observed by or reported by several serosurveys that have been conducted in different Indian cities. And just to give one example of this study done in Mumbai, this was done in 6 slums and non-slum communities across 3 wards in Mumbai. And as you can see, urban slums had much higher seroprevalence, even more than 50% seroprevalence compared to non-slum areas. Well, then based on the seroprevalence, we estimated the disease burden. We estimated the number of infections at different time points. So we estimated about 6.4 million infections in May, which increased to 74 million in August, and about 271 million infections by December. And at that time, the reported number of COVID cases were 79 in May, 79,000 in May, 2.3 million in August, and 10 million in December. So, when we compare the number of cases reported and number of to that of number of infections, and calculated what is called as infection to case ratio. So during May or by May, the infection to case ratio was 81.6, indicating that for every COVID case, there were 82 infections, which declined to 32 by August and 27 by December. This high infection to case ratio in again indicated 2-3 points. First is at that time we were prioritizing symptomatic for testing. We also had limited testing capacities initially in the pandemic, and also the testing rates were variable across states. And the decline in infection to case ratio subsequently was on account of increasing in expansion of testing strategies, as well as expansion in testing capacity in the country. And just to highlight about the expansion of testing labs, when the first case of COVID was diagnosed, we had only one lab, that is NIV Pune, which was doing confirmatory assay. By first week of February, we had 14 labs, which by August it increased to more than 1,500 labs. And this was possible with efforts from several organizations, including involvement of VRDLs in diagnosis, involvement of mentor institutes. ICMR identified 14 mentor institutes, which mentored 300 medical colleges to upscale COVID testing. Scientific and technical institutes in the country like CSIR, DBT, DRDO, and ICR were involved in testing. TrueNat and CBNat were deployed where testing was peripheral facility. Private sector was roped in testing, and we then in June also introduced rapid antigen test for doing testing on a massive scale. Similarly, the testing strategy, in February, it was a symptomatic international traveler who were tested or who were prioritized for testing. Whereas in March, this testing strategy was expanded to context of positive case, include healthcare workers who are having symptoms, and healthcare workers who are managing SARI patients and who are symptomatic. And also asymptomatic direct contacts of cases were also prioritized for testing. In April, when containment zones were identified, all the influenza-like illness patients or cases were also prioritized for testing. In May, again, the testing strategy was expanded to include all hospitalized patients who develop ILI while hospitalization were tested. The migrants and returnees from different states were also prioritized for testing. And June, again, there was expansion of in testing strategy, which included asymptomatic high-risk contacts as well as pregnant women who were hospitalized. And very importantly, testing on demand was began by June. So the improvement in infection to case ratio was on account of both expansion of testing capacity as well as testing strategy. Another important use of sero surveys is estimation of infection fatality ratio. Well, based on the number of cases and number of deaths that have been reported, India has a case fatality ratio of about 1.4%. And this is one of the lowest CFR in globally. And so we basically estimated infection fatality ratio, which takes into account the total number of infections as against total number of cases in CFR. So in 70 districts where we conducted the survey, the infection fatality ratio was 11.7 COVID deaths per 100,000 COVID infections. Which in May, very similar in August, that was 9.4 deaths per 10,000 infections. Compared to other countries, the IFR, the infection fatality ratio in India was much lower. A meta-analysis which was published in July, August indicated IFR 68, and compared to that, the IFR in India was very low. So there could be several reasons for lower IFR in India. One was is about death reporting, and we know that the death reporting is incomplete in many districts, in many states. And also, it also could be because of differential access to testing for patients or for declaring COVID confirmed death. It also, the lower IFR that we observed also could be because of difference in age structure. We have a much younger population in the country, and also could be because of prevalence of comorbidities in different countries. There have been studies where they have estimated IFR by age group, and this is one sero survey conducted in Chennai. In fact, two sero surveys, one was conducted in July and second in October 2020, which again, if you see the IFR by age group, and you can see that the elderly had a much higher infection fatality ratio compared to younger age groups. Similarly, males had higher IFR. So this is again consistent with many studies conducted through. Well, we all know healthcare workers are at higher risk of infection because of their occupation. And in the third sero survey, especially because healthcare workers were targeted for vaccination in the first phase, we decided to estimate seroprevalence in among healthcare workers in the third sero survey. And as a part of this sampling, we sampled 100 healthcare workers from who were working in peripheral health facilities. So we sampled about 7,000, more than 7,000 healthcare workers, and the seroprevalence was again comparable to general population. It was 25%. But we really didn't find any difference by categories of healthcare workers. So again, indicating that the infection is widespread in the community. Well, coming to the last part of my talk about herd immunity. Well, sero surveys can give an idea about antibody-mediated herd immunity or immunity in the population. And for any infectious disease, once a large proportion of population is already infected, transmission is expected to come down. And for COVID-19, the expected herd immunity threshold based on the classical R0 of 2 to 3, the threshold is 50 to 60. I've been seeing number of COVID-19 cases in India since October. And one very important question that has been asked is, has India achieved herd immunity? Well, this slide describes the seroprevalence that have been observed in different sero surveys in India. And although by December 2020, as per the third ICMR survey, about 25% of India's population was exposed. If you see, the several mega cities or several metros had much higher seroprevalence, and this decline that we are seeing in India could partly, or even I will say could largely be due to higher seroprevalence that have been that is seen in many of the urban areas. But we must keep in mind that only 25% of the India's population is exposed to the virus, and a large proportion of population is still susceptible. And we need to keep this in mind. People are still susceptible, and there is a need to implement this non-pharmaceutical intervention. Moreover, herd immunity acquired through natural infection can't be a solution for reducing transmission. And I just want to conclude by giving the recent, citing a recent paper from Science about an experience from Manaus province in Brazil. This particular province had a very bad COVID epidemic, and because a large proportion of population was affected, The second, at that time, they thought second wave is unlikely, and the several serial sero surveys that were conducted indicated a very high sero prevalence. So as much as 76% of the sero prevalence was as much as 76% by October in that country, and therefore the second wave was virtually thought unlikely in this particular province. But beginning January, there was abrupt increase in number of cases. More than 3,000 cases were reported in 2 weeks' time. And in spite of the fact that the country had a very high seroprevalence. So, and then several theories were suggested that antibody-mediated immunity might have come down. But one of the very important point was, or possibility was emergence of variants. And the studies then identified some immune escape variant, especially the P1 lineage that was detected in this province. So the point which I want to make is, despite the fact that we are seeing decline in number of cases, despite the fact that 25% of the population is affected, we should keep in mind that large proportion of population is still susceptible for infection. And we need to continue the non-pharmaceutical interventions. We need to continue the ongoing surveillance, which is ongoing in different health facilities. And since the prevalence was much lower in rural areas, now since from beginning March, elderly will be vaccinated. The focus should be on rural areas so as to vaccinate people or elderly people from rural areas. So thank you very much.
Thank you very much, Dr. Murekar, for a very, very comprehensive summary of the 3 sero surveys, as well as of giving us sounding a note of caution that all is not over yet, and we need to keep our guard. Thank you very much. I think we are completely out of time, and I would request any questions to be please posted in the chat box, which we will take up later. So thank you, Dr. Manoj. Once we now proceed to the second talk of the session, and which is to be delivered by Dr. Amit P. Sharma as a Professor M.G.K. Menon Lecture Award 2020. It is indeed a privilege for me to introduce Dr. Sharma, who is an ex-crystallographer, and he is very briefly, he completed his PhD in the US and his postdoctorate in UK at the University of Oxford, and briefly served there as a faculty. Subsequently, he has joined ICGB, and it's also up on your screens now. He has joined ICGB, served ICGB for several years, and currently he is working as the director of Malaria Research Center in New Delhi. He has been a recipient of very many major awards, including the Shanti Swarup Bhatnagar Prize, and he is a fellow of all the academies. And in addition, he has got the National Bioscience Award for Career Development, the Infosys Award, Ranbaxy Award. I think the list is very long. I will not spend time and not come between the speaker and you. So I want to give his talk on drug discovery against the malaria parasite. Dr. Sharma, please.
Thank you very much. I hope I'm audible. And my slides are visible.
Yes.
So thank you very much for, I'd like to thank the organizers for this really great opportunity to present some of our work. But before I start that, I'd like to just reminisce about Professor M.G.K. Menon, in whose name I'm giving this lecture, who I knew for close to 4 decades. And I think just about everybody in this conference probably knew him for the same time or for even longer. And we all have just some absolutely amazing memories of him. And I won't speak much more about him because all of us have had an independent and very fruitful relationship with him over a long time. So I do remember him at this moment. And I also like to thank the organizers for slotting me into this session, and Dr. G.P. Padmanabhan especially, because he's already given you a precursor of malaria research. I'm going to just discuss some of the work that we've done over the past 10 years towards discovery of new molecules that can work against malaria.
Amit, you need to share your slides. I don't think we can see them.
Yeah.
Yes.
Can you see them now?
Yes. Yes, yes, we can.
Okay, so the very first slide here.
Projection. You may like to go into presentation mode also. Yes,
Yeah,
yes.
this is
Yeah,
fine.
great. Great.
And so the very first slide here summarizes some of the ideas that I will discuss in the subsequent parts of this talk. See, human beings actually did not evolve at all to be able to look at cells or tissues actually even, because the human eyes evolved essentially to look at objects of similar size during evolution. And so our interest in molecules and cells that we could not see was actually promoted and supported by developments in physics, chemistry, and mathematics, and especially later on in biology when all these three fields contributed to biology. And so if you look at this slide, basically, in summary, if you want to look at proteins and complexes and nucleic acids, etc., how they interact at atomic level, you have a few techniques here which are shown here in X-ray crystallography and single particle electron microscopy, which has come up more recently. These are the techniques that you need to use really to be able to reveal how nature has worked, because our eyes are not good enough for this. And so this, you really need to rely on developments in physics, etc. So most of this talk is going to be based upon data that we've generated using X-ray crystallography to reveal how some of the proteins might work in malaria and why and how they may be druggable. So the talk is going to be focused on malaria, but there are bigger issues. I think the talk will have, I hope, has some implications on other pathogenic diseases which are related to malaria, and I will discuss them very briefly. I think part of the reason the talk has been made in a sort of a general format is to drive home the point that some of the enzyme families that we've been looking at for the past decade are in fact of great interest, not only for malaria, but for many related infectious diseases. And I hope I'll persuade you about that by showing you some data. But we all know about malaria. We are not going to discuss this at length for the moment. And we know some of the magical drugs in the past, which were both plant-based, that have actually saved literally millions of lives across the world. And so we have to keep this in context of new drugs that we may discover that in principle will work in combination with either artemisinin or if and when required with chloroquine. So we will be, the rest of the talk will be specifically focusing on specific protein structures from the malaria parasite. The enzyme family that I'm going to discuss very shortly, it spans a very vast evolutionary time. In other words, it's actually, this enzyme family is ubiquitous. All cells need these enzymes, and there are no cells that don't have these enzymes. And therefore, they are essential part of life and cannot be done away with. So the fact that these enzymes are conserved throughout evolution immediately opens up this applicability of these studies to wider systems, as I discussed briefly. And those wider systems in this context would be pathogen-driven diseases. And all of this work, the structural biology tools that I will show you shortly have been central. So I mentioned to you that the main focus is malaria, but what's significant is that some of the lessons we've learned from this work in malaria can be easily applied and has been applied to other related diseases like cryptosporidiasis, schistosomiasis, leishmaniasis, and also toxoplasmosis. And so there are wider implications looking at these enzyme families because they have autologous proteins naturally in other infectious diseases, and therefore whatever we learn from malaria can in some cases be extrapolated and can in fact be useful for targeting these related pathogens. So I will introduce this particular enzyme family and then give you the malaria parasite perspective on how much we've been able to understand a few of them. So the specific family is called amino acid tRNA synthetases, and they do a very important job. These enzymes put together amino acids and tRNA, and these specific tRNAs, which are then charged with amino acids, then go to the ribosome to complete protein synthesis. In other words, protein synthesis is not possible if even one of these enzymes stops to work, because the whole machinery will be stalled when the ribosome is generating protein-based is doing protein-based protein synthesis, then it will require, for example, in this case, let's say the next residue in the protein chain is a tryptophan. However, if the enzyme that is able to generate charged tRNA responsible for tryptophan charging hasn't worked, the proteins will stall, and therefore you'll have aberrant proteins, misfolded proteins generated in the cell, and the cell will eventually die. So these enzymes are ancient. They are very important because they do this critical step that allows translation of proteins, and they're highly conserved. But on the other hand, each of them recognizes a different amino acid. So very loosely speaking, on the whole, there are 20 amino acids. And so you expect at least 20 of these enzymes, each for one amino acid. What makes these enzymes really interesting from a structural perspective, and this is work that's been done over almost 3 decades, much before we got into this field, was that these enzymes are rather large given the sizes of usual enzymes that bind to amino acids or to ATP, etc. And these enzymes have lots of cavities, sites, and pockets in them, basically for binding the substrates. But also there are other pockets and cavities whose purpose has not been clear throughout evolution. So what we see is high evolutionary divergence in the structures of these proteins as well as in their sequences. And of course, the sequences diverge. That's exactly why the structures diverge. But no matter how much the divergence is, each cell must have at least 20, and in some cases up to 40 or more, distinct tRNA synthetases, because as I mentioned, each of the amino acids needs one per amino acid. And over the decades, there's been a lot of structural information that's been gathered on bacterial and eukaryotic tRNA synthetases, including from our lab. And there have been a huge number of studies that have been already published showing how natural inhibitors, some plant-based and others, or fungal-based inhibitors can initiate drug development against this enzyme family. And I'm going to give you in this talk 3 examples of this from work in our lab. So this slide essentially summarizes in a cartoon fashion some of the compounds that we'll be discussing very briefly. So what's shown in orange is a tRNA synthetase enzyme, and what's shown in this clouded sort of subpockets are the substrates that these enzymes bind. So they all bind to an amino acid, a particular amino acid, ATP molecule, to tRNA, and in some cases they have subsites whose functions aren't clear at the moment. And in some cases, the enzymes have editing sites, as in when these enzymes make a mistake and put the wrong amino acid or a wrong chirality of amino acid, then these enzymes are able to fix or correct the error themselves. And what's shown on the right-hand side are some of the molecules that are actually quite advanced in terms of, as much as we understand, in terms of their anti-malarial potential. So we are going to discuss in this talk cladosporin, we are going to discuss halifignon, and we are going to discuss this enzyme family that's been targeted by azitidine, the FRS family. We are not going to discuss berylidine and this AN6426 compound because one of the latter ones is already out in the market as an antifungal, and berylidine is specifically something that we are working on, but that's of great interest, but it would be too long to discuss that for the moment. So we'll be discussing 3 specific molecules that we've been working on for the past 10 years. But before we do that, because there is a general interest in understanding how malaria parasites are able to do the protein synthesis very well. As you know, during the erythrocytic stages of the malaria parasite life cycle, these parasites are resident in your red blood cells and are therefore their occupancy of your red blood cells is the reason whenever the red cell bursts, the infected red cell bursts, you get feverish symptoms, etc. And all the pathogenesis that's been associated with malaria is essentially associated with infected red blood cells. So inside the infected red blood cell sits a parasite. Now this parasite is very interesting because unlike say even the human cell, it has in fact 3 protein translation compartments. So it has to do protein translation in its cytoplasm, in its mitochondria, which is common for all eukaryotic cells, but also it has a special organelle, which is a plant-like plastic hangover from when these parasites probably ate some bacteria. So this specific organelle is called apicoplast. So you have here a situation where there are 3 independent protein translational compartments, and you actually need 60 enzymes for that if you were to do the math, 20 enzymes multiplied by 3 compartments, but that's not the case. And it's taken us almost a decade of work, and there are all the papers or most of the papers that we've published on this are listed here over the past decade. It's taken us, and of course, many other labs have been doing this as well. It's taken us around 10 years to understand that the enzyme, 36 enzymes from the P. falciparum family do the job of 60, basically by dual localization of single copy tRNA synthetases, by tRNA trafficking, etc. I won't dwell on the details, but suffice to say that the parasite has found clever mechanisms to be able to supply charged tRNAs to all the translational compartments. And that's obviously self-evident because the parasites survive very well. But it was interesting what are the mechanisms of sharing the resources. You know, as I said, 36 have to do the job of 60. So a couple of years ago, when I was going through this really incredible book on periodic table and all the politics, scientific politics that went on and probably still goes on in terms of discovery of new elements, I thought that it'll be really nice to be able to generate a periodic table of the tRNA synthetases from the malaria parasite. And there it is in front of you. This is basically the 36 enzymes that P. falciparum encodes. They are color coded because the ones in epicoplast are in orange. Mitochondrial one, there's only one, partly localized there is FRS. There's cytoplasmic enzymes shown in green, and then there are dual enzymes, which are as in they are localized to the epicoplast and the cytoplasm, and they're shown in yellow. The point is that this sort of a periodic table information depiction makes it really easy to be able to focus on specific enzymes. And for the rest of this talk, we'll be actually focusing on 3 enzymes from this green, from these green boxes shown here. Another fun part about these enzymes is, or in fact, I should say, it's easy to target these enzymes in the sense of doing screening. And the reason is that the amino acylation assays, you know, when you want to discover new drugs, the enzymes that you want to target, you have to have robust assays so that you can actually check whether the compounds are able to bind and inhibit them or not. There are very robust assays. So there is one specific assay called malachite green assay, which actually detects the released, degradation of pyrophosphate. And then there's second assay, which is an AMP detection assay, which indicates when the charged, when the tRNA has been charged with a particular amino acid. So here's the tRNA, here's the amino acid. And when this enzyme leaves behind this product, which is a charged tRNA shown here, it also leaves behind an AMP molecule, and there's a detection kit that just picks up AMP. So for every molecule of charged tRNA made, a molecule of AMP is made. And so that, and it's ELISA-based assay. This makes it very easy to then screen a large number of compounds. And so using this and many other tools, in fact, I'm actually giving you the whole mystery of this talk. What has been shown over the years is that the specific molecules that we're going to discuss, for example, tiodosporin, halofignon, and azitatine compounds, they in fact target different sites within the same tRNA synthetases. So tiodosporin goes to the ATP pocket, halofignon goes to tRNA binding pocket as well as an amino acid pocket. And azitatine, as we very recently shown, this paper came out a couple of weeks ago, goes to the amino acid pocket and to a subsite which was not discovered earlier. So I'll give you the examples, just a little more detail on these particular compounds. Halofignon is the first one. In fact, it's actually, its parental compound is called fabrifagine. It comes from a Chinese herb. And as many things in China that have been discovered and have been of great public health for the rest of humanity, fabrifagine also has been used over thousands of years for malaria-induced fever and for other symptoms. Because of its some side effects in terms of causing gut dysbiosis, its halogenated versions were made, and these are actually being very actively explored not only for malaria but for cancer therapy as well, because the same compounds inhibit the human ortholog as well. And what we were able to show is that these compounds, in fact, halofignon shows a very interesting mechanism of action. It's in fact an ATP-dependent dual site inhibitor. So you remember ATP is a natural substrate for these enzymes. But so I'll just very, very briefly show you one piece of data. You can see here on top and side the thermal melting curve. So the enzyme alone has a melting curve, well, TM of 50 degrees or so. And when you have the drug bound, you have it goes up very substantially to 65. But when you have ATP or its analog or non-hydrolyzable analog bound, it goes up till 90 degrees. That means ATP or the natural substrate aids the binding of this particular drug, which is quite incredible. So that's been shown and proven both for the human enzyme as well as for the parasite enzyme. So now we understand from data from the past many years that helifignon in fact goes to, in blue is shown the tRNA synthetase site. Helifignon goes to the tRNA binding site. One moiety of the drug goes here. The second moiety goes and kicks out proline. So this is specifically the proline tRNA synthetase. Proline is a substrate for this particular enzyme. It kicks out the proline, clearly has higher affinity for this enzyme than proline does. And by virtue of occupying two sites, therefore, it is able to block the enzyme activity and therefore stall protein synthesis and leading to cell death, in this case, the death of the parasite. Of course, ATP still continues to bind to this enzyme. And in fact, ATP binding to the enzyme in the presence of drug, sort of promotes the binding of the drug because there are specific hydrogen bonding interactions that happen between the drug and the ATP molecule. So this was quite striking in its mechanism of action. You have to keep this in mind because we'll be also discussing shortly other mechanisms of action of these particular series of compounds that I'm going to show you. So the second example that, again, very briefly, I'm going to give you is of cladosporin, which is again a natural product. It's actually made by fungus. The previous one was a plant-based product. This is a fungal product. And the beauty of this product is that it kills malaria parasites, as shown here in the lower panel. When you add it in the plus, under the plus, when you add it, the parasites fail to progress through their erythrocytic life cycle and are stalled, whereas in the absence of that, the parasites go from rings to schizons over a period of 48 hours, as they should. And it was shown earlier that this compound in fact inhibits protein synthesis, which is the straightforward assays to show that the process that any compound is inhibiting. But what is specific about this compound and what's really incredible is the fact that this compound is able to kill the malaria parasite enzyme with really very exquisite specificity. And it's, sort of the IC50 values are between 10 and 30 nanomolar, depending on who's done the enzymology, but between the range of 10 to 30, which is very potent. But this enzyme, the human version of this enzyme is never touched by this compound. So remember that both the lysine tRNA synthetase from the human and the parasite do exactly the same job. They do exactly the job of charging tRNAs with lysine. They are absolutely identical in their functional significance, and yet in sequences they diverge, and therefore perhaps one of the reasons we see the difference in selectivity is because the sequences have diverged. So this is convergence in the sense of the functional convergence, but sequence-based sequence divergence. What we were able to show was that this enzyme family, lysine tRNA synthetases, they bind to cladosporin at the expense of ATP. So if you measure the KT value or the binding constant for ATP alone, it comes in micromolars, say some odd 4 micromolars or so. But when you measure the KT value of cladosporin for this enzyme, it is in the range of 10 to 14 nanomolar. And so you can see here that there's almost a 300-fold higher affinity between the drug and the enzyme over the substrate. And so we know now that this compound is actually an ATP site competitor. But what's more interesting is that if you look at some sequences, and it's really always good to look at protein sequences, not just at structures, you can see here that in the top panel here, what I've shown you are 3 in black, Homo sapiens and 2 fungi. And you can see these specific residues labeled here are Q and T. But when you look at P. falciparum, these residues are V and S. And these V and S residues are really critical because from our structural analysis, what we've shown is that what's shown in the lower panel here are the V and S residues that in cases where you have bigger than V or S, which is of course Q and T, in those cases, the drug doesn't bind as well. In fact, can bind really poorly, and therefore it won't be a drug. And so that's okay. That explains the specificity for the human, for the parasite enzyme over the human enzyme, but doesn't explain other things. But one of the things it clearly tells you is the fact that wherever V and S are conserved, it's possible that this compound will also bind to the particular lysine tRNA synthetase. And in fact, we've shown this over the past 2 to 3 years that this enzymes from Schistosoma mansoni and Loa loa, which are also pathogens, and some of the other pathogens here, Tutsi, Vivax, etc., can also be inhibited with equal potency by the same compound by virtue of the fact that their VNS are conserved. And we've come up with a framework to explain this, and I'll come back to that a little later. The third brief example that I'm going to give you is some work that has just come out from our labs. And that is this particular compound, which is the azitidine series, which is shown here, azitidine-based compounds, which were shown to have extremely high affinity for the malaria parasite enzyme, in this case, phenylalanine tRNA synthetase, whose structure is shown here in the bottom on the left-hand side. It's a complex heterodimeric structure that, well, it's actually a pseudo tetramer. So alpha beta 2 is the overall structure of this FRS molecule. And what we were able to show over the past 5 years or so is that this compound actually goes and binds to this enzyme and kicks out the phenylalanine, the amino acid, and part of the drug goes to an accessory pocket or a subsite that I had discussed with you earlier. Again, completely unexpected results. These particular series of compounds are able to inhibit the malaria enzyme very specifically. The KD values are in nanomolars, their IC50s are in nanomolars, and their selectivity index ranges from 500 to 1,000. So they are really specific compounds, and we think we are beginning to understand why these compounds are selective, although that work is still ongoing. But in a related effort, as I mentioned, because there's a conservation in the active sites in pathogenic enzymes, in this particular case as well, to show that the same set of compounds in this paper with Vinayak et al., we showed that the same set of compounds are able to inhibit Cryptosporidium as well, which is a related parasite. And that goes back to the point that I made earlier, that there is sequence conservation as long as there is selectivity between the human system and the pathogenic system, and there is conservation within the pathogenic sequences. There is a possibility to use one compound to multi-target enzymes through several diseases. So we'll come back to this. We've come back to this slide. I'm repeating it because I've shown you very briefly how cladosporin, halofignone, and azitidine work based on structural data as well as enzymology data, and how they may be selective against, selective for the parasite enzyme and selective against the human or the host enzyme, which is what you would like in an antimalarial drug. So the question is now, what makes this a little more interesting for me as well is the fact that these enzymes have so many subsites. So if you think about this very trivially, if you had only one site or one subsite or whatever you want to call it, there'll be only one possibility for a compound to go and bind there. If you had two sites, the possibilities increase. You can have three options. You can have a compound going to each of the two sites, or you can have a compound going to two sites at the same time. As you have greater number of subsites here, 3, you can see that the combinations go up to 7. When you have 4, you have 15 possibilities. And so this really opens up a huge gamut of possibilities, a whole new universe of multi-targeting the same subsites in an enzyme with basically copy-pasting scaffolds from molecules. And so let me show you an example of that by what I mean. About 2 years ago, in fact, this analysis is done earlier, but 2 years ago, this paper came out from our lab where we thought that it may be possible to do multiple druggable sites within the same protein using different compounds. And the reason we thought that was because, as I showed you in the previous slide, an enzyme like proline tRNA synthetase has several subsites, which are shown here in various colors, yellow, green, purple, and orange, if you like. And you can see here that these are allosteric pockets, and there's an ATP site, and there is a amino acid as well as a tRNA binding site. So the question is, why can't you put two drugs in one enzyme as opposed to one drug going to two enzymes, which is also possible. So we took this upon. As far as I know, I may be mistaken, but as far as I know, there is no other example of this so far, and this work will be going out for publication soon. But so we've actually been successful in dual drugging this particular enzyme, and what's shown here is we're talking about halofignone and a compound that Takeda Pharmaceuticals has shared with us, 595, which is also a very potent compound. These compounds independently bind to the enzyme and inhibit the enzyme as well as the parasite in nanomolar affinities. So they are really good compounds in their own right. But we asked the question, given that they go to different subsites, why can't we mix them? And in fact, when we mix them, this is what you see. This is an electron density map for the two drugs, and here's an electron density map of the two drugs. And in the background is showing the protein structure. Dr. Varatharajan might appreciate that immediately, that this binding is very nice. And so the point is that it's possible, I think, going forward, that you may be able to drug multiple sites with multiple compounds in the same enzyme. So you don't need to do target hopping. So we've sort of loosely called these ideas STOP, which is also going to be very soon the stoppage of my presentation. But STOP stands for structure-based targeting of orthologous parasite proteins. And so here are some of the examples from published literature, not only from our labs, but we've collated data from other groups as well, where, for example, here we've shown you cladosporin can go to multiple pathogens, Falciparum, Schistosoma mansoni, and Loa. We know that the leucine tRNA synthetase compounds, this AN series, this work was done by groups in France, targets malaria and toxoplasma and candida. And another group from the US has shown that specific compounds from this series, GNF series, are able to go and hit not only the protein translation machinery as I've shown earlier, but protein degradation machinery, in this case, the 20S proteasome machinery. And they've shown that this set of compounds are actually quite potent against these related pathogens, all three of them being human pathogens. So I think STOP is a good strategy if you're interested in infectious disease drug targeting, as long as there is some conservation in the function and divergence in sequence. And I think I'll end there by thanking really a large number of people who have been in the group. But for the moment, there are my lab members are shown here who are in ICGB, and some of them are here in NIMR now with me. And over the past decade, we've been funded really very heavily and very positively, very constructively by DBT. But we've also had a huge amount of funding from MHB, Medicines for Malaria Venture, from the Wellcome Trust, from GHIT Fund, which we have presently. We have very active collaboration with Harvard and Broad Institute, Washington University, with groups in Japan who are really state-of-the-art medicinal chemists, and Takeda Pharmaceutical Company, as well as we have very close links and very fruitful collaboration with CSR lab in NCL Pune, who have been really doing very nice work with us on cladosporin-based compounds that I didn't discuss for the moment to save time. So I'd like to thank you for your attention, and again, really privileged to have this opportunity to discuss my work. Thank you.
Amit, that was a very fascinating talk, and at least 3 points I will just sort of briefly highlight. One is that you have shown beautiful visualizations of the interactions between compounds and the target enzyme, the amino acids tRNA synthetases. Second point, which I was very impressed by, was that you have a potential for the HF molecule, if I'm correct, targeting the lysine tRNA synthetases of multiple pathogens. So you can have almost like a broad spectrum anti-parasitic molecule. And the third thing which I found again very fascinating was your targeting using multi-drug therapy, using a single enzyme target, because you are able to target multiple subsites on an enzyme. So really, thank you very much. If there are any questions, I urge you to kindly request you to please put it in the chat box. I'm sure Amit would be more than happy to answer. And I believe it is now time to also make the formal award to you. And I think Santosh is going to be projecting it on the screen. Unfortunately, these are very unique times when the award also has to be given in a very novel way. So, but believe us, our good wishes are with you, and I think he's projecting the citation on the screen at this point of time. So, congratulations, Dr. Sharma, for this MGK Benin Lecture Award 2020. There you go. There's the citation. It is being conferred on you. And one more award in your long list of awards, and perhaps many more to come. Congratulations very much.
Thank you, madam. Thank you, madam. Just I want to also share here
Thank you, Dr.
that
Jari. Thank you very much, and thank you to NASI for again another honor which I'm absolutely floored with.
Sir, we want to share here that for this lecture award, the awardee will be given a medal and citation with the cash award. Because this is earlier we organized this function in a very grand way, but because now we are in virtual mode, so the medal, citation, and cash award will be sent to you at your address, sir. And on behalf of NASI, we again, sir, congratulate you and thanks for receiving this award. Thank you very much, sir.
Thank you. Thank you very much, Dr. Santosh, for filling in on the details of the award. And now I request my co-chair, Dr. Paramjit Khurana, to kindly continue with the proceedings of this first session. So over to you, Dr. Paramjit Khurana.
Thank you so much, Jaya, for this opportunity. We have had some brilliant expositions on various aspects of serobiology and keeping with the continuity of the
Louder.
protein conformation. Hello, am I audible?
Yeah.
So I said we've had some excellent expositions on serosurveys and protein engineering for malarial parasite. And keeping the protein theme in continuity, it's my proud privilege to invite Dr. Vardarajan Raghavan, who is a very brilliant Indian biophysicist working at the Indian Institute of Science. He did his PhD from Stanford and is currently one of the vice presidents of the Indian Academy of Science. But most importantly, I think he is known for his work in the field of protein structure and folding. His special contributions have been in developing vaccines and drugs for treating a fatal type of influenza and HIV. And very recently, there have been, I understand that they have also initiated work on immunogens derived from the spike glycoprotein of the SARS-CoV-2, the causative agent of this COVID-19 pandemic, and the reason why we are here in a virtual mode. Dr. Vardarajan is a very brilliant scientist who has been conferred not only the Shanti Sarup Prize for Science and Technology, one of the highest science awards in 2002, for his contributions to biological sciences, but he's also the J.C. Bose Fellow and Fellow of not only NASI, but INSA and the Indian Academy of Sciences. So it's a pleasure to welcome you, Dr. Vardarajan, to this, and we look forward to your presentation.
Thank you very much for the kind introduction, Professor Khurana. And also, I would like to thank Dr. Manju Sharma for the opportunity to present over here. Now, without further ado, let me start my talk. Can people see my screen?
Yeah, the screen is audible. You can make it in the presentation. Yeah.
Okay. So I'll just give a general overview of COVID-19 vaccines and briefly touch upon a little work that we have done. So there are, a huge number of vaccines that are in development. And as of date, there are 11 which are approved in one geography or the other. And these include 2 mRNA vaccines. These are by BioNTech and Moderna. 3 non-replicating viral vectors vaccines, 5 inactivated virus vaccines, and 1 peptide-based vaccine. And in India, approximately 12 million people have received at least 1 dose of the vaccine after 40 days since the program was rolled out. So, of course, we have a way to go. Now, I've just mentioned on this slide the various different kinds of vaccines, but really the significant part is just the summary that's highlighted over here. Okay, now the way that the primary target of current vaccines in development, the vast majority of them, if not all, is the spike protein of the SARS-CoV-2 virus, which is the causative agent of COVID-19. So the spike protein is the major surface protein of the virus, and what it does is to bind to the ACE2 receptor on its target cells, and this initiates the process of entry. And ultimately, there are conformational changes which are triggered by proteolytic cleavage, which lead to fusion of the viral and host membranes and entry of the virus into the cell. So If we can block this initial entry process, then we would be stopping the infection process. And so antibodies that block this entry are called neutralizing antibodies. And the major goal of a vaccine is to elicit high titers of such neutralizing antibodies. So, vaccines, as you all know, are given to healthy people, and they seek to elicit protective responses in the absence of infection. Typically, I request others to please switch off their.
You know everything about NASI and this thing. Or Kasturi Rangan they carry.
Okay, so these B cells, which are antibody producing cells, they make antibodies that can block viral entry. And they can also target infected cells which display viral proteins on their surface. Whereas the cytotoxic T cells identify and destroy virus-infected cells. And so far, the available data suggests that neutralizing antibodies are the clearest correlate of protection against the SARS-CoV-2 infection. Of course, the objective of a vaccine is to produce long-lived memory B and T cells that can recognize the virus and then provide long-lasting immunity. So this is a summary of existing clinical trial data. As I said, there are data for these basically 4 different kinds of vaccine modalities. And the important points are that the titers, the quantities of neutralizing antibodies, which have been elicited by the vaccines in humans, are similar to those which the same vaccines have elicited in small animals. And these have been low both for the inactivated virus and the adenoviral vectors, which are the constituents of the vaccines we have in our country. They're better for the mRNA vaccines, and they're highest so far for the subunit vaccine. And overall, higher neutralizing antibody titers are associated with higher protection. Now, one issue of some concern is that in different parts of the world, especially Brazil and South Africa, there have been mutant strains of the virus which have acquired a large number of mutations, several of them in the spike protein itself. And the Oxford AstraZeneca, that is the Covishield vaccine equivalent, was recently, that rollout was halted in South Africa because of limited response against the local strain. The good news is that relatively low titers of neutralizing antibodies are sufficient to protect non-human primates from severe lung pathology. And although we don't know the detailed correlates of protection, but nevertheless, neutralizing antibodies are likely to be very important. Finally, all the current formulations require refrigeration, and especially for the mRNA vaccines, very ultra-low temperature storage prior to the vaccine administration. So, again, this is just recaps the different kinds of vaccines. Inactivated live attenuated, we don't really currently have licensed vaccine for COVID-19 for this, and the protein subunit vaccines, and of course the mRNA vaccines. Okay. Now, just a brief recap of some of the early preclinical, the early clinical trial data. So the Covishield vaccine was given in an initial phase 1 to trial to about 1,000 healthy adults. And the good thing is that it did elicit neutralizing antibodies. However, the titers of neutralizing antibodies were relatively low. And they were roughly comparable to those which are seen in human convalescent sera. So people recovering from infection. Okay. So these are the titers in recovering people, and these are the titers in the AstraZeneca vaccines. I'll also focus a bit on the Covaxin. That's an inactivated virus developed by Bharat Biotech. And there's published data available very recently in Lancet Infectious Disease on a phase 2 clinical trial. And there again, there were, it produced a Th1 biased immune response, which is desirable for a vaccine to avoid immunopathology, and it appeared to be in this small study safe and well tolerated. So the adjuvant that's used is relatively new. When the neutralizing titers once again were approximately comparable to those in convalescent serum samples, and the titers persisted to similar levels 2 months after their last immunization. So the good news is that both vaccination and in convalescent sera, the antibody titers last at least for several months. Initially, there was concern that they were behaving atypically, but there's no current evidence for that. Okay. Now, let me get back to some of the issues of concern, which are these variants that I talked about. So, namely this UK variant B1.1.7. These are all the different mutations it has in the spike protein, and this is the South Africa variant. And one of the mutations, the N501Y, is common to both the variants. So this is a summary of the existing neutralization data that's available in the literature. For the UK variant, the neutralization titers were by and large unchanged in convalescent sera, as well as, so sorry, this should be also convalescent sera, as well as sera from the Pfizer and Moderna mRNA vaccine recipients. Whereas with the South African variant, there was on average about a 10-fold drop in neutralization titers both in convalescent sera and in the Pfizer Moderna vaccine. So this data is shown here. And a separate study showed a complete loss of neutralization in about 50% of convalescent sera from actually people recovering in South Africa. And so this was one of the reasons that the Covishield vaccine rollout was stopped, because of, I wouldn't say complete lack of efficacy, but certainly a lack of efficacy against mild and moderate cases of COVID-19. So this is a summary of some of the available data. So these are the different vaccine modalities: inactivated viral vector, RNA, and protein. These are the doses which are given. So there are a few micrograms for the inactivated and protein vaccines, and about 10 to the 10, 10 to the 11 viral particles for the viral vectors. Now, these are the neutralizing titers, and it's not very easy to compare these numbers exactly because they all use slightly different assays. And so this is a problem. So what is probably more relevant is to compare the titers with titers in convalescent sera measured by the same assay. And so by these criteria, most of the vaccines produce titers, the inactivated and viral vector vaccines produce titers comparable to convalescent sera, and the RNA and protein vaccines produce titers that are higher than those in convalescent sera. Now, this is the efficacy, and the efficacy also for the same vaccine, they are widely differing efficacy estimates depending upon the measure of efficacy and depending upon the geography in which the efficacies are measured. So a good example is, so with the Novavax vaccine, for example, the efficacy was 95% against the wild type virus, a little lower against the UK variant, and these are preliminary estimates, and about 60% against the South Africa variant. Whereas similarly for the J&J vaccine, again, the efficacy is different in different geographies and lowest in South Africa. Okay. But overall, all these efficacies are above 50%, which is, of course, good news. So overall, there are multiple vaccines with reasonable efficacies, but the emergence of mutant strains is certainly somewhat of a concern. So now I'll touch on a little bit of work in the remaining few minutes that we have done. So all the existing licensed vaccines use the whole spike protein as their primary antigen. We and others focused on a small part of the spike called the receptor binding domain, which binds to the ACE2 receptor. And the reason is that virtually the bulk of neutralizing antibodies target the receptor binding domain. There's a small fraction which targets something called the N-terminal domain, and a single site on the N-terminal domain. This is, of course, much smaller than the spike protein, so it's relatively easier to produce. And so we constructed a glycan engineered receptor binding domain, which we expressed in a number of different systems. The protein was highly thermotolerant, and formulations elicited good titers of receptor competing and neutralizing antibodies. And in the one hamster challenge study that we did, the protein performed relatively well. Okay, so, and very interestingly, the protein was highly thermotolerant. So this is a thermal scan of the protein before and after. This is the first thermal scan. We cool it down, and the second thermal scan. So they exactly overlap with each other. We can take lyophilized protein and expose it to very high temperatures for quite a long time. And at 37 degrees for over a month, without any loss in its thermal transitions, and also in terms in its functional properties. So if we look at the ACE2 binding by SPR, you can see that the lyophilized protein retains ACE2 binding even after quite long storage at 37 degrees. Okay, so it's exceptionally thermotolerant. When we looked in guinea pigs in our initial study, we showed that this elicits receptor competing antibodies which block binding to the ACE2 receptor. And the titers that you get here are roughly equivalent to what you get in a neutralization assay with replication competent virus. Unfortunately, the Pichia produced protein did not give neutralizing antibodies. In mice with a different antigen, we again got good ELISA titers and some degree of neutralization. So if we look at our neutralization titer, this is of course in guinea pigs compared to other vaccine modalities, this is a log scale, we are doing okay. So this was all done last year, and then we carried out a hamster challenge study. These are if we look at the weight, these are the unvaccinated in this sort of magenta color. The ones immunized with our RBD are also gaining weight after challenge, whereas the unimmunized and the spike immunized actually show poorer outcomes. We see that there are the viral load is extremely low in the lungs compared to the control, and the cytokine profile is also quite favorable. If we look at the lungs themselves, this is group G6. It is similar to the G9 unchallenged animals. So overall, these animals were protected from challenge. There was little lung pathology, and they showed negligible lung viral titers and favorable cytokine profiles. So subsequently, we have designed oligomeric versions of the receptor binding domain. And I'm just showing here some data. The most relevant is the pseudoviral neutralization titers. Again, this is a log scale. You can see that the convalescent sera have neutralization titers centering around 100, whereas ours are well over 10,000. Okay. So we are able to elicit neutralizing antibodies about 30 to 300 fold higher than in convalescent sera. So if we compare what we have with existing vaccine modalities, so shown in blue are the mice neutralizing titers, and the more important is shown in peach, the ratio of the mice neutralization titers to those found in human sera in the same neutralization assay in the same study, because it's difficult to compare across studies. So you can see that some of the Moderna and BioNTech vaccines and the Novavax are about 10 to 20-fold higher, this pink, peach bar. Whereas this is the data that we have, and so we are, in mice, the titers that we get are 200-fold higher than what we see in the human convalescent sera. And interestingly, we've also carried out ACE2 competition assays with the receptor binding domains from both the wild type as well as the South Africa triple mutant. And we see that the ACE2 competition assays are completely identical with the sera generated by our oligomeric immunogen. So this is very encouraging news. And we are in the process of carrying out proper viral neutralization assays, as well as hamster and mouse viral challenge studies. But our experience has been that the ACE2 competition assays are very predictive of viral neutralization, because it's essentially the same thing that we are targeting, prevention of viral entry. So I'll just wrap up now. So to summarize, there are 11 COVID vaccines that have been granted approval and authorization in various geographies, and with about close to 16 different clinical trials. And the reported interim efficacies are in the range of 50 to 90%. And recent mutations are a particular concern. And the neutralization titers against the South Africa variant are 10-fold lower than those elicited against the original wild type virus. And convalescent sera also are unable to very well neutralize the South African virus. Now, of course, neutralization is not the only mechanism of protection. And so it is very likely that convalescent individuals have other means of protection which will render them somewhat resistant to these mutant viruses. Now, the current oligomeric formulation that we have developed at IISc, and along with Minvax, the startup that I work with very closely, these are highly immunogenic and thermotolerant, and the neutralization titers were about 20 to 300-fold higher. So 20 in guinea pigs, 300-fold higher in mice, and they showed much better immunogenicity than the currently licensed vaccines when compared in the same animal model. We made permanent cell lines, and importantly, in a surrogate neutralization assay, the titers are identical against both wild type and South African mutants. And because these mutants don't require a cold chain, they, I think, have, because these formulations don't require a cold chain, they likely have a lot of promise to combat COVID-19, which is likely to be with us for some time. So I must most important thing is to acknowledge all the people who have contributed to the work. These are all the people in my own lab who've been working very, very hard over the past 1 year. Samir did a lot of the imaging designs. There's a lot of yeast display that I have talked about. And Dr. Somnath Datta was responsible for all the structural work, a lot of cryo-EM structures. Again, I have not had an opportunity to present. Our early pseudoviral assays were done in collaboration with Professor Stalin Raj at ICER Toronto, and more recently with Dr. Rajesh Renge at CSR in Tech. The viral, with replicative virus, the neutralization assays were done at THSTI by Shailendra Mani and his colleagues, and the hamster challenge was done by Zaidam, who works with Amit Awasthi at THSTI. We have been funded for this work by the Bill and Melinda Gates Foundation. All the animal studies and a lot of the work is done in very close collaboration with Minvax. And without them, we could not have made this kind of progress. And the human sera were obtained, data were provided by Dr. Rajesh Pandey at CSR, IGIB, and Max Super Specialty Hospital. So our initial RPD work was published last year in JBC. It's got a lot of press coverage. This is our more recent work, which some of it we have put out. So I'll stop here now, and thank you all for the opportunity to tell you some of our work.
Thank you very much, Dr. Varatharajan, for a very beautifully presented, simplified version the work which is going on in your lab. I'd love to see if anyone has any questions, so we can address the questions to either this speaker or to the other two speakers as well. I don't know, Jaya, has anyone written any questions? No, I think they're all congratulations in the chat box only.
Yes,
Over
there
to
are
you, Jaya.
specific questions. Yeah, there are no specific questions that I could. Everyone has been very congratulatory and rightly so. Thank you.
I had one clarification to seek from Raghavan.
Yes, sir.
Oligomeric, how many units are there in the oligomer?
We have done a variety of things. We have done trimers as well as nanoparticles. In our experience, a trimer works quite well, and it's from a manufacturing standpoint is much simpler. So
you make the monomer and then oligomerize that.
No. It has an oligomerization domain, and so it spontaneously oligomerizes. It forms very clean oligomers.
Okay, just wanted to get that clarification. Yeah, thank you.
If there are any more questions from it. If not, it is really my pleasure to congratulate first Dr. Varadarajan for a very lucid account of your own work and summarizing for us really the kind of competition that you are facing from across the world. I think congratulations. We are hoping to see more about this oligomeric form of the RBD vaccine. And I also take this opportunity to thank all the three speakers. Very beautiful talks, very simply presented, and from my side, I think it's a big congratulations for all three speakers. And Paramjit, would you like to think before we actually formally close the session? Over to you.
I think it was a wonderful exposition. We had a very nice survey, the Sero survey, giving us a holistic picture. And when Amit spoke about his work, how multiple drugs could be targeting the same subsite and also affecting different organisms also simultaneously, I think that was an eye-opener for me at least. And I thoroughly enjoyed the last lecture also. I mean, I was constantly wondering what is the basis of this thermotolerance, because it scared me a little bit also. But I think I'm very happy with the way things are working, and I look forward to more work coming from their labs. I'm sure in the coming years we will be seeing much more positive work both from NASI as well as NIMR and the other work also. Congratulations to all of you.
So thank you then, and I really hand over to Dr. Neeraj and Dr. Santosh to carry on with the proceedings of this annual meeting. So over to the headquarters of NASI.
Thank you, madam. Thank you very much. And generally, we should have felicitate to our chair, co-chair, and the speakers. But we could not possible to this felicitation. But I want to share my screen as well as also on behalf of NASI. We want to share heartfelt gratitude and thanks to the chair, co-chair, especially Professor Jaya Tyagi madam and Khurana madam, who are always kind enough to have with us if any type of need and any help for guidance we need. You always kindly to guide us, madam. We are thankful to you, as well as we are very much thankful to the speakers, Dr. Manoj Morhekar sir, Dr. Amit P. Sharma sir, and Dr. Raghavan Varadarajan sir, who has enlightened so much information to this our new young generations and given a lot of things to go forward for coming future in their careers. We are very much thankful to all of you, and we, with the permission of the organizing Chairperson, Honorable Madam, I think Madam is here with us. And we could announce for the lunch break or how much time, Madam, we want to ask you. I hope Madam is also with us, Professor Manju Sharma, Madam.
Actually, according to the program, if I may just add one point, according to
Yeah,
the
madam.
program, the session is supposed to start at 2 o'clock. So I don't know whether all the speakers are already there. I do not know that.
So that we can announce a lunch break for that, and we will meet here again after at 2 p.m. I think so.
Yeah, perhaps that would be right.
So,
Yeah.
once again, many thanks to you and all the participants as well as the IVC persons. Thank you very much, madam. And we will be here at 2 p.m. But the link is on.
Santosh,
Yeah, madam.
do it earlier. I mean, why should we waste time, na? So instead of 2 o'clock, let's have it at 1:45.
Okay, madam. But we will let me contact with the Sanjeev Sinha sir and the co-chair. If they will be here, we will start as early as possible, madam.
At 1:45. Huh?
Okay. So we will accompany here. We will assemble here and join. Everybody requested to kindly be with here at 1:45.
Yeah.
Okay.
Thank you, madam. Thank you, madam.
Karo band karo.
Now, welcome to all of you in the post-lunch session of the Symposium of NASI on towards a new healthcare regime for the nation. Now, this session is chaired by Dr. Sanjeev Sinha, who is Professor of Medicine at All India Institute of Medical Sciences at New Delhi and an eminent physician and excellent researcher in the field of pulmonary medicine and infectious diseases. And this session is co-chaired by Dr. Padma Priyadarshini. The speakers of this session are Dr. C.S. Pramesh, Dr. Sangmitra Pati, Madam, and Professor Priya Abraham. Now I request Dr. Sanjeev Sinha sir to start the session. So please.
Thank you very much for inviting me. First of all, I would like to thank NASI for invitation. And can I please invite to Professor Priya Abraham? Dr. Priya Abraham is director in ICMR National Institute of Virology at Pune. And our topic for lecture is COVID-19 pandemic scenario research, prevention and control. So, Dr. Priya Abraham, please. Is Dr. Priya there?
May I request Priya Abraham, madam? Are you listening, madam?
सबको भेज दिया?
जी मैम, कंप्लीट। अर्चना मैम को सब भेज।
नमस्कार डॉक्टर साहब। नमस्कार।
Hello. May I audible, sir? Sinha, sir. Sir, you are unmuted, sir.
Yeah.
Sir, can we request to Sangmitra Pati, madam, because she is available here. Firstly, we can deliver the talk, then we can contact to other speakers, sir.
Okay. So I would like to invite first to
Sangmitra Pati.
Dr. Pati. She is director in ICMR Regional Medical Research Center at Bhubaneswar. And it's my pleasure to invite her, and her topic for talk is magnitude and outcomes of multimorbidity landscaping in terrain. So, Dr.
Pati, madam.
Pati, please.
Madam, are you available, madam?
If she is not, then Pramesh can come.
Professor Priya is there, I think, Doctor.
Ma'am, if you like, I can give my talk. Good afternoon to you, ma'am. Good afternoon to you, sir.
Thank you very much. You're welcome. Please.
Shall I give the talk, sir?
Yeah, please.
Okay.
Just a minute, let me just check the program. Oh, Dr. Sinha,
Yes.
your session is being co-chaired by Dr. Padma Priyadarshini. Are you there, ma'am? Dr. Padma Priyadarshini? No. Then Pramesh is here. Dr. Pramesh?
Yes, Dr. Sharma, I'm here.
Yeah. So, Professor, it has to be after him.
Sure,
So,
sure. I
Pramesh,
understand.
so Dr. Sinha, you can start with Pramesh.
Okay, ma'am. Okay, I'll do that. So good afternoon to everyone again. I'm Dr. Sinha here. And first of all, I would like to invite Professor Dr. C.S. Ramesh. He is director in Tata Memorial Hospital at Mumbai. And it's my pleasure to invite him. And his title for today's talk is Furthering Cancer Research, the National Cancer Grid Initiative. I would like to invite Dr. Ramesh for his talk. Thank you.
Thank you very much. I'll take this opportunity to thank the organizers, Professor Sharma, and the other organizers for this opportunity, and the chairs for the kind introduction. I'll just beginning to share my screen. So is my screen visible to you?
Yeah, it is visible.
Yes.
Thank you. So over the next few minutes, I thought I'll share
Maybe you can go into the presentation mode.
Yes, so I have done it into the presentation mode. I think there'll be a bit of lag in the display. So has it come into the presentation mode now?
Yeah.
Not yet, at least not for me.
Not yet.
So I've done that again. Is it on the presentation mode now?
We can read. Actually, it is not on presentation mode, but I think you may proceed.
Okay. Are the slides moving at least? I've gone on to the next slide. Okay, great. So thank you. Over the next few minutes, I thought I'll give an overall overview of what the challenges are in cancer research in India and how we have within the National Cancer Grid attempted to address these. So this was a study that was done at the Tata Memorial Hospital almost 12 years back, sometime in 2008, where we looked at what proportion of our patients who were eligible to receive a targeted therapy for breast cancer were actually receiving the treatment. So we looked at 440 patients who were HER2 positive, which meant that they would have benefited to a large extent by this drug called trastuzumab. And what we found was that out of the 440, only 38 had received the drug. And out of these, roughly half of them received it because they were part of a clinical trial, and the remaining half were the patients who were able to actually afford it. So though this was a blockbuster drug in breast cancer, the reality of the fact was that only 4% of India's patients were able to afford the drug. And it was primarily because of cost that they were not able to afford it. So what I'll cover here is how much has cancer research improved outcomes in general? What does it cost? I'll make this distinction between value versus cost, and what should we as an LMIC do, and our own approach within the National Cancer Grid. So when you speak to cancer physicians across the world about how much cancer research has improved outcomes, you're very frequently met with curves or graphs like this. This was a clear blockbuster, imatinib in chronic myeloid leukemia, which showed close to 90% long-term survival with just taking a pill every day. So this was the beginning of targeted therapy, and it revolutionized the way we treat cancer, at least in some subtypes. We also have more recent examples of this. This is a very uncommon type of lung cancer, but still seen in 4 to 5% of patients with lung cancer, which is the EML4 ALK translocation. And the drug called crizotinib showed dramatic responses in the way patients responded to this drug. There are several other examples. This is a very good example because metastatic melanoma had a very dismal prognosis prior to the advent of immunotherapy and checkpoint inhibitors. But very soon we realized that even with one drug, you could reach 3-year survivals of 34%, and with the combination, reach even close to 60% survival, which was something unheard of with metastatic melanoma. A more recent example is the use of this targeted tyrosine kinase inhibitor called osimertinib in non-small cell lung cancer. And again, very wide differences in survivals between patients who received the drug and those who did not. But all this has come at a cost. And very similar to the breast cancer example with trastuzumab, which I mentioned, there is this tension between cost and value, and these don't always move in the same direction. So we looked at FDA approved drugs and the monthly and the median cost of these drugs. And from 1970 till 2020, we've had a steady increase in the cost of these drugs. And for those of you who might think that this is probably okay adjusted for inflation, this is not a linear curve. What you can see on the y-axis is actually a logarithmic scale, which means that what cost $100 in 1970 actually ended up costing $10,000 by 2020. This was the median monthly price for cancer. We also look at cost per quality adjusted life years, and as you can see with these several examples of monoclonal antibodies for several cancers, the cost per quality adjusted life year ranges anywhere from $250,000 to $1,200 or over $1.2 million dollars. And this is something that is
Dr. Ramesh, excuse me, please, your slides are seeing only on this side. Please make it on the whole screen. I want to see it a little better.
Okay, it is, at least on my screen, it is on the whole screen, but I'll probably try again if it is not clearly visible.
Now it is.
So are you able to see the whole slide now?
Yeah, now we can see it.
So probably I'll continue with this mode itself, not on a slideshow. I'll continue on this mode. So clearly the cost of these drugs were unaffordable for many. And this brings us to what value is. And this quote from Warren Buffett is very apt, because price is what you pay, but value is what you get. And I think several of those examples illustrated that. So we did a study to look at whether the established value frameworks, and there were two value frameworks which are well established in literature. One is by the American Society of Clinical Oncology, and the second by the European Society of Medical Oncology, which look at value and see whether the cost of a drug is worth the survival benefit that it provides. So this is a complicated graph, but just to mention that the blue bars are the ones which did not meet the benefit threshold of ESMO, which is one of the societies, whereas the red bars are the ones which meet the benefit threshold, meaning that they are more beneficial and they are value for money. The height of these bars is proportional to the ASCO score. So as you can see from this, there is virtually no correlation between these which are well established in literature. So even our ways of measuring value are not very well measured. But what we found in our study was something even more surprising. When you plot cost of a drug to the benefit, you actually had a negative correlation between the two. This is completely counterintuitive. And as this graph on the right shows, the cost of something which was found to be beneficial was actually 1/3 of the cost of something which was not found to be beneficial. So this is completely counterintuitive, but this was the actual truth of what we found. So it clearly shows that what breakthroughs we were achieving in cancer research were not actually something that was translatable into the real life scenario. So in a country like India, where traditionally we've spent under 1.5% of our gross GDP on healthcare, what do? Do we take the same approach that was taken in the West, or do we promote clinically relevant research which is contextually important to India? And secondly, how do we translate research into policy? So these comes back to 2 or 3 main points, which is what are the steps through which we go through to translating a clinical question that you see in the clinic every day into research and then subsequently into policy in a public health scenario. So these steps are known to all of us who are researchers. You identify the problem, you think through the solution, you incorporate value into the solution. Next, you go ahead and do the experiment to show that the solution works. Then you demonstrate scalability, and then you advocate with policymakers to make it work. While we are very good at identifying the problem and showing that the solution works, we are not as good in thinking through the solution. And we found that, at least in the trials that we've been looking at, many of them have not been thought through before the actual trial has been launched. But though I say this, we've gone through several trials and they have shown that certain new interventions work. But what we are really poor at is to incorporate value into our solution, to demonstrate scalability, and to advocate with policymakers, with the result that the results of our research very rarely actually translate into routine practice. So we looked at several research priorities a few years back to look at what we should work towards. And I'll go through 3 of these examples. The first being one on oral cancer. We chose this to be a research priority because it's a very common cancer amongst Indian men. There is no effective systemic therapy, and the outcomes were not great. They were suboptimal as far as survival was concerned. Similarly, cervical cancer was a fairly common cancer in women until very recently was the most common cancer. It's now been overtaken by breast cancer. It was clearly shown in studies abroad that screening was useful, but the ICMR had commissioned a study in the mid-1970s where they said that the established screening methods like pap smear were not feasible in a country like India, mainly because of issues with expertise and logistics. HPV DNA, which requires less logistics, however, is more expensive and therefore again not feasible. Breast cancer has now become the most common cancer in India. It's also the most common cause for cancer death in India. And we also know here that mammography is a useful screening method in women over the age of 50. However, again, similar to the cervical cancer screening story, population-based mammographic screening is not feasible primarily because of the lack of equipment and expertise to interpret the mammographs. So we looked at each of these. So for the oral cancer problem, we looked at a surgical solution rather than look at systemic solutions, systemic therapy solutions. And we looked to see whether a more extended lymph node dissection would improve outcomes. And at the point that we started the study, there was no consensus on what was the optimum extent of lymphadenectomy in patients who had oral cancer, but no clinically palpable nodes. And therefore, we conducted a randomized trial comparing an elective nodal dissection with a wait and watch policy. So we recruited 600 patients who were stratified on tumor stage and site. And we showed that there was a 10.6% improvement in overall survival by performing the surgery electively rather than wait and watch, thereby answering a long held question debate in cancer circles. So this was subsequently published in the New England Journal. The next problem example that I'll use is on cervical cancer. As I mentioned, screening is associated with decreased mortality, but conventional screening methods are not feasible in India. So we consulted Government of India and ICMR and global experts, and we narrowed down to a simpler, low-tech solution called visual inspection with acetic acid by trained health workers. So we went ahead and did a cluster randomized trial. This was a very large study, one of the largest screening trials done anywhere in the world, where we randomized over 150,000 women into 2 groups, one which had a screening arm with visual inspection with acetic acid, and the second, which was the control arm, had no screening. The cancer incidence was similar. One of the big problems with many screening methods is that there is overdiagnosis. It was not seen in our study. The visual inspection with acetic acid did not pick up innocuous cancers. And again, this was published as a full paper and an ASCO plenary session. And what we showed was that by a simple low-cost method, over 30% of cervical cancer mortality could be reduced. So this has subsequently been adopted by the Government of India in its NCD screening method as the method of choice in most states that perform screening. So this has been initiated in 100 districts, and it is likely to be scaled to all districts in India in the next 5 years. And we also went ahead and started a training program for health workers on how to do this method of screening, and have developed a blended learning module which trains health workers, ANMs, to screen. My final example is one on breast cancer. It is now, as I mentioned, the commonest cancer. Screening we know is useful. Population-based mammographic screening is not feasible, though the other options that are available are breast self-examination, which had been evaluated in an earlier trial and found to be not of benefit. And the only other method which was available was clinical breast examination. So we built this into the cluster randomized trial, 150,000 women, half of them undergoing clinical breast examination by trained health workers, and the other arm, which was the control arm, getting no screening. And this is virtually hot off the press. This was published in the British Medical Journal a few hours back. And this gives the long-term results after 20 years of our prospective randomized trial on breast cancer screening. So what this trial again established was that there was a 15% reduction in all women when it came to breast cancer mortality. And when you looked at the women over the age of 50, where mammography has been proven to have the maximum benefit, this showed a 30% breast cancer mortality reduction in women over 50 years. So with these two studies on population-based screening, we not only did establish that these were efficacious, but also the fact that this was scalable. It comes to the next section of my talk, which goes towards how do we translate this research into policy? And this is the valley of death as far as several new interventions are concerned. And we realize that it takes an entire systems approach and not just fix small problems as they come along. And we needed to look at the entire system to be able to provide a solution. So 8 years back, we created this large network of cancer centers in India called the National Cancer Grid. This is a Government of India funded initiative. What started off as a small, modest initiative 8.5 years back has now grown into 236 cancer centers, research institutions, patient groups, professional societies, and charities across the country, which between them treat 750,000 new patients with cancer annually, which is roughly 60% of all of India's cancer burden. Clearly, the initiatives that are taken within the network have the ability to be widely extrapolatable across the population. The three pillars on which the cancer grid is built upon is to have uniform standards of cancer care, to develop trained human resource to tackle the increasing burden of cancer in India, and to provide a ready platform for collaborative multicentric cancer research. So these are some of the research initiatives that we have taken. The first being to promote context-specific research for LMICs in India, which is some of the examples which I have shown earlier. Systematic efforts to identify gaps in evidence, which is done by a biennial meeting where over 200 of India's top cancer experts brainstorm on what are the next questions to answer in cancer research. We go ahead and back that with well-conducted and well-planned trials. We build research capacity by training early career faculty on research methods. And we also have taken several steps to facilitate the actual conduct of the research. We have created a clinical trials unit and a academic contract research organization, which between them help researchers design studies, help them with biostatistical help, with the regulatory submissions, help them with contracts, insurance, and multicentric studies, and also go ahead and conduct monitoring so that the integrity of the data is absolutely ensured. This is a unique initiative by the National Cancer Grid. This is one of 4 such workshops worldwide. There are only 3 other workshops of a similar nature, one in California, the second in Europe, and the third in Australia. This is the only one of its kind in any location other than a high-income country. So primarily what this workshop does is convert a one-page research summary in the back of someone's mind, an early career researcher, over a period of 6 days into a full-fledged protocol which is ready for ethics submission. It's kind of a hackathon for or a boot camp for performing research. And many of these, we've trained over 300 participants so far, and most of them have gone on to academic careers with very flourishing trials after they've completed the workshop. So the feedback that we got from many of these participants was that the workshop was extremely useful, but when they had doubts and questions and mentoring required after the workshop, they found this difficult. So we started virtual research boards a couple of years back, much before COVID started, and leveraged the virtual platforms to train people on virtual research and to troubleshoot if they had any problems with the actual conduct of the study. We also fund clinical research, provided conditions are met. We fund only multi-centric collaborations. We do not fund single-center studies. The cancer that is being studied has to be common or unique to India, clearly making sure that we are evaluating context-specific and relevant research to the country. We look at cost-effective treatment options, meaning that when something is proven to be effective as a consequence of our research, it is implementable readily and cost doesn't come to be a constraint. All researchers funded by the National Cancer Grid have to commit to share their data for other researchers to mine and to come up with newer conclusions. We have a heavy emphasis on public and patient involvement and insist on this increasingly as a necessary prerequisite for being funded by us. There are, as I mentioned, there have been a systematic brainstorming effort at identifying gaps in evidence and answering them, clearly becoming a national cancer research initiative. So these are some of the studies that we have funded. We funded many more after this. But just to illustrate that in a disease like acute lymphoblastic leukemia, where the average clinical trial size is in the range of 300 to 400, and these are some of the best clinical oncology groups in the world which do these kind of studies, the sample sizes have ranged between 300 and 400. Our study has a sample size of over 2,200. Similarly, a 1,500 study on curcumin and metformin in head and neck cancers. As you can see, most of these are relevant to the Indian situation. Add aspirin is a study which we co-developed along with the Medical Research Council in the UK, aims to recruit 2,000 patients in India. We've recruited 2,000 patients on a COVID-19 and a cancer cohort. And all of this has been done with funding from the National Cancer Grid and from other organizations. And as you can see, it is possible to do this at budgets which are much lower than what you would in the West. So to summarize, clinical research in countries like India have many hurdles. There are a lot of challenges, but we need to look at solutions in a systems approach if we were to promote clinical research in the country. Training, logistics, mentorship, networking, funding. These are all challenges and all need to be addressed. We cannot have a band-aid solution to just one of these without addressing the other. So we need to look at it at a systems level. And we've demonstrated that the National Cancer Grid model is feasible and scalable to other diseases as well. So I'll stop with that. And if there are time for questions, I'm happy to take any questions as well. Thank you.
Hello. So thank you very much for excellent talk, Professor Pramesh. I think it was having a broad spectrum of cancer research, and it was very good regarding the National Cancer Grid initiative. So now session is open for question and answer and comments. If someone have any question, please. Is there any question? Yeah. So if there is no question, then I would like to thank Professor Pramesh for excellent talk. And I would like to invite second speaker, Dr. Saggamitra Pati. She is Director at ICMR Regional Medical Research Center at Bhubaneswar. And it's my pleasure to invite her for her talk. The title of her talk is Magnitude and Outcomes of Multimorbidity, Landscaping the Terrain. So Dr. Pati, please. Yeah. Thank you.
First of all, my slide is visible.
Yes, ma'am, it is visible. Yeah.
Is it on
You
full
can,
screen, sir?
yeah.
So first of
Can you
all,
have it in the slide mode?
yeah, I have it
The-
now.
Okay, thank you.
So good afternoon, everyone, and namaskar, Manju madam, because for only I am here. So my heartfelt gratitude to ma'am for considering me for this symposium, prestigious symposium. When I look forward to more such association. And as my topic is magnitude and outcomes of multimorbidity, basically what I will be doing is that I will be just landscaping what has been done. The reason for that is that multimorbidity is an emerging concept in low and middle income countries and more so for Indian context. Though multimorbidity has been very much well researched in high income countries in developed nations, but in India and low and middle income countries, it is yet to take up that pace that is desired. In fact, today morning, our DG has very emphatically highlighted the importance of non-communicable diseases, wherein he had told that if at all there are, if you are going to score which in terms of morbidity and mortality, hypertension, cardiovascular disease, they are going to be in the top leading position. It dwells upon that. However, over a period of time, its conceptual paradigm or its conceptual horizon has little bit broadened. So basically, I'll be discussing that. And my outline is what I will be doing is that I will just give the introduction to the concept of multimorbidity, how it evolved over a period of time, and what is its landscape globally, low and middle income countries, South Asia, India, and of course, what we need to do at this juncture. And with the onset of COVID-19, much more forefront attention than it was getting before. So I'll also be discussing a little bit about the interplay of multimorbidity with COVID-19 because my successor Priya ma'am is going to speak on COVID-19. So what is multimorbidity? Because many times we confuse multimorbidity with comorbidity. Multimorbidity is little novel or little different concept, which is more democratic. Why? Because it encompasses all the medical conditions that is coexisting in an individual rather than taking one particular condition and looking at the associated morbidity, which is comorbidity. So to start with, popularly, it was defined as the co-occurrence of multiple chronic diseases and medical conditions within one person, but later on, World Health Organization defined it as existence of two. The count, the number was fixed that it is two or more. Then we would say, sorry, ma'am. Any problem? So the existence of two or more chronic health conditions in the same individual as articulated by World Health Organization. At the same time, Academy of Medical Sciences went little further and they broadened the definition, wherein they define multimorbidity as the co-occurrence of two or more long-term conditions, but not confining themselves to non-communicable diseases. So as we can see, it contains either of these conditions may be a non-communicable disease of long duration like cardiovascular disease, diabetes, hypertension, or ischemic heart disease. It can be a mental illness of chronic nature like dementia or mood disorder. And again, it can be an infectious disease of chronic duration like HIV or hepatitis C. So whenever two chronic conditions, so what they made is that they shifted from NCDs to chronic conditions because the patient has to deal with chronic conditions throughout. And while comorbidity, which was popularly was being used before, this refers to any distinct additional entity that has existed or may occur during the clinical course of a patient who has the index disease. For example, if a person has diabetes and he develops nephropathy, nephropathy is considered to be a diabetic comorbidity or in that sense. But multimorbidity does not look at which is the index disease, rather it looks at all the conditions at any point of time that exist in a patient or in an individual. So basically, when we do a diagrammatic, this is what multimorbidity, the pictorial or the diagrammatic representation would be, and while this is comorbidity, as you can see, from the index disease and its associated comorbid conditions, while multimorbidity looks at all the conditions, what is the effect on the patient. And very pertinent quote of Aristotle who told, the whole is more than the sum of its part, and multimorbidity very truly fulfills that. Because 1 plus 1 is never 2 in multimorbidity, rather it becomes 4 because of the cumulative complexities of each chronic conditions on the patient. This is what if we see this diagram, that the coexisting conditions are of less importance. Treatment usually tries to induce or remission of the index condition. So basically, which is a disease-specific approach, and very traditionally we have been doing in our medical system, that we look at medical condition A if the patient has it, and whether he has B and C, then we look at all conditions. But multimorbidity is more, as I told, it's a more democratic term where all conditions are given equal importance, but treatment is oriented towards outcomes that matter to the patient. That means it is a holistic or a patient-centric approach to look at a research. That means medical A, B, C, all are given same weightage, and what effect they are having on the patient, that is mainly taken care of. So the difference, the primary difference between comorbidity and multimorbidity is that comorbidity takes a specific approach, and the associated medical interventions are primarily towards the index condition, while multimorbidity is a patient-centered approach, which you really need now with Ayushman Bharat in place, and also health and wellness centers are being set up across the country, wherein we are not looking at a particular condition, but we are looking at the promoting wellness, which includes both physical and mental, and of course, social well-being in a person. So why it is important and why it has been coming to the prominence? Because now it is being observed that majority of people who are aged 65 or more have multimorbidity. And why it is important? Because multimorbidity has got a broad range of adverse outcomes, which include impaired physical of life, increased health service utilization, which is very important for the health system point of view, then more prone to premature mortality, and increased health service cost, because a multimorbid person uses more health services and incurs more out-of-pocket expenditure for multiple treatments. And in high-income countries, already multimorbidity has been proven to be a norm, not the exception. At least 50 million people in European Union alone have been diagnosed to have multimorbidity. It is, and with urbanization, with globalization, so becoming a pervasive problem in low and middle income countries as well, which means multimorbidity is the true global health challenge of 21st century. However, despite its huge problem the multimorbidity brings in, surprisingly, little research into its impact on patient, healthcare system, and society at large has been done. This is particularly more for low and middle income countries, and that is why I am going to put the point that what needs to be done to fill this knowledge gap so that we can improve care and outcomes for patients. Because if we look at our patient care protocols, nowhere it discusses or tells or deliberates on multiple conditions, rather it is very mono disease specific or mono condition specific. So how can we have a clinical care protocol when a person has got two chronic conditions which may be totally discongruent or incongruent? So let me just take a how this concept demography. So initially comorbidity was being used for the first time in 1996. Marian van den Akker of Netherlands, she started this question that comorbidity or multimorbidity, what is in a name? Is there a difference between these two? So she was the person behind coining the term multimorbidity. However, it was struggling to come to prominence. In fact, there was no mess on multimorbidity in the PubMed till January 2018. So in January 2018, NACE has designated a definition and a different classification term for multimorbidity, which is distinct from comorbidity. That shows that now multimorbidity has become an issue of prominence for researcher as well as for.
Sorry, madam, your slide is not moving forward, madam. Sorry, madam. Sorry to interrupt. Madam, slide is not moving forward, madam.
Oh, okay. Is it? So what should I do? Should I again do? Now?
Madam, you can just give enter because the slide is on first page. Your slide is.
Oh my God. Okay. I was. Okay. Now it is coming. Is the flash
Yeah,
slide,
yeah,
right?
it is coming. Please,
Now?
please go forward. Please go forward.
Coming?
Not yet.
No, still in the first slide. You are in the first slide. Please
Stuck on the first
scroll
slide.
it down.
Yeah, I scrolled it down. Now?
No,
No?
still the first slide.
Should I stop sharing and again speak? What should I do? Should I stop sharing and again do? Or is there anything at that side? Now also?
Madam,
No,
yeah,
it is
now
not visible.
reshare again, madam. Reshare.
Reshare. Please reshare.
Or should I forward it to you? You can maybe then. Should I send the slides to you so that you can go ahead? Or
Okay, madam. Okay.
should I send the slide set to you then?
Yeah, please.
That would be helpful. Then let me stop sharing and again do it. If not, doesn't come then.
Yeah, first let try one time, madam. Let's you try reshare. If you're not properly moving, then you can send me.
Now it's coming. Now it's coming.
First share, madam. First you share.
Okay.
Santosh.
Yes, madam.
We can't see the slides.
Yeah, just madam is sharing, madam. Just wait. Otherwise, if you problem, madam, kindly say mail your slide. We will share it from here. No problem.
But nobody can see.
Just wait, madam. We are trying to share. Sangeeta madam, if you have problem to share, kindly mail to us just now, and we will.
I've sent you.
Okay, madam. Okay, just please. Yes, ma'am, I've received. Just please.
Please, go ahead and I will just go on. No problem, please. Sorry for all this. I didn't know that it was not moving.
Okay.
Yeah. Now where we were?
Okay, I can just flip through if you want for 2 minutes. You can from the very beginning if you want. Ma'am, should I start?
Yeah, yeah. Please start, please.
Okay, so please, can you, sir, can you please go back to the first one? I will just quickly flip through.
Now we can see.
Yes, ma'am. Sorry, ma'am. If I could have been told before, then I would have sent it. So basically, sir, can you go to the first slide, and then after that, I'll just quickly move and I'll come to the first slide where I was.
Please let us know when you say for the slide. We will move accordingly.
Okay, so this is fine. I mean, I can start from here only. So second.
Okay, when you say next, we will go.
Yes, next. Next. Okay. So basically, when we see this picture, comorbidity versus multimorbidity looks at the conditions as a whole rather than importance to any index condition. And next thing, so this the title that is the whole is more than the sum of its parts is very true for multimorbidity because it is 4 on the patient because of the cumulative complexities, the illness burden, the multiple medications, multiple physician consultation, and of course, quality of life being inferior. Next. So when we see the measurement of multimorbidity, we can see there are 2 approaches. Comorbidity is a disease-specific approach, while multimorbidity is a patient-centered approach. Why? Because we see all the A, B, C, for example, all the medical conditions, what is their impact on the patient, and multimorbidity looks at the patient-reported outcomes rather than looking at a clinician-reported or a disease-specific outcome. And in comorbidity, the coexisting conditions are of less importance. Treatment primarily seeks to induce the remission of the index condition. If a person has got diabetes and got rheumatoid arthritis later on, but the index condition is given priority, the rheumatoid arthritis might be causing much more problem for the person concerned. That is the reason multimorbidity has been coined, because it is a more holistic concept, more patient-centric, more democratic, and more oriented towards improving the health outcomes. So why multimorbidity is important? Because it has been shown that majority of people who are aged 65 years or above have multimorbidity. And why? And what are the adverse effects, or what are its long-term consequences? Multimorbidity, because of its multiple chronic conditions, leads to impaired physical, social, and psychological quality of life, increased health service utilization and mortality, and increased health service costs. In high-income countries, multimorbidity has been proven to be a norm, not the exception. In fact, already 50 million people have been diagnosed with multimorbidity and are living with it. And it is urbanization, with globalization, with changing lifestyle, multimorbidity is emerging an equally important public health challenge in low and middle income countries. And that means it is going to be a true global health challenge for the 21st century. However, despite its huge implications for healthcare practice or healthcare delivery, there has been not much research into its impact on patient healthcare. Next. And let me tell about how this term before multimorbidity term was coined, it was more of comorbidity designed by Feinstein. He was the first person to coin the term comorbidity. But it was Marion van den Akker in Netherlands who was doing a PhD. And during her PhD, she coined or she thought of this term, that multimorbidity. In fact, her question was, what is there in a name? Is it comorbidity or multimorbidity? How does the two names differentiate? From 1996 till 2018, multimorbidity struggled to be on the forefront, and in fact, only in January 2018, there was a special mesh or a different mesh term coined by PubMed, so that to symbolize that now multimorbidity is a distinct term from comorbidity. So it took a long time for multimorbidity to come into the focus of the researcher and the journal or the scientific community. Next, sir. So now let us look at the publications in Medline when we use the word multimorbidity. As we can see, slowly the rising interest in multimorbidity can be evident or can be seen when we see this rising graph. Like for example, in 2000 and 2002, 2004 and 2006, we look and suddenly there is a spike in the number of publications. So till 2006, the publication was not much, but after 2006, the interest gathered, the momentum gathered, and people thought of what is multimorbidity and how it should be studied and why should we study. In fact, the fact that there is a journal of multimorbidity and comorbidity by SAGE, which is an international journal, which exclusively looked at the problem or the magnitude of multimorbidity with special focus on its etiology, its prevention, its management. Is there any genetic or is there any what are the mechanism behind clustering of conditions, and what is the outcome or the trajectory of multimorbidity, why certain conditions lead to higher mortality, why few do not, and why certain conditions require multiple medications, but still there is no improvement in outcomes, and how to optimize medicine. So all these articles are considered by this journal. And similarly, when we look at the global scientific research and multimorbidity, you can see, yes, sir, please go ahead, sir. Go ahead, sir. So as we can see the spike. So that means that only last 5 years or 6 years, multimorbidity has come to prominence. Next one. And absolutely. And in fact, World Health Organization started multimorbidity, looking at multimorbidity in 2016. Again, it's hardly 5 years. And 2018, Academy of Medical Sciences told that multimorbidity is a priority for global health research. So these two landmark documents created the momentum for or the conducive path for taking up multimorbidity research globally. Next slide. Now, how can a research be patronized unless there is a funding? As we can see, MRC started a dedicated fund called for global multimorbidity. That means that because as long as there is no dedicated funding, there is no dedicated grant, it is very difficult for any kind of research to nourish, to nurture, or to grow. So, and not only that, GSCD, Global Alliance for Chronic Disease, has already announced that its 2023 call is going to be only on multimorbidity. Why? Because in particular, which area? How can we have principles of implementation research which will be used to integrate interventions for optimizing management? Most important, the COVID-19 has also taught us how do we care for a chronic diabetic when there is COVID-19? How do we give steroid when the person is already having hypoglycemia? So these kind of puzzling questions are needed, and we must have those kind of facts and figures with us. And not only that, they have also emphasized on using artificial intelligence, big data, medicine, which is very true for now COVID-19 context, into NCD implementation science. Next slide. And these are the three important studies which are, in fact, this Lancet study kind of created the former of, or created the map for doing the multimorbidity study, wherein Barnett is very famous, in fact, there's 4,000 citations till now, wherein she is the first one who found out the epidemiology of multimorbidity. Yes, it's a excellent. And similarly, low and middle income countries also geared up. China also geared up. Now comes, what are the challenges of multimorbidity for today's healthcare system? Right? But there are multiple challenges. First, we do not have any evidence base, any standard guidelines to manage multimorbidity till now, because we still work on single disease specific guidelines. We are yet to think of, if two diseases are there, what should be the optimized protocol? How can we reduce the medicine number? It's not that we go on adding medicines as the number goes up, counts goes up. And the most important side effect or challenge is polypharmacy. Polypharmacy is defined when a person takes more than five types of medicines per day. Polypharmacy is a major challenge in multimorbidity. And third is that it has been shown that when there is physical multimorbidity, it somehow or other induces psychological distress. So the combination of mental, physical, dual multimorbidity is very important, so that we include in different professionals when they are delivering the care. And treatment burden. Remember the medicine, remember the visit, remember the laboratory test. So too many factors are playing to increase the treatment burden. At the same time, our system is morely moving towards specialism. We are more concerned about specialty rather than generalist. While multimorbidity is more concerned about generalist approach or a patient-centered approach. So specialization of medicine, too much specialization of medicine is also going to be contrasting for multimorbidity, because we have to look at the patient. And of course, our primary care system has to be the linking pin for delivering or for being the contact between the patient and the specialty care. They should be not only the gatekeeping, but they should also be doing multiple range of care, so that a person gets everything under one roof. What is that is envisaged under our health and wellness center. And third, the very important thing is how do we not so much of resources, because with aging, increasing life expectancy, people are living longer, but everybody should have healthy longevity. So how do we ensure healthy longevity with chronic condition? We need ample resources for providing care, for managing their treatment burden. And most importantly now, interface of multimorbidity with chronic and acute infectious diseases, because all of us know the most vulnerable were those who had comorbidities, and those who have two or more, they were in fact most vulnerable. So how do we address, or how do we protect these vulnerable patients when there is any kind of pandemic outset or any kind of onset of pandemic? Next slide. सर जल्दी चलिए हो गया सर प्लीज. This is basically the study that UK Lancet. They have found out that 16% of any adult person have multimorbidity. When we see females have got more compared to male, in Western countries, though in India it is little different. Next slide. Sir, आप quickly चलते रहिए, मैं बता दूँ. And this is very interesting when we see with age how the number of conditions are going up. 25 to 29 versus, for example, 75 to 79. The percentage of conditions, and in fact 0 to 7, how even people have got 7 conditions also. And it can be, life can be very difficult for those who have got so many multiple chronic conditions existing. Next slide. And but one thing is that even though people, proportion of people with multimorbidity is greater in older people, there are more people aged 65 years with multimorbidity. That is very worrisome for a country like India, where majority of our people under 65. We have very young population, young demography. If our young demographic is having multiple chronic conditions, then it becomes very difficult in terms of national productivity and societal output. Next slide. And most important is that socioeconomic deprivation. Ideally, it is always thought that those who are having low socioeconomic status, they should be having less number of NCDs, but it is not true. Those who are living at the greatest deprivation, in fact, they have more multiple chronic conditions slowly because they are more disproportionately affected with globalization and lifestyle changes. That is very important when it comes to equity. Next slide. Quickly move quickly. And this is very important because I want to highlight this pattern. What we can see is that the combination, the desired combination of conditions. Someone is having, for example, respiratory along with anxiety disorder. Someone is having mood disorder with musculoskeletal. So how do we affect so many variety of permutations and combinations? So how do we develop care protocols for such kind of mixed and varied patterns of multimorbidity? That is a challenge now for all the clinicians across the globe. Next slide. And impact, what is the impact? Any versus, when we see this multimorbid versus not multimorbid, here let me point out that not multimorbid means they may be having single chronic condition. So if single versus 2, if we see the physician consultations or the consultations with doctors, a referral, hospital admissions, and healthcare costs are much higher compared to single. So when second comes, the amount becomes much more. Next. So this is our study which we did look at middle income countries. We looked at the impact of non-communicable disease on healthcare utilization and out-of-pocket expenditure, because that is what our health protection scheme is planning to do. And we found that, next. So what we found is that this is China, Ghana, India, Mexico, Russia, and South Africa, all these 6 countries. When we see the prevalence of multimorbidity by different age group, and if you can see, China and Russia are most high, and India as well. Green color is India. So you can see that along with age, there is increased prevalence of multimorbidity, and India is also into that trend. Next. And this is the out-of-pocket spending. We see for each person how much they spent on outpatient services, and for each type of, for both inpatient and for outpatient, if you see medicine is taking too much of cost, similarly followed by healthcare provider, followed by medical tests, followed by transport and others. These are the different cost breakups, out-of-pocket expenditures. Where do people spend more? Because accordingly we can design our financial protection schemes. Next slide. And this is, for example, if you see the outpatient visit, the number of NCD 1 versus 2, 0 versus 1 versus 2, how 2 is much more. The outpatient visit in terms of multimorbidity. Adjust both after ANOVA regression analysis as well as adjusted odds ratio. Next. And most important is physical mental multimorbidity has been shown that it leads to disability, reduced work productivity, and of course decreased social participation. When we look at depression, we can see that those who have got more number of chronic conditions, depression is sets in. Psychological, physical multimorbidity are very closely related, which means we have to think of mental wellness along with physical wellness whenever we are designing any kind of care delivery plan. Next. is what all the different, only if they are early retired, if those who are having more chronic condition, their chances of retiring early. Those who have got more chronic condition, they are more rate of sick leave, they have got more reduced chance of social participation. So different dimensions. So along with a very strong correlation with multimorbidity. Next. To see how, I mean, it's a very complex, but this is the truth. How do we manage when a person is having atrial fibrillation, osteoarthritis? You can see the complexity. I mean, that I just wanted to put it. How a doctor is going to prescribe medicines when a person is having so many chronic conditions together? Because one might be contraindicated in the other. What do we do in that case? So this was the patient complexity. So how the diseases and medications affect one another when a person has got. For example, if it is concordant, it's fine, but when it is discordant, it becomes more problematic because antidepressant may promote something cardiological problem. So how do we optimize the treatment? That is going to be a challenge in the coming years. Next. So most important now is polypharmacy. Now most all of us have got this plate full of, this basket full of medicines because of our multiple chronic conditions. Every person must be taking 4 or 5 medicines per day because they have to manage each individual chronic conditions. Next. So this is the concept of how it is because of the different conditions. The network, how it leads to different this network diagram. Next. So basically, when we see from a holistic point of view, multimorbidity leads to morbidity burden, and that leads to patient's complexity. So this is how whenever we look at multimorbidity, we have to look at this complete picture rather than looking the patient per se. Because the gender might be influencing the morbidity burden, age might be influencing, other health-related attributes like frailty or like body strength might be influencing, then non-health related like income, access to hospital, then literacy, then availability of family members. So they also contribute. So all contribute to a patient's complexity. It is not only the condition, there are many factors which contribute finally to a patient's complexity, and we have to take cognizance of that. Next. So we also, but however, compared to low and middle income countries, we did a systematic review in South Asia. Much study has not yet been done on multimorbidity. We could find only 13 studies, but none of them had a uniform methodology. Hardly their sampling size was not very uniform. Most took secondary data analysis. So what I'm saying is that South Asia needs to reinvigorate in terms of multimorbidity research. We must now take up multimorbidity as one of the research priority. I would request ma'am through NASI and through if she can promulgate or if she can advise the research agencies how to have a dedicated grant call on multimorbidity, because that is what is needed now. Next. So we had done this study and we found that there was very high prevalence. Our first study showed that in primary care, 30% of people had multimorbidity attending primary care. And not only that, we also found low socioeconomic status had got a greater morbidity burden, which is a double burden for them. Next slide. And if you see how with the age in Odisha, how the population more than one, how the number of chronic conditions went up along with age. And this is the different patterns we had. Sorry, before. Just I will show like what zero morbidity had in the young age group. As we can see, 60% had zero morbidity, but still one chronic condition have. I mean, even if they are below 40 years, we are seeing even 3 chronic conditions have certain people. But as the age goes up, we can see that the number of 2 and 3 goes up, increases along with age. And this is similar for both male and female. Next. And in fact, now we analyze the NASI data, that is the longitudinal study on aging, and what we found is that older adults have also got 53% of older adults have also got multimorbidity. And this is among urban poor. We selected the urban poor and looked at the multimorbidity patterns. So social deprivation is strongly associated now with multiple chronic conditions, and this is going to be a real challenge for their meeting their healthcare cost by the system. And this is the self-reported health among, because as we can see, multimorbidity versus no multimorbidity, those who have got multimorbidity, they have reported that their health is not good. So that means the self-reported, self-rated health or self-reported health is worse in terms of multimorbidity among urban poor also. Next. So now I will just take 2 slides to show. We analyze the COVID-19 data and multimorbidity. Just sir, so basically what found is that in Italy or in other countries, they found that presence of multiple comorbidities or multimorbidity contributed exponential mortality independent of age. So here age is not a factor. Even if a person is young but has got 2 chronic conditions, his or her chances of mortality is much higher compared to a person of higher age but has got less number of morbidity. That means multimorbidity is a strong risk factor for morbidity and for mortality or adverse outcomes in COVID-19, independent of age. Next. So the risk of dying was significantly higher among patients with two or more than 2 comorbidities with higher hazards ratio, as we can see. Next. And as we can see, and not only that, multimorbidity also increases the risk of getting COVID-19 infection. Data from UK Biobank, they show that they followed the longitudinal cohort, and those showed that those who have got 2 or more long-term conditions had a 48% higher risk of acquiring COVID-19 compared to those who had 1 or no morbidity. Next. Next, sir. This is the different factors relative risk by long-term condition count, and what are the prognostic factor like ethnicity, then gender, age, and of course income. Next. And polypharmacy also was found to be very strongly associated with multimorbidity and had a more adverse outcome in COVID-19. So we did a small study on how people managed multimorbidity amid COVID-19 pandemic, and we found that there were disrupted care for those people who had NCDs, and the most common reason were physician consultation, access to diagnostic services, transport, mobility restriction, and multivariate analysis revealed that older adults living alone had higher challenges than their rural counterparts. In fact, rural people were much better off compared to urban, which calls for we have to think of patient activation for self-care because a doctor cannot be all the time present, and technology-enabled teleconsultation. So what should we go now? Where from here, now that we know so much about multimorbidity, what should our research be? That I will just give one slide on what should be the research and end up there. Next slide. So we need research for what should be our research. First is we have to have a nationwide study on our multimorbidity magnitude. Until now, we do not have any data. And second, we have to develop evidence-based protocols for managing multimorbidity. Third, which risk factors are important for developing multimorbidity, that is from 1 to 2. Many people continue throughout their life 1, but many quickly develop 2, 3, 4 chronic conditions. So what are those risk factors? And fourth, what is the trajectory of multimorbidity and the outcome? Which type of clusters have got worse outcomes compared to those other clusters? Next. And for this, we must focus on health system, not only leadership and governance, how to train our health workforce, how to strengthen medical technology, how to have a health financing, and how our service provisions should be aligned. And at the end of the day, we must protect people against catastrophic expenditure, and our health system should be responsive for improving the health of the population with across different levels, different strata, and to ensure equity. With this, I stop here. My heartfelt gratitude to Manju ma'am, Manju Sharma madam. Madam has been a great source of inspiration and role model for many women researchers like me, and NASI and the organizing committee of the NASI Symposium, all the distinguished speakers and chair, the esteemed audience, and my own organization ICMR and DHR, and of course, without my colleagues, nothing is possible. It's everything is a teamwork. Next.
Thank you.
With
Thank you
this, I
very
thank
much,
you for all your
Dr. Pati.
attention.
Yeah. Thank you very much, Dr. Pati, for excellent talk. And now session is open for comments or any question, please.
And I once again apologize for the inconvenience, and that shows that everybody is subject to technical.
Madam, just may I ask one query?
Yes, please, sir.
Santosh here. Madam, you said about the polypharmacy. It was very nice and catchy word, polypharmacy, for this morbidity topic. And but can we have also considered in the our India context, we are using the banned drug. Plenty of banned drug is used in the so many, not on the medical store, the plenty of society is using. So banned drug is also be considered in the one factor of this.
No, what happened is that we have to also look at the access to care, because majority of the drugs, to be honest, are being procured through OTC, over the counter, because access to healthcare, and then that is what you rightly pointed out, and the informal care providers, we won't call them other name. There are, every community has got their own system of local healers or local care providers whom people have trust in. I mean, because why? Because they are easily accessible. They know that at the midnight, at the end, anytime I want, I can see that particular person. So that could be one of the reasons. But definition of polypharmacy will remain same.
Thank you. Madam, your mic is mute, madam. Madam Manju Sharma, madam, your mic is mute. Madam, mic mute hai. Please unmute yourself.
No.
Yes.
Yes, ma'am. Please.
We can ask the audience to send their questions and we can proceed. We can ask questions late in the end if we have time. Otherwise,
Okay,
we
thank
can
you.
proceed.
Okay, ma'am. Thank you very much. Thank you very much, Dr. Pati, for excellent talk. Now, I would like to invite speaker number 3, Professor Priya Graham, Director, ICMR, National Institute of Virology at Pune. And it's my pleasure to invite her. And the title of her talk is COVID-19 Pandemic Scenario, Research, Prevention, and Control. Dr. Priya Graham, please.
Thank you. Thank you,
Would
ma'am.
you like to share? Would you like to share your PowerPoint from yourself, or we have to share?
So I will try from this end. Seeing the problems that other speakers faced, I've also sent you the PowerPoint. I will try and share from this end.
Okay, okay. We have received your PowerPoint.
Yes. Good afternoon, Professor Manju Sharma, Dr. Sinha, Dr. Shukla, other esteemed scientific colleagues, and my fellow directors from ICMR. I take this as a big honor to be able to speak at this important occasion of the 90th annual session of NASI on a symposium topic that is very relevant today towards a new healthcare regime for our country. So with that, let me try and share my slides. Is it visible to all?
Yeah.
Yeah, yeah.
And is it moving, ma'am?
Yes.
Okay. So the Honorable Chairperson has already spoken about my topic that was given to me. And this is about the pandemic scenario, some of the research initiatives in this scenario, as well as prevention and control. So the outline of my talk would be to particularly put stress on this concept of variations in the virus genome, the evolution of the virus, variants, by defining them, the implications of some of these variants, and then touch very briefly on prevention and control. So we all know that this SARS coronavirus 2 or SARS-CoV-2 belongs to the family Coronaviridae, virus family Coronaviridae, and it belongs to the subgenus known as Sarbecovirus. And its other cousin in this subgenus is the SARS-CoV-1 or the earlier SARS that we encountered in 2002-2003. But this simplified phylogenetic tree here at the right bottom of your slide actually gives you the relationship of the SARS-CoV-2 and the degree of relatedness with the SARS-CoV-1 and the MERS coronavirus, both of which were also very pathogenic. So if you compare SARS-CoV-2 with SARS-CoV-1, there's about 79% homology. With the MERS-CoV, it's only 50% homology, but its closest relatives actually are a bat coronavirus, which has 96.1% homology with this virus, and a coronavirus from pangolins, which has about 91% relatedness. We know in November 2019, and that's where the pandemic is dated back to, this virus originated in bats, made its debut in humans. And from the time it got introduced into humans, it has been trying to adapt to the human host, whereby this virus has been undergoing some variation in its genome constantly. So because this has generated a lot of interest, as of today, in the month of February 2021, we have more than 5.6 lakh whole genome sequences deposited into public databases, of which the biggest contribution comes from Europe, followed by North America and Asia. Oceania contributes about 3%, with very little contribution thus far from South America and Africa. Now, the ICMR, National Institute of Virology, as you know, was the first institute to report this virus in the Indian continent. We reported it on Jan 30th. We were very caught up with establishing the assay further, equipping multiple labs, virus reference diagnostic labs over the country, sending them SOPs, sending them reagents, having quality control. So we deposited the whole genome sequences of our first positives only by the first week of March. And by then, already 240 sequences had been submitted globally. These are our two initial whole genome sequences from NIV. And even though both these sequences had a 99.98% identity with the Wuhan seafood original SARS-CoV virus strain, you could see that they seem to have some different lineage, as you can see from this. When we looked at the same phylogenetic tree, looking at all the sequences submitted a week later, by which it had almost doubled, we could see this tree was getting much denser, and these same two sequences were clearly grouping into distinct clades. We published this in IJMR as per the desire of our DG. And then we looked at the whole genome sequences, early sequences that we had from travelers and tourists, as well as returnees from the countries of Italy, Iran, and China. And again, if we look here, sequences that came in from Italy, sequences that came in from Iran, which clustered here, and then our first two sequences, clearly grouped into distinct clades. And today we look at the major clades circulating within our own country, which is clade O, also called as the unclassified clade, and clade G with its subclades GH and GR. Most of those introductions into our country have been from China, Europe, Southeast Asia, the Middle East, Africa, and Far East. And that is why we have in our country, to some extent, a mishmash of clades. Now, I wowould like to clarify the nomenclature that has been used interchangeably by different scientists and in different publications. There are three major nomenclatures. One is the GISAID. GISAID stands for the Global Initiative on Sharing All Influenza Data. This was a global science initiative started in 2008 for influenza. But today that is shared for also the use of submitting major SARS-CoV-2 whole genome sequences. We have the next strain nomenclature, which is also an open source initiative. It does real-time tracking of this pathogen. And we know, whereas in GISAID we have 8 major clades, with next strain we have about 11 major clades. And then we have pangolin. Pangolin does not refer to the animal pangolin, which I spoke about earlier, but its expansion is phylogenetic assignment of named global outbreak lineages. And this nomenclature focuses on the actively circulating virus lineages in a region and those that spread to a new location. This is an extremely dynamic nomenclature. And as of today, we have 6 major lineages, but as lineages expand, we'll see the alphabet expanding and these numerical numbers also expanding. So you could become 1.1.373 and so on. You know, it could just expand as new lineages are recognized globally. Now we look at the GISAID classification, the initial 2, which we had, even our first 2, and globally, the first 2 clades recognized were the L and the S clades. And within the L, L expanded to G, V, and O clades, and the G clade further expanded to other subclades, namely the GH, GR, and the GV. I've already spoken to you about the expansion of these acronyms, about the nomenclature. And if you see the 3 nomenclatures and their trees, aligned next to each other, the GISAID deals with the amino acid sequences in the whole genome sequence. The next strain is basically giving you a temporal dating, a dating in time. And the pangolin deals with nucleotide differences in the whole genome sequences. The table here is actually a head-to-head comparison of a particular clade in one classification or nomenclature against its comparative clade in this nomenclature and this nomenclature. And the column on your far right actually refers to major marker mutations seen in these clades. And I draw your attention to a particular mutation in the spike protein found in the G clade and its subclades GR, GH, and GV, wherein the a particular amino acid at position 614 of the spike protein changes from actually aspartic acid to glycine, and we will see the impact of that shortly. So actually what happened is initially we had the spectrum of the VSOL clade, but this was soon overtaken by the G clade, GH, GR, GV, and today the major subclade is the GR itself. And all of them express this mutation at the 614 amino acid position from aspartic acid to glycine. And you can see this even in continents, be it Europe, North America, right down to South America, across different continents. The predominant clades belong to G and its subclades. Here, Africa has yet to submit its data for the month of February, so this will be populated in due course. What about India? India has submitted over 5,000 whole genome sequences into these databases, of which Maharashtra has done maybe the largest amount of whole genome sequencing, followed by Telangana and then Gujarat. And the states colored in slate or dark gray are states where they've submitted some sequences. Those in pale gray are those where we have no sequence data available. Now, if you look at what is known as a sunburst diagram of clades and their individual lineages within clades within our country, we'll find again there's a predominance of the G, the S, GH, the GR, and the V subclades, of which the G, GH, and GR are the most predominant. And we also, I'm sorry, these arrows have lost their alignment somewhere along the line, but actually these arrows were supposed to point at the predominant lineages within these clades, namely B.1.36, which is somewhere here, and B.1.1.306, which is somewhere here, and so on. So the arrows as they display now don't make any sense, but they've moved orientation. But basically to tell you, within the clades, there are lineages that also have relative dominance, so to say. If you look at across states, yes, Maharashtra has, as I said, largest number of sequences. And if you look across the board again, predominance of G and its subclades, though some Delhi Union Territory has a good representation of the O clade, likewise Telangana, Maharashtra to some extent, Odisha, and so on. This tracking of different clades and their subclades and lineages between states have also helped us to track the movement of migrant workers even after the religious gathering that happened in March 2020. We could actually track the movement of individuals from the place of origin to different states. Now, just to make the nomenclature a little more clear and to tell you the significance of the word variant, a mutant is a natural byproduct of viral replication. It's more so in RNA viruses. A variant, on the other hand, it has a demonstrably different phenotype, unique phenotype, which may impact on its transmissibility, maybe its virulence, or even its capacity to bind with neutralizing antibodies. So what is the consequence of having these variants? It could, as I said, impact on transmissibility, impact on morbidity or mortality. It could evade detection by certain classic diagnostic tests, particularly those that target the S gene of the virus, which could lead to false negatives in some assays. It could lead to decreased susceptibility to certain antiviral drugs, decreased susceptibility to neutralizing antibodies, particularly therapeutic neutralizing antibodies in the form of convalescent plasma or monoclonal antibodies that have become quite widely available. It could also evade natural immunity, which could lead to the risk of reinfection, an entity that we have been hearing about, and could also have the ability to successfully cause infection in an individual who's already been vaccinated. So I want to home in to some X-ray crystallographic picture structure model of the spike protein, wherein you can see the S1 part of the spike protein, the S2. This is one monomer. There are 3 such monomers for the entire spike protein, and this is the RBD region, and I'll show you in close up the specific region that binds with the ACE2 receptor on host cells. So this is the specific receptor binding domain, or the RBD, that everybody talks about. It runs from amino acid position 437 to 508, and it has some important amino acids, 501, 417 and 484, significance of which I will be telling you shortly. And also in the spike protein is a particular amino acid at position 614, which I had mentioned earlier, seen in clades G and its subclades GH, GR, and GV. And normally this amino acid has got aspartic acid at that position, but when it's replaced by glycine, we will see what happens. So when aspartic acid is replaced by glycine, what actually happens is it causes an opening out of the spike protein in an increasing partial or in a fully open fashion, thereby enhancing the binding with the ACE2 protein. And I will detail this even more. Now, if we were to look at this binding, this actually is a hydrogen bond formation between the amino acid position at 614. This is the S1 protein, and this yellow structure is the S2 protein. And it forms an important interprotomer bridge or latch or a hydrogen bond between 614 position of the S1 protein and the 859 threonine position of the S2 protein. But when this aspartic acid changes to glycine at the same 614 position, it causes a weakening of this interprotomer bridge, so to say. And what actually happens is where here it is more compact, and one would say the latch is on, the latch is off. And what happens is, because of the weakening of the interprotomer contact, the spike protein assumes an increasingly open conformation, which facilitates the binding to human ACE2 and also spike-mediated membrane fusion. Effectively, this increases infectivity in cells. So this is what this particular mutation, the D614G, can do. Now we look at some of the other variants. All of you have heard of these variants, the B.1.1.7, also known as, people colloquially call it the UK variant. We are not meant to actually call it by the country name because it leads to some amount of stigmatization. But if we look at this variant, you can see that in the spike protein, the nucleocapsid, and the other regions of the structural genes, there are several substitutions as well as even deletions, with a few more other changes in the ORF1. When we come to another variant of concern, B.1.351 from South Africa, again we see some substitutions in the spike region as well as the nucleocapsid region, and maybe one more in the ORF1A. When we come to the P.1, which originates from Brazil, we can see again a whole slew of substitutions as well as substitutions and deletions happening in other non-structural genes of the genome. The same information is depicted in the form of a table, and I want to draw attention to the fact that there are several changes happening for these variants of concern in the RBD region of the spike gene, as well as other regions of the genome. And these signature mutations, namely aspargine to tyrosine at position 501, at position 417, lysine to aspargine, and again at position 484 from glutamic acid, from glutamic acid to, from glutamic acid to lysine, we see mutations seen in the spike region of these variants of concern. Brazil also has another variant emerging. And we also have some other variants which are still under investigation, a B.1.525 seen in Nigeria and in the UK. Again, we are seeing substitution happening in this receptor binding domain of the spike protein. Likewise, a B.1.49, also known as cluster 5, which was associated with a big outbreak among minks in Denmark, where they had to cull many minks in Denmark with a mutation in the RBD region. This actually caused an outbreak wherein the virus spread from humans to mink, spread between the mink, the animal's mink, and then there was it came back from minks into humans. So there was reverse zoonosis and zoonosis. So that led to actually culling of several minks in Denmark. And if we look at the distribution of lineages with variants, we can see that this really happened to be noted from about September of last year with the B.1.7, the one from UK, increasing in number This is the month of February. This is not to depict that these variants are coming down. It's just that in February, the data is only partially got. We need to get the full data with the close of the month. So we would imagine that some of these variants would actually have increased in number. You can see these variants of concern actually slowly mounting in numbers with time. I come to this ribbon model of the AC2 receptor protein, and this is the RBD, receptor binding domain of the spike protein. Again, I draw attention to these important amino acid positions, and in the wild form, there's nothing too much that would draw too much attention. These are the binding areas of these amino acids. But in the mutant, wherein the asparagine changes to tyrosine at position 501, we actually see that there are increased hydrogen bonds and pi stacking when the asparagine changes to tyrosine at position 501. At these positions where mutations have happened, the Sol bridges are actually abolished, which actually does not much impact on affinity at all, increased affinity, but certainly this mutation impacts on higher immunity, higher affinity and enhanced transmissibility. That's why we say that the UK or the the variant which has the N501Y mutation, or the one that originates from UK, has got an enhanced transmissibility. Now let me look at the RBD domain and its capacity to bind with the neutralizing antibody. This is the heavy chain of the neutralizing antibody with its light chain. And you can see here that in the wild form, again, not too much changes, but in the mutant form, where lysine changes to asparagine at 417 position, and where glutamic acid changes to lysine at the 484 position, what actually happens is there is an abolishing of key interaction. Here you can see an interaction. Here this key interaction is lost. Likewise, at this position, key interaction is lost. You can see here. And therefore, this leads to poor binding of this variant with neutralizing antibody. Now, that would be of concern with those who have been vaccinated, who have had natural infection. Will this impact on poor protection, because neutralizing antibody is binding less because of these two mutations? We will also note that the 501 position doesn't seem to impact so much on binding. You can see both in the wild and the mutant form, it does not seem to impact so much on antibody binding. And this was proven by work at our center, wherein we looked at individuals who had been vaccinated with Covaxin. We looked at these people who had neutralizing antibodies. We looked at the neutralization titers when we incubated it with the UK variant, which had the N501Y mutation. We also compared it with other clades of G and O, etc. And we found this did not alter the neutralizing ability. The titers were very comparable because, as I said, the 501 position does not impact so much on binding with neutralizing antibody. So what are the steps in the wake of the variants of concern? We need to isolate and fully characterize viruses obtained from individuals who, despite vaccination, may have picked up COVID and are admitted in the hospital. Countries need to maintain active surveillance of active genomic surveillance of these different variants, which may be variants of concern. We need to create a central repository of sera from vaccinated individuals. We need to limit the spread of particularly the Brazilian variant and the South African variant. Also, today, vaccine companies who make mRNA vaccines, who make adenovirus construct replication deficient vaccines need to be ready to include key changes in the sequence changes of their the makeup of their vaccines. In fact, today Bharat Biotech, even though they make a whole virion activated vaccine, are tying up with us to take back from us the Brazilian variant and the South African variant as and when it is available, so that they can incorporate them in the making of their whole virion inactivated vaccine. And then, of course, the wearing of masks, frequent hand hygiene, and physical distance remains very important. In the era of this avalanche of COVID-19 vaccines, you can see that at least 181 are in the preclinical stage, with about 22 in phase 1, 26 in phase 2, 21 in phase 3, and at least 10 vaccines that have been approved for use. The color code here, what's in orange are nucleic acid-based vaccines. In dark blue is a virus vector-based vaccine. Adenovirus is an example of that. Protein-based vaccines are in red, and whole virion inactivated vaccines are in a lighter blue. And we can see that this is going to be a moving target, and we're going to see more and more of them getting licensed with time. So is India in the right direction? Today, we know as of yesterday, 1.23 crore vaccine doses were delivered to healthcare workers and frontline workers. All of us have gone through vaccination, at least most of us have. And this has been a very well-organized effort, and you're issued a vaccination certificate which says you have got the first dose or the second dose or vaccination complete. As of March 1st, we know vaccination of persons over 60 years and those with comorbidities of 45 years and over are going to receive the vaccine. The Union Health Secretary also announced that they will be taking in the private sector to help with increasing the vaccine coverage and increasing speed of vaccination. We also know a genomic consortium known as the India SARS-CoV Genomic Consortium, the INSACOG, has come into being from December, which includes eminent scientists from DBT, from ICMR and CSIR. And we also know at today, we are also performing 16 lakh tests per day across our country. So is India in the right direction? Yes. But with regard to vaccination, given the stupendous size of our population, we would probably have to get into seventh gear. There are some new approaches to vaccine delivery. These are not in practice in our country, I would like to emphasize, but these are initiatives being taken by countries like the UK, where probably they do not have enough vaccine doses. So where vaccines were initially licensed for delivery with a gap of 3 to 4 weeks, that means the first dose and then 3 to 4 weeks later the second dose, they are considering extending the interval between the first and the second dose from anywhere between 6 weeks to 12 weeks. The rationale that they claim is that this will ensure that at least a larger number of the population will get their priming first dose. And we know with some vaccines, increasing the gap between the first and the second dose has contributed to longer term efficacy of the vaccine. So this is the rationale they use. They've also been thinking of what we thought was not allowed, a mix and match of different vaccines. This is because of implementation logistics. It makes it easier, wherein they use a heterologous combination of, say, a Pfizer vaccine, and then which is followed up with an Oxford AstraZeneca, what we know as the Covishield vaccine, as the second boosting dose. So we mix and match two different vaccines. So it's called a heterologous prime boost. This is not being in our country, I once again emphasize, but this is being done in nations where they have they want to speed up the vaccination and where the morbidity and mortality is very, very high. So this is what they are going, their studies are underway. What is the COVID forecast? Yes, many modelers have many forecasts, many timelines, but very clearly the virus is here to stay. Even without testing or the vaccine, human behavior can impact hugely on where this virus will be several months from now. The future also depends on many unknowns. In all humility, we have to accept. The duration of immunity of this virus, we really don't know. How exactly will seasonality affect the, the peaks that might appear in different regions of the globe? And it will also be determined by the choices made by governments and the individuals in each country, which could put a stop to the rampage of this virus. So with that, I would say that COVID appropriate behavior remains the need of the hour, with respiratory hygiene, maintaining physical distancing, staying away from crowded places, not touching our face, eyes, nose, mouth, good hand washing, hand hygiene, and of course, the use of masks at all times. And so with this, I thank you for your patient listening.
Yeah. Thank you very much, Professor Priya Abraham, for excellent talk. And I think Dr. Abraham have very nicely explained about the different strain like South African strain, UK strain, Brazilian strain, and another Netherlands strain. And she have explained very well about the different kind of mutations. So I would like to thank her for excellent talk. And we are running out of time. We can have one or two questions. We already have to the time.
Can I make a comment?
Yeah, please come.
Yeah, we started with one lab for testing and went on to 1,500 labs perhaps. I do feel we need to ramp up our sequencing. The world is talking about 400,000. We are talking about 8,000, 9,000, despite the formation of INSACOM. I am only appealing that we need to ramp up the sequencing effort enormously. This is just a comment I want to make.
Yes, if I may respond to that, yes, Dr. Padmanabhan, I think your point is very well taken, and that is why the INSACOG came into inception, and they are ramping up big time. This is a DBT funded initiative where even ICMR and CSIR are also helping. We ourselves have gone through several hundreds of sequences. In fact, we were sequencing even before these variants of concern came to be. That is why we have several publications with whole genome sequences. And your point is very well taken. We have to ramp up and be, this genomic surveillance has to be in real 7th gear.
Any other comments or question, please? Okay, so if you don't have any comments, then I would like to close this session, and I would like to thank my co-chair, Dr. Padma, and I would like to thank all the speakers for excellent talk for this session. And I would like to thank NASI and Professor Manju Sharma for giving this opportunity.
Thank you,
Have
sir,
a good evening.
for chairing this session. On behalf of the NASI, I am very thankful to the chair, co-chair, and the speakers of this session for delivering such an informative lectures. We are our heartfelt gratitude to all the speakers for not only delivering the talks, but also their contribution in the respective fields. Thank you. Now the next session will be coordinated by Dr. Santosh Shukla. May we start the session, Madam Manju Sharma, Madam?
I think we can have the session. If anybody wants a tea break, they can just log out for 1 minute and then come back again.
Okay, so after 5 minutes, we will assemble here and the next session will be started. Okay, thank you all.
Thank you.
Excellent lecture.
Hello. Hello. Ma'am, Sally Avasthi, madam is online, ma'am. Is Dr. Sally Avasthi, madam, are you online? Ina Ravi, madam, namaste. Namaskar, madam.
नमस्कार नमस्कार नमस्कार.
I think Patro sir is also connected with us. Patro sir.
Yes, I'm here.
Sir, let me call to Sally, madam.
नमस्ते. Oh. तुम बोला.
Namaskar, Petro sir.
Namaskar.
I hope your health is all right now, much better.
Yeah. To a great extent, yes. There are still certain things remaining.
Mm-hmm. Well, we all pray for you.
Thank you so much, sir.
You can start now. Dr. Santosh Shukla, can you hear me? You can start the session.
Sir, but I am searching to Sally Avasthi, madam. Sir.
I'm
You are
here.
waiting for her.
I'm here. I'm here. Dr. Shukla, I'm here.
Yeah, okay, madam. Thank you, madam. Just I was trying to call you, madam. So, shall we start, sir?
Yeah, we can start.
Sir, yeah, welcome you all in this session 3 for this 90th session of National Academy of Sciences on the towards a new healthcare regime for the nation. Sir, this session 3 is chaired by respected Professor Ishan Patro sir and co-chaired by Dr. Veena Ravi madam. Professor Patro sir is presently in the Department of Neuroscience, Jivas University, Gwalior, and he was former Vice Chancellor from the Ravinsha University, Cuttack, Odisha. Honorable Dr. Veena Ravi madam is also Professor, Department of Surgery, AIIMS Rishikesh. She is also with us. So I request Professor Patro sir and Veena Ravi madam to kindly start the session 3, and the learned speaker Dr. Sally Avasthi and the Vijay Lakshmi Ravinathan will deliver her talk. So kindly invite our first speaker, Dr. Sally Avasthi, with her formal introduction. I request to Patro sir. Sir.
Yeah. Good afternoon. Namaskar. I'm extremely grateful to Professor Manju Sharma ma'am and Professor Padmanabhanji for giving me this opportunity to chair this session of NASI. And we are a little bit unfortunate that this session could not happen in Ravinsha University, where it was supposed to be. Due to COVID, we could not really do that. and now we are all here. It's anyway, it is a good thing that we are all doing it from our headquarters, NASI, and all respected former presidents and senior faculty fellows, Professor Vijay Lakshmi Ravindranath and Professor Sally Avasthi, who are going to speak, and the members of NASI, ladies and gentlemen. It's my pleasure to introduce the first speaker. Before that, I should say that the co-chair for this session is Professor Veena Ravi, who is a senior professor at Department of Surgery, AIMS Rishikesh. And the first speaker will be Professor Sally Avasthi, and she is presenting the Dr. Mridula Kamboj Memorial Award Lecture 2020 on the topic management of possible serious bacterial infection in young infants and an innovative approach. Professor Avasthi is a professor and pediatric pulmonologist working at King George's Medical University, Lucknow. She is an elected fellow of all major Indian science academies like the National Academy of Sciences, Indian Academy of Sciences, National Academy of Medical Sciences, and Indian Academy of Pediatrics. The Department of Science and Technology, Government of India, honored her with the National Award for outstanding efforts in science and technology communication through innovative and traditional methods in 2016. She is a recipient of several awards, including Basanti Devi Amit Chand Award, Dr. H.B. Dingley Memorial Award, 1996, by the Indian Council of Medical Research, Government of India. May I request Professor Sally Avasthi to present the oration, please.
Thank you very much, sir, for your very kind introduction. I would like to share my screen. I hope I am able to do that. Okay. Can you see my screen? Is my screen visible?
Yes, ma'am. Yes.
Okay.
Yes.
So I would like to thank the organizers, the National Academy of NASI, and I owe my gratitude to Dr. For the Oration Award, Dr. Mridula Kamboj. Dr. Mridula Kamboj was a very renowned obstetrician from Lucknow, the city from where I am, and I know many people who have been her patients, have been treated by her, and in her memory, I'm getting this award. It's a proud privilege for me. Thank you, Nasi, and thank you, Dr. Kamboj and her family for instituting this award. So I would be talking about management and treatment of possible serious bacterial infections in infants, young infants, in low resource settings like India. And I would be sharing the experiences we have for this. So as you know, India has been struggling tenaciously to tackle morbidity and mortality among neonates. Now, we have a birth cohort of about 25 million newborns every year. And among these 25 million newborns, as you can see in the pie chart here given by UNICEF, 33% deaths are due to neonatal infections. Now, if you see this box here down below, the under 5 mortality rate in India is 42. In Uttar Pradesh, it is almost 60. The neonatal mortality rate in India is 23.5. In Uttar Pradesh, it is 37.1. So we are having high neonatal mortality, and this is the setting where this work was done. Now, what is the pathogenesis and epidemiology of neonatal sepsis? Because that is the leading cause of neonatal death. In approximately 2% of new infants, this infection is intrauterine. However, 10% get it in the neonatal period. Either it is transmitted vertically or after birth. This can happen as it ascends the maternal vaginal canal or by the environment in which the infant is there. There is immunological prematurity in the neonates, with preterms being more immature than terms. The diagnosis of sepsis is difficult because of presence of comorbidities in the neonatal period. And the neonatal sepsis has a wide spectrum of manifestations, from intrauterine sepsis, to subclinical sepsis, to severe sepsis, focal or systemic manifestations, even syndromic manifestations. And it also depends on the time of exposure, the inoculum size, immune status of the newborn, virulence of the organism, which all affect the clinical picture. Many a times, maternal infection goes undiagnosed. Hence, the newborn is infected, whereas we could have treated the mother and prevented the infection. And to top it all, there's a wide variety of etiological agents, bacteria, virus, fungi, protozoa, including mycoplasma. So why are neonates having low immunity? One, their immunoglobulin levels have problems. The IgG is transmitted through the placenta. It is produced from the third trimester in the uterine life. It is effective for opsonization of gram-positive bacteria. The cord blood IgG is proportionate to gestational age. However, IgA, IgM, IgE, and D do not cross the placenta. And with decreased IgM level, these newborns are predisposed to gram-negative sepsis. Also, complement, which is synthesized in the fetus from 6 to 14 weeks of gestation, doesn't cross the placenta, maternal complements won't help. There is diminished classical complement pathway and impaired alternative pathway, reduced C9, which predisposes to sepsis by E. coli and group B streptococcus. Likewise, neutrophils have decreased function and there are decreased stores in the bone marrow. So you can't even mobilize them from the bone marrow. Natural killer cells, a subgroup of lymphocytes, which are cytotoxic to virus, in cells infected with virus are decreased in number and activity. And the cytokines, there's a disbalance between pro and anti-inflammatory cytokines. Th1 response is impaired. Hence, all these lead to severe sepsis. So this results, what we see now is under 5 mortality in India has decreased from 83 in 2000 to half, 42 in 2017. Neonatal mortality has not reduced to that extent from 38 to 25 per 1,000 live birth. And there is a substantial variation between states in the magnitude and rate of decline in mortality. Even higher variation between districts of India. So, the child survival targets are 25 deaths per 1,000 live birth for under 5 and 12 deaths per 1,000 live birth for neonatal mortality set by the United Nations for 2030. To achieve this, we have to do something drastic, bring about a drastic reduction in mortality due to sepsis. This map is showing that in 2020, all the red dots are areas which have high burden of neonatal mortality. And when you come to 2017, some of the red areas have cleared, but yet a lot of work has still to be done to reduce neonatal mortality. So the sepsis, a study was done where India was also included, published in the Lancet 2018, where approximately 10% of newborns had sepsis, called possible serious bacterial infection. Only in 28% of episodes, etiology was found, 16% bacterial, 12% viral. The leading pathogen in the virus was respiratory syncytial virus, followed by ureaplasma. However, among babies who died, cause was attributed in 46% of cases, and in 92% it was bacterial. Therefore, we are likely to prevent deaths which are due to bacterial sepsis. And the bacterial isolates were susceptible to drugs like penicillin, ampicillin, gentamicin, or a combination of these drugs. So we did a work which was identification and management of young infants with possible serious bacterial infection, where referral was not feasible in rural Uttar Pradesh, funded by the World Health Organization through Bill and Melinda Gates Foundation. For those who don't know, the Indian health system, we have a public and a private system. The public system has primary, secondary, and tertiary care. In the primary care, you have the community health center for 50,000 plus population, primary health center for 30,000 population, and a sub-center. There's a sub-center for 5 villages. And when we are talking of in rural India, we're talking of the villages of India where these births are occurring. Majority of the births are in government facility. Yet, within 48 hours, these mothers go home with their infants. So the rationale of this study was that WHO and Government of India recommends postnatal visit by healthcare provider to facilitate early identification of danger signs and promote care seeking. Hence, our government launched home-based newborn care program in 2011 through the ASHA worker, who has to visit 6 times in case of an institutional delivery, day 3, 7, 14, 21, 28, 42, and additional visit on day 1 if the baby is born at home. Despite this, NMR is high. Therefore, implementation research was done to evaluate acceptability, effectiveness, scalability of simplified treatment to cases of PSBI where referral is not accepted in HPNC programs. Now, how do you identify PSBI? The signs are so subtle that you have to be very alert. Either the baby who has been feeding after birth stops feeding. Baby has convulsions, fast breathing, more than 60 breaths per minute in infants less than 7 days of age, severe chest indrawing, fever, low body temperature, or the infant doesn't move or moves only when stimulated. And the other danger signs are fast breathing, again above 60 breaths per minute, red draining umbilicus, skin pustules, diarrhea, and yellow palms and soles. So it's not easy to identify PSBI. So therefore, we trained the ASHA workers to identify PSBI. We retrained them. And when the PSBI was found, all the mothers were said, please go to the hospital. Please go to a government hospital. And let me tell you, in red it is written, majority did not accept referral to hospital for various reasons. So when the family did not accept referral, their criticality was reassessed. Very critically ill baby who was convulsing, no movement at all, on even stimulation or not feeding, very sick, had to be rushed, but family said no. Clinically severe infection, less movement than normal, not feeding well, severe chest indrawing, hypo or hyperthermia, and in less than 7 days old when there was severe pneumonia. So we offered these children whose parents did not go to the hospital despite persuasion, simplified treatment, oral antibiotic amoxicillin plus intramuscular gentamicin when the child had severe pneumonia or the child had clinically severe infection. But when the child was critically ill, instead of amoxicillin, we recommended use of injectable ampicillin or gentamicin. And those who were just fast breathing were given only oral amoxicillin for 7 days. So our primary objective was, can it be accepted? Is it effective? Can we scale it to other districts of UP and India? And we did it in 4 geographical blocks, which had about 1,000 square meter area with about 8 lakh population from 2017 to 2019. There were approximately 25,000 pregnant women and 25,000 live births. It was a big project. There were consultations and approvals by policymakers at the state, national level, state level. Upgradation of skills of the healthcare workers were done across all cadres. Infrastructural strengthening was done, and we did supportive supervision throughout. Training of 700 plus ASHAs, 137 ANMs and staff nurses, medical officers, pediatricians, and There were master trainers from our institution. So what did we find? In the home-based newborn care, they are supposed to visit the baby within first week. We divided our project timeline into establishment phase of 3 quarters and implementation phase of 4 quarters. You are seeing in orange live births and in green those who have been visited in the first week where they're more likely to be sick. During the implementation phase, only 3/4 of the babies were visited in the first week. And this visit was largely a token visit where the Ashas went to the door and said, "Bacha theek hai," and came back. They did not do all the components they were supposed to do. Then in the second quarter, not only the percentage being visited improved from 75 to almost 90%, the quality of visit had substance and they did what they were supposed to do. So you see the result. In the first quarter of establishment phase, only 1.2% cases of PSBI were found. You know, the global standards are that there should be 10% cases of PSBI. In the second quarter, 3%, 3%. And in the implementation phase, it did not reach what we thought was 10%. It ranged from 6.5 to 8%, double that in the establishment phase when they started actually going and doing quality visit. Now, how do you know they did it? We observed them, we documented what they did, but we also assessed the change in knowledge of mother, because when these Ashas go, they're supposed to counsel the mother. When we went early in baseline, the mothers, you see here, 23% mothers did not know and they were given no information about danger signs. But at midline, this reduced from 23 to 8%. And all the individual components of illness was improved. Therefore, yes, they were counseled. The Ashas started doing what they were doing. Likewise, even though the Ashas were trained by the government, their knowledge was deficient by virtue of fact they were not practicing what they were taught. They were not shown what to look for. There was no hand-holding. And with hand-holding and reinforcement and revision, they started recognizing danger signs and they started performing. So what happened as a result? As a result, you see that in the 25,000 newborns, 1,302 had PSBI. All were referred to higher center. And the starking figure is that only 7% were ready to take the child to a government hospital. The rest said, we won't go. The mother is there. She's just delivered. Who'll look after her? And 93% did not accept referral. To them, we said, okay, we will offer you simplified treatment. Stay where you are. About 3/4 accepted simplified treatment. 20% refused and went to either private providers or went to traditional people. And there were a few which said, okay, we will not treat this child. That was the sad part, despite counseling. Now, what happened when we offered them simplified treatment to 959 who were offered simplified treatment? You will see that here, simplified treatment was offered to about 650 children with clinically severe infection. Majority of them, 94%, were cured. Critical illness, 72% were cured. And when you had fast breathing, almost 100% were cured. And those who had severe pneumonia, 100% were cured. So here, in the real life situation, if parents are not going to the hospital, we have something which was proven in other countries, unproven here, unpublished and reviewed, that it works, we should give it to them. Here you see, these were patients with danger sign, about 200. If you combine PSBI and fast breathing, the numbers come to 1302. And then there were other causes of those who are pediatricians will understand that there were jaundice, diarrhea, local bacterial infection in these children. And again, you see, I told you that in these children with PSBI, in those who were clinically severe infection, 9 died. Remember that? In critical illness, 16 died. You see here. Even though 9 died, in real life, among those who had clinical severe infection, 39 died. 30 deaths occurred in those who did not accept simplified treatment, went to either private provider or took no treatment, or were very ill. Same for critical illness. With simplified treatment, 16 died. 94 died elsewhere, who did not accept simplified treatment or who were very sick. And of course, for severe pneumonia, there were no deaths at all. Now, another interesting analysis I will show you, when the patient had only one sign, only 3% died, sign of illness. 2 signs, it increased to 14%. More than 2 signs, it increased from 14% to 22%. So as you keep having more and more and more signs of illness, logically, the chances of death are higher. And if they go to a district hospital late in the disease, when 1 sign becomes 2 and 2 becomes 3, even the best district hospital, best setup, will have mortality in our centers. So what I'm trying to say is, then we went back and interviewed. There was a finite time. The doctor said, we will continue simplified treatment. It works. The A&M said, yes, we will revise, we will give injections. They were not giving injections so far. So we concluded this innovative, simplified treatment, where in the public program in Northern India, does work. It gives good cure rates. When it gives good cure rates, we require system strengthening, we require supportive supervision. And when we finished this work, we trained the paramedic, the doctor, the nurses of all the adjoining districts. And we were very happy to note that addendum to operational guidelines came out, which recommended use of gentamicin by A&M for management of sepsis in young infants under specific conditions. What were the specific conditions? Specific conditions were when the parents refuse, where referral is not feasible or refused, medical officer can give it. This was legitimized. And under the current program, SAS, which has been launched in Uttar Pradesh last week, this is an integral part of management of these children with SARS. So our results were widely published. Simplified treatment can cut neonatal mortality. And we calculated and we said in this birth cohort of 2,500, there would have been many more deaths in 21 months. And if you calculate by the mortality rate of Uttar Pradesh at that point in time, there was a 40% reduction in neonatal mortality. We agree this was very intensive, because even though the work was done entirely by the government, we were there as a technical support unit for supportive supervision. However, I feel that now that the government has taken it in their own hands, they are likely to get similar or even better results. And I think within the next 2 to 3 years, we would have required 40% or more reduction in neonatal mortality. Similarly, it was covered everywhere. Now, what is the future? Future is that in states and districts where neonatal mortality is high, this nationwide implementation has begun, and we should do it with a lot of enthusiasm to achieve the SDG. However, I must tell you that in the Lancet study, in 3/4 of episodes of PSBI, no cause could be attributed. Bacteria could be attributed in about 16 to 20%, and among all those who died, 92% were bacterial. Till we develop and start using metagenomic approaches to facilitate decision making, and we have invested in saving lives among very young babies, we cannot limit unnecessary antibiotic use, and we have to continue treating them with antibiotic. But I should say sooner than later, we have to start using metagenomics to have algorithms at point of care to decide which young infant has sepsis, bacterial in etiology, which is non-bacterial in etiology, and then use antibiotics with caution so that we prevent emergence of antimicrobial resistance in the community. The innovation is home-based treatment where treatment is refused. It has been recommended by the government of India. We will use it, but we understand that much more work has to be done in as basic science and in translational field so that we can benefit our community. So with this, I would again like to thank the organizers, and I bring greetings for my medical university. Thank you all very much for your listening. Thank you, sir.
Thank you very much, Dr. Awasthi. It's a wonderful lecture. And now I would invite Professor Vijayalakshmi Ravindranath.
Sir, sorry, sir. Before inviting the next speaker, I would like to request you to kindly felicitate our this speaker as well as the awardee of Dr. Mridula Kamboj Memorial. I would request to read the citation for the awardee.
Okay. Please go ahead.
Yes, sir.
Screen, Santosh.
Madam, just may I request to the Beena Ravi madam to kindly read the citation so that we can felicitate the awardee. Even this is because this is virtual mode. Otherwise, this prestigious award is being celebrated in a very good grand way because Dr. Mridula Kamboj Memorial Lecture Award was instituted in NASI year 2016, and it is conferred to so many prestigious people like Dr. Earlier recipient of this award was Dr. MD Padma Srivastava from All India Institute of Medical Sciences, as well as Dr. Smita Mahale. And in this award, the awardee will be given a medal, a citation, and a cash award. So we will send the medal and citation with the cash award to the address of the awardee. I request to Dr. Beena Ravi madam to kindly read the citation to the Dr. Sparsili Vasturi.
Thank you very much, Nasi. Dr. Sharma, madam. Dr. Shelly Avasti is my junior and a very dear colleague also. I can read the citation now. The National Academy of Sciences of India, Dr. Mridula Kamboj Memorial Lecture Award for year 2020 has been conferred to Dr. Shelly Avasti, Professor and Head of Pediatrics, KGMU, Lucknow, for her significant scientific contributions in the applicability of innovative and traditional methods in pediatric pulmonology. Thank you very much.
Thank you very much, madam, and thank you very much, Nasi. Thanks, ma'am.
Congratulations.
Thank you, ma'am. Thank you, madam Manjuji.
Thank you very much, madam. And on behalf of NASI, we congratulate you and thanks for sharing your time. Now I request to the chairperson, Professor Patru sir, to kindly invite our next speaker, Dr. Ravidath madam, and give a brief introduction, sir.
Congratulations, Professor Awasthi. Now I invite Professor Vijayalakshmi Ravindranath to present our Professor MGK Menon Memorial Award Lecture 2020 on aging and dementia, mechanisms to interventions. Professor Vijayalakshmi Ravindranath is an eminent neuroscientist and is currently a professor at Center for Neuroscience, Indian Institute of Science, Bangalore. She was the founder director of National Brain Research Center, Gurgaon. 2000 to 2009, she was the director of the center. Then she founded the Center for Neuroscience at Indian Institute of Science. She is an elected fellow of several Indian academies like National Academy of Sciences, National Science Academy, National Academy of Medical Sciences, Indian Academy of Neuroscience, and the Third World Academy of Sciences. She also has received many awards like Santiswarup Bhatnagar Prize for Science and Technology for Medical Sciences, K.P. Bhargav Medal of Indian National Science Academy, Om Prakash Bhasin Award for Science and Technology, and Padma Shri Award in 2010, to name a few. I welcome and request Professor Vijayalakshmi Ravindranath to present her lecture, please.
Do you see her?
Good afternoon, everybody. At the outset, I want to talk.
We want to see you.
Madam, I'm there. चेहरा बदल
वीडियो
गया
ऑन करिए.
ऑन है मैडम
नहीं.
Yes. अभी
हाँ अभी ठीक है.
Good afternoon, everybody. And at the outset, I would like to thank the Council of NASI for bestowing this absolute honor on me. For me, I do not know how I should react. Receiving Professor M.G.K. Menon Memorial Award is, I think, so precious, a deep personal honor. Professor Menon, as we all know him, of course, was an unparalleled scientist, an efficient and empathetic leader, and of course, a visionary scientist, a visionary in science and technology. My own interaction with Professor Menon began when I started working towards the establishment of NBRC in 1997, 98, and Dr. Manju Sharma, Professor Tandon were helping and advising me, and there began my interactions with Professor Menon. He guided me on how to navigate the myriad ways in the government, as I had to go through several processes for the establishment, and gave me the big picture that I needed to always think about. Dr. Manju Sharma was always on hand. I can never, ever forget those times. And Professor Menon was just an awesome mentor who understood the need for an interdisciplinary approach to understanding complex biological system. And he encouraged me as I started building an interdisciplinary team of people to study brain and understand its function. In addition to the mentoring that he did and advice that he gave us, I think what I remember most about him is his utmost kindness. There were times at NBRC when I was very dejected, and I remember him calling me one night and saying, "Please don't stay there alone. Come to my house and Indu will take care of you." Induji, his wife. That's the kind of person he was. And even today, as I was putting it together, tears were in my eyes as I thought through all those occasions, our travels together to Allahabad, where all of us would be around him and he would be telling us stories. And I pay my homage to Professor Menon today and thank him for a large part of making me the person I am, who could set about and build NBRC from scratch. And I think it is his teachings and his mentorings that I still follow today as I go on to build new enterprises in public-private partnerships, as I am doing with the Center for Brain Research today. Thank you all, Professor Padmanabhan, Dr. Manju Sharma. Thank you so much for this absolutely deeply personal honor. Thank you. So today I will be talking about aging. That has been the part of my research for the last 35 years, ever since I came back from NIH and started my own lab. And as we look at how a person ages, we definitely understand how complex it is and how different components play a role in the way our cognitive ability changes, and how our life experience and education, diet, exercise all impact on the aging process, which we see as changes in our brain structure as well as our brain function. Of course, of consequence and importance to us are the neurodegenerative disorders, such as dementia, Alzheimer's dementia, Parkinson's disease, motor neuron disease. And most of these diseases, although the genes play a role, either as a risk gene or as a part of a complex network of genes that impact us, the environmental factors and how we live our life often has a role in the way we age. If we look at this slide, that was this data that was put out by WHO and the World Alzheimer Report in 2010, it predicted in the years to come, the low and middle income countries will contribute to new cases of dementia and people living with dementia. And because of our large population base, the number of elderly in our country will rise dramatically. We were 104 million, we'll be 173 million in 5 years to come. And by about 2050, 20% of our population will be the elderly people. And this is true for a country which is considered as a young country, because substantial number of the population of the country is young today. One of the things we learned in the last, I should say, decade is that although dementia, and including Alzheimer's disease, manifests in the 60s and the 70s, the 6th and 7th decade of our lives, almost 2 to 3 decades before, changes in the brain take place. This happens in terms of accumulation of amyloid, dysfunctions in the connections and transmission of signals in the brain, the synaptic dysfunctions, injury mediated by other pathogenic proteins such as tau, structural changes in the brain where hippocampal volume decreases. And of course, this then is manifested as changes in our memory initially and other cognitive functions such as decision making, judgment as the disease progresses. And it is only in the 60s and 70s that the patient is hampered enough to go to the physician, or the caregiver often takes the person. So in effect, I often say that we pay the price for how we are, or what illnesses we get in our 60s and 70s on the way we have lived our life in our 30s and 40s. This is something that is totally new in our understanding. The disease starts much early, and we need to start tackling this right at that time, because today we have no cures or treatments available. Now, in India, there are certain very unique profiles. We really do not know what is the genetic susceptibility and what is the occurrence of that genetic susceptibility. We have tremendous diversity in terms of language, education, social, economic backgrounds, and also urban and rural India, which are literally two Indias. Risk factors such as diabetes, hypertension, dyslipidemia, midlife obesity, smoking, lack of exercise, all of these contribute. The differential cognitive engagement with elderly living alone rather than in a joint family system, as was common, say 2-3 decades ago, also play a role. How do we understand what is happening, and how do we bring in measures to prevent this? For this, the only way to do it is to start looking at and studying people as they age and understand what are the risk and protective factors that preserve our cognitive function while we age, as also offer the risk on us as we age. While such studies have been done in the West in the last 50, 60 years, I think the most famous among them is the Framingham study, which changed the way we treat heart disease. Such studies are completely non-existent from low and middle income countries. And I should also add that even in the West, those studies that people have done have been in urban middle class educated people. We do not have studies anywhere in the world that looks at the rural, marginalized, low literacy people and how they age and what are the problems that they have. So knowing that $15 billion that have been spent in research have been set aside, they haven't yielded any new treatments for Alzheimer's. We were now at CBR, we felt that studying human subjects as they age and taking that information into mouse models and reductionist studies with mouse models and cells could help us bring in new interventions that would help the elderly. So the longitudinal studies we all felt were important, and a major thing was to identify risk and protective factors. I should now tell about the true extraordinary philanthropies that came our way, which helped lay the foundation for such studies. In 2014, Mr. Ratan Tata, through the Tata Trust, gave a very generous grant of 75 crores for us to look at Alzheimer's disease and especially early phases of the disease. And this grant also supported the setting up of a 3 Tesla MRI, a state-of-art, the only one in the country, Prisma. This is a new MRI, as well as a whole genome sequencing platform along with the necessary computational infrastructure. Subsequently, Mr. Gopalakrishnan, the co-founder of Infosys, through his trust, the Pratiksha Trust, gave a gift of 280 crores that has enabled us to establish this Center for Brain Research, this building that is going to be ready very soon. We plan to do it next week. And with these two philanthropies, we initiated initially the Tata study of longitudinal aging and then the rural study. So the Tata study of rural aging that we started in 2016 looks at individuals from urban Bangalore, mostly middle class, highly educated, from 45 years onwards, but the average age of the cohort is about 70. And although it is study is conducted in Bangalore, it has people from all over the country represented in this study. And I'll tell you briefly how we go about doing this study. Whereas the rural study, the parallel rural study that is done through Pratiksha Trust funding is done in this taluk of Srinivaspura in Kolar district in Karnataka at about 120 kilometers from Bangalore. And this comprises of rural agricultural workers, again age 45 and upward, that we study exactly. Both these studies are perfectly harmonized, helping us to look at both urban and rural burden of the disease. So the features are, we picked Srinivaspura Taluk because there is low degree of migration. They are all mango orchard farmers, low literacy, low socioeconomic strata. They are mostly bilingual. They speak Kannada and Telugu. And their lifestyle has not altered through these years. And our target cohort size is 10,000. We have now recruited 2,000. And currently, it's about 1850, and it began in 2018. The Tata study, on the other hand, has migrants from other states. Their education is quite a large number of them are above 15 years, middle class, white collar jobs, multilingual, major lifestyle changes in the last 3 decades. The cohort size is 1,000, and currently we have 500 with follow-ups, and it began in 2016. What do? We first run awareness program, and we tell them about our research. We go to retirement communities in Bangalore. In the case of the rural study, we work with the primary healthcare professionals, especially the ASHA workers, and we run awareness camps in villages. And this is followed by a survey at home where we tell them what we are going to do and take their informed consent as per our ethical norms. Then in the case of the urban, they come to IISc where we put them through a clinical assessment, cognitive assessment, blood investigations, genetics, which consists of both genome association studies as well as whole genome sequencing. We have the MRI done at IISc. In the case of the Kolar people, after the survey, we have established a site office in the Taluk headquarters where they come for clinical assessment by a trained physician, cognitive assessment. The blood is collected in a camp. We go at 6 o'clock in the morning. Our team leaves Bangalore at 3 a.m. and goes and runs the camp where we collect fasting blood samples. And we also give the reports of the previous blood camps, and they get a small consult with the physician, because if they detect any hypertension, diabetes, we inform them at once so that they can go to a physician. Of course, blood is brought back for genetics, and they come, a subset of them come to IISc for their MRI. And then this is repeated in a cyclical manner every few years. And in addition, we do a cardiac workup. We are going to start the abdominal ultrasound to look at the fat, belly fat, and also looking at optical coherence tomography and eye vasculature and pulmonary function. These are the new things we are adding also to this. So as you can see, this is a pretty complex study, and I'm very happy to say that from the minute we make the contact, it's all done completely on a tablet. Everything is digitized, and the data seamlessly flows into CBR servers. These are the variety of clinical assessments that we do. I just wanted to give you a brief glimpse of how deep the study is. And for the cognitive assessments, we look at attention, memory, language, and visuospatial ability. The tests are adapted and also translated language that the subjects can understand. So this is just telling you how all this data comes back into the CBR database for data integration and, of course, then for analysis. So if we just look at the males and females, between these two, we have pretty much equal numbers. Of course, the SANSCOG cohort is a little younger than the Tata cohort, but generally the average age is about 58, and it is 66 in the Tata cohort. And of course, one has to remember that the lifespan, rural lifespan, is less than the urban lifespan. This is where the difference starts showing. In SANSCOG, 70% of them have less than 7 years of education. And I should mention here that in the 70%, most of them are women. Illiteracy is much higher in females as compared to males. Of course, in the urban cohort, this study particularly has very well-educated people. Now, if we look at the medical comorbidities, we find that hypertension, diabetes is very low in SANSCOG, whereas it is very high in the Tata study. So 42% of the study subjects have hypertension and nearly 30% have diabetes. This is one very big difference that we see. Now, if we look at the MRI, we have done 284 subjects for MRI in the urban cohort, but the rural cohort, it's much less. We have done only about 9. And what we look at is, among other things, one important factor we look at is this so-called white matter hyperintensity that you see as bright blobs in the MRI. These indicate that the blood vessels to the brain, especially the small vessels, have infarcts, or they have had mini strokes across the brain, and which accumulate over a period of time. These are small strokes, don't directly cause a deficit in large vessel strokes, but over a period, these do impact our cognition. So if my colleague Vivek Tiwari, this before that, we also see that more the white matter, it has a direct correlation with the deficit in cognition. That is, more white matter leads to more cognitive deficits. And we see that in the Tata cohort, 61% have ischemic changes in the white matter. And my colleague Vivek Tiwari, who joined CBR recently, has went ahead and started doing the segmentation and quantitating the white matter. The first thing we see is 80% of SANSCOG cohort between the ages of 45 and 54 have white matter hyperintensity, a number that you only see at 65, above 65 years of age in the rural and urban. So that means the white matter hyperintensity burden that you see in an individual age 65 and above in the Bangalore city is seen, the same amount is seen in the rural cohort between the ages of 45 and 54. That means the burden starts much early and builds up. And this was quite a shocking revelation to us. And then when Vivek did some careful analysis, we found that in this young age group, 45 to 54, the amount of white matter hyperintensity in the rural cohort is actually double that of the urban cohort. That means either genetics, or is it the impact of smoking, which leads to decreased pulmonary function, or environmental pollutants that have caused this big change in their brain at a very early age. What Vivek has now found is that the decrease in gray matter that happens with aging in all of us, that decrease is far quicker in the rural as compared to the urban, showing that the structural changes in the brain is occurring far earlier, or the rural cohort is aging much more quickly and at a much younger age as compared to the urban cohort. So when we looked at this age prevalent of mild cognitive impairment and dementia, this is where, so the psychiatrists who evaluate our subjects also grade them in the clinical dementia rating. So clinical dementia rating of 0 means we are cognitively normal. 0.5 indicates that we are starting the mild cognitive impairment. And if we take 1 and above, we have full-blown dementia. And what you see here is at the age of 55 to 64, the burden of CDR more than 0.5 is 3 times in SANSCOG as compared to the urban. That means the rural people have a 3-time higher burden at a younger age as compared to the urban, and it is statistically significant. If we look at the next stage of 65 to 74, it is little more than double. Again, the same trend continues. It is only beyond 75 years that we see that they both even out. What we are showing by this is the rural people live longer with dementia. They get the cognitive impairments much early and live longer with the disease as compared to the urban who get it only far later in their 70s. Whether this is an impact of education or an impact of the kind of changes we see in their brain in terms of vascular dysfunction, that is the small vessel disease as we call it, or because of the more rapid gray matter loss, or because of the pollutants, is something that we need to figure out as of now. But these studies and the results are pretty startling because such data has never been seen before, and we have rarely understood the burden of the elderly in our rural settings. Of course, on 13th March last year, our whole study completely stopped. Both our studies stopped. We continued to maintain telephonic contact with subjects and inquired about their well-being, and also made sure that they were given adequate help through the telephone calls, and we linked them with the primary health care. But an important thing we did was we conducted telephonic interviews that would help us grade their depression, called the geriatric depression scale. And what we found here is again a startling revelation. So when you looked at the prevalence of depression among the elderly, again in the rural, you saw that about 7.7% had in the rural cohort had depression. But post-COVID, this number went up almost 3.5-fold to nearly 29% because of the pandemic. We also did the anxiety score, and that did not change. And interestingly, among the urban elderly, when the same study was conducted, there was no difference in the depression or anxiety in them because of the pandemic. I think they felt safe, and they knew that as long as they stayed in their homes, the urban elderly were reasonably safe from the pandemic. But in rural, this increase in depression is, we believe, is mostly because of the tremendous economic setback that they suffered. But we will be following them up because we are very concerned. Depression is a forerunner of dementia. And one is definitely concerned that this increase in depression should not lead to increased dementia in the rural setting. So what I'm trying to tell you by this is there are two Indias that are widely different in the way they manifest, in the amount of burden they carry. And of course, it's also a very big challenge on how we can reach out to them and correct very early on the problems that occur. So what we are now doing, even as we speak, because our studies have begun and they are back to the pre-pandemic levels in the terms of speed, is to actually start scanning the brains of people who are younger than 45 and see how early the vascular starts, and then try to bring in corrective measures through diet, exercise, and cognitive therapy, and also, pulmonary deficits if they have, to see how we can correct this early on before this burden transforms itself to dementia. At this point, I also want to tell you briefly about Genome India. This was an initiative that we thought about and had the first discussion meeting at IISc to start cataloging the genetic variations of the Indians across different ethnicities and genetic lineages through whole genome sequencing. And I'm very glad to tell you that the Department of Biotechnology funded us. It's a consortium of 20 institutes across the country that are involved in sample collection, sequencing, and data analysis. And the major goals of this project are to sequence 10,000 Indians, prepare an exhaustive catalog, and try and do genome-wide array specific for Indian population so that genetic tests can be done at reasonable cost. We are planning a biobank, and this I'm happy to tell you, in spite of the pandemic, we have collected over 3,000 samples, and as we speak, the sequencing and analysis is going on. And in the end, what I want to mention, I want to thank all the three places that I have worked for the last 20 years. But more importantly, I want to talk to you a little for a minute about the new initiative we'll be launching at CBR. We have now realized that aging and brain disorders of the aged brain are very important areas to focus on. We now plan to start funding others in the country to also start working in this area. And these funds will come from the Pratiksha Trust that is set up by Mr. Chris Gopalakrishnan. He has been extremely generous with his funding from his own personal money. And importantly, he has also been a very proactive and donor who is eager to see that we reduce the burden of the elderly in our country. I should also mention that I have not given up my lab work. We continue to take insights from this. So we have now developed a vascular dementia model, and we are examining the molecular mechanism. So that hasn't gone from our lives. But I thought I would present to you some new data that has come in, and also invite the other scientists in the country to participate with us and join in our mission for reducing the burden of brain disorders in elderly. Once again, my homage to Professor Menon and my heartfelt thanks to NASI. Thank you.
Congratulations, Professor Vijayalakshmi Ravindnathji, and it was indeed a very interesting and very informative lecture. And what I love the most is your invitation to people to come and join in this venture. But definitely very much overwhelmed with the kind of neuroscience you have encouraged in this country. Now I would request Santosh to get the presentation done. May I request Professor Ravi to read out the citation, please?
Sir, before reading the citation, may I request to the chairperson and madam to kindly allow me to share few slides of Honorable Professor Menon sir, because this is a prestigious award constituted by the Academy in the from 2015, and this prestigious award has been conferred with many prestigious scientists like Professor Priyan Tandon sir, Professor Govind Swarup sir, Professor Manju Sharma madam, Professor K. Kasturi Rangan, and earlier this Bharat Ratna Professor C.N.R. Rao sir was also the recipient of this prestigious award. So if you would allow sir, I want to share few slides, then we read the citation sir. Yeah, Professor Menon sir is one of the most renowned scientists and architect of the Indian science and technology, and need not any introduction. Just I want to share few photographs which are self-explained, sir. Thank you very much. Thank you, sir. Thank you for allowing me. Many thanks. And now I request to share the citation to read by Dr. Beena ma'am for the Dr. Ravindra. अरे सर ये हट नहीं रहा है. दीपक अब देखो हो गई प्रॉब्लम यार, हट क्योंकि ये शेयर हो रहा है ना।
शेयर नहीं हो रहा, आपको स्केप आपसे बाहर आई है, कोई प्रॉब्लम नहीं।
साइटेशन होगा ना।
बीना जी को भेज तो फिर ऐसे ही पढ़ेंगे।
Yes, got it now. Thank you. The National Academy of Sciences of India, Professor M.G.K. Menon Memorial Award 2020 has been conferred to Professor Vijayalakshmi Ravindranath, Director, Center for Brain Research, Indian Institute of Sciences, Bangalore, for her significant research contribution in the area of neurosciences, toxicology, and pharmacology, especially in psychoactive drugs and environmental toxins. For you, Dr. Professor Vijayalakshmi.
Thank you very much, madam. Thank you very much.
Can we ask some questions, Madam Vijayalakshmi?
Yes, madam. The awardee will be given a medal, citation, and cash award. And because we are in the virtual mode, so we will send the awardee the citation and medal with the cash award to her address.
Thank you very much. Deeply indebted to the Academy and all of you. This is just, as I said,
Congratulations, Vichi.
Thank you, madam. This is deeply personal. This is deeply personal. Thank you so much. Such an honor.
So wonderful
Thank you.
to see you again. And
Thank you all. Thank you very much. Padmanabhan, thank you so much.
Congratulations to both the parties. All the very best.
so, can we conclude today's program?
Hearty congratulations, Viji.
Neeraj?
Thank you, Dr. Mishra. Thank you.
Neeraj?
Yes, madam. We can conclude.
So, I want to tell all the distinguished speakers that today's symposium program is concluded, and we start our symposium again tomorrow between 9:30 to 11. We will have the first session. I mean the next session. So tomorrow we have to start early at 9:30 because the sessions are too long and we won't be able to finish otherwise. So please join us at 9:30 tomorrow. And the first lecture will be by Professor Sudhir Kumar Satpathy from Bhubaneswar. He will be speaking on the public emergencies.
Hello.
So thank you all, and we meet tomorrow at 9:30.
Also, excuse me, madam. Hello. Can you hear me? Madam, we have the sectional after the symposium tomorrow. In the end, at 4 p.m., we have the sectional presidents' addresses also, and
Yes,
followed by
that is given in the program.
Yes, and followed by one invited talk by the chairperson of the INEAS. So we are concluding today's program, and many thanks to everybody, and a special thanks to Madam Anju Sharma ji and our Honorable President Professor G. Padmanabhan sir, and thanks to everybody.
Sir, on behalf of myself and NASI, I also give to thanks to this session, the chairperson and co-chair, Professor Shan Patwa sir and Professor Beena Ravi madam. And also, we are very much thankful to Professor Shelly Awasthi madam and Professor Vijayalakshmi Ravindranath madam. Our heartfelt gratitude and thanks for sharing this. Many, many thanks, madam.
And in the end, I would like to thank our dear colleague, Dr. Santosh Shukla, Dr. Pavitra Tandon, and Archana Pant for coordinating the events today. Thank you very much. And Mr. Deepak.
एक ऑनलाइन फोटो ले लेना प्लीज.
Yes, we are taking it.
Okay.
Actually, the whole program is being recorded, madam.
Right.
Thank you very much. Thank you. Thank you.
Thank you.
Bye-bye.
Good night. Yeah.
Good night, sir.